I'm not going to be talking. You hadn't seen the final agenda? You got it?
Oh, okay. You have
Yeah.
15-minute talk. Yeah.
Even if I just
Good morning. My name is Roger Bingham, and I'm happy to be here. Thank you. This is obviously the Autism Tree Global Neuroscience Conference. We've been doing this for some years now. It's finally picked up a wonderful pace. Last year's, those of you who are watching, I was sitting in my office at UCSD doing it. And one of the things that people have said to me today is, what on earth are you doing wearing a jacket and shoes and things like that? And that's in fact true. The last time I actually was wearing grown-up clothes in this room was February 24th, 2020. And haven't been doing that since. So it's been a remarkable period. It's remarkable to be back in grown-up clothes, and I'm looking forward immensely to doing this meeting. Let me just say very quickly that we're going into the 20th year of the Autism Tree Project Foundation. We'll hear more about that later on, but I just want to say that from my involvement in it, I've felt delighted, honored, overjoyed by being part of this operation. You'll meet Dana Hoff later on. There's many other people involved. Rebecca Barron, who's been helping today, Lisa, who was helping for years before that. So I just want to say that the feeling that one got, and this is particularly relevant after this last 2 years, is when you can go on Twitter and find all sorts of raging going on. The feeling I always have with this gathering, with this group, is one of family. And I just want to thank everybody who's involved for that, in allowing us to do it, allow us to do it this way. I'm going to introduce Alison Motri pretty quickly and get on with it. Normally, those of you who've seen me work before, I'm usually carrying a microphone and rushing around the room, getting people to speak and so on. I can't do that today. And this is not just a fashion statement to match the scarf. Actually is a mask, and I still think that we're in according to the reports I see from people like Eric Topol, we still have some things to worry about. So I also wish you good masking. Alison, you know, I was looking back at the Science Network. I came here, started the Science Network in 2003 with a lot of help from Rusty Gage, who's now the president of the Salk, and Larry Goldsmith here. The first meeting we did was called Stem Cells, Science, Ethics, and Politics at the Crossroads. And things have evolved hugely since then. There was a lot of ethics talks then. Then it drifted away again, but now the ethics stuff is coming back again. So that's really interesting. But one of the people that was involved very early on, who I met a long time ago, was Alison Motri. And you can actually see him presenting at the stem cell meeting on the Mesa, the second edition of it, in 2006. So we've been family for all that length of time. And it's great to see you here again, sir. Alison's been doing amazing research. There's a very lovely profile of him that was done in the Spectrum review, which the interesting point that it made was that what he does was to get an idea and push it, even if it's not quite sure where it's going. He's just very interested in picking the right idea and then following it. And I loved that quote from your childhood, your first deep thought around age 7, when he tried to figure out how a light bulb works. My idea was that the light bulb was not there to send light, but to suck up the darkness, he says. So, you know, we're gathering here today again to suck up some of the darkness, get rid of it and have more light. One of the things, the thing that he's possibly best known for most recently is moving from the stem cell field, pluripotent stem cells and so on, moving to brain organoids, brain in a dish. I'm not going to say any more about this because Alison will deal with it, but it's a topic that has caught off hugely. And in fact, today is November the 4th. Only yesterday in the journal PLOS Biology, there was a paper, SARS-CoV-2 infects human brain organoids, causing cell death and loss of synapses that can be rescued by treatment with Servisphere. This was a paper by Alison. So you had a paper yesterday talking about brain organoids, actually to do with the virus. But you have a better way and a better use for the organoids that you hope will be useful in treatment of autism as well. So let's hear more about that. Alison Motri, thank you.
It's hard to beat Roger's introduction. That's great, Roger. Thank you so much. And it's really nice to be here back in person after the hiatus of the pandemic, and we are all so happy to get our family together in this room. We've been doing this for a while. For those who are new here, welcome. For those who are longtime friends, welcome as well. I mean, and I hope you meet somebody new today. And we'll have like a full agenda. And we like to mix different people. This is one of the, I would say, hallmarks of the ATPF conferences. We put together medical doctors, we put together neuroscientists, we put together families, therapeutics, and everybody who cares about autism. We put together in the same room, and we take opportunity to hear each other and to discuss how we can move forward together. So this is very different from what sometimes I see happening in other places, where there's more like a disconnection between people. Everybody wants to say something, but nobody wants to hear the other. So this is our effort to kind of set an example to the community, and hopefully others will follow as well. And before I start, I would just like to take this opportunity to thank Joe for the very nice, I mean, he always comes with nice graphics for the conference. This one is a really cool one. I'm very proud of that. Because he took advantage of our experiments in his space. We sent brain organoids or stem cell derived brain cells in his space to understand neurological conditions. And so we are always pushing the edge of science, and he captured that really well by providing these images to all of us. So thank you, Joel, for these very nice thoughts of destiny. I love it. All right. So, so the topic of the talk here is autism and gene therapy. And I've been working on ideas for gene therapy for a long time. And I would like to tell you, I mean, what is the status, what we are doing for that. And also take this opportunity to really set the stage on what is gene therapy and why we should care about gene therapy and autism. This is because I feel that sometimes my colleagues working on gene therapy, when they present or when they talk, I think they're often misunderstood, even by the community of autistic individuals. And again, because there is no contextualization of what the scientists are trying to do here. So I decided to divide my talk in different topics. But before I move on, I just would like to say that I'm a faculty here at UCSD. I'm also the director of the stem cells. You are in a place where human stem cells are taking place. The labs are in that building. There are people from my lab here. If you meet one of them, you are welcome to ask them to tour the lab. So you can go and check it out. My lab manager is around. She can organize those things. But also, most importantly, on my CV is that I'm a father of Ivan, and some of you know him. So Ivan is an autistic boy with 16 years old now. And so most of my motivation is actually because of him, because we wake up together, we interact, and I recharge my energies because of him. So if I'm full of energy, it's because I've been around him for so long now. All right. So the way I decided to structure this talk is in 5 steps. The first one is, as I started to say, to contextualize the autism spectrum and exactly what we are trying to do here. And then I want to provide a very clear definition of gene therapy. I still think that there are confusions out there. It is not a new concept for science, but it's becoming kind of a mainstream in society. So make sure that everybody understands. Then I'll introduce the human model. I mean, why we use a human model to study autism. I'll show you the core of the science, which is the proof of principle that a gene therapy approach might work and why we are excited about that. Then I'll talk the next steps, what we are doing and what I think needs to be done as a next step for these to become successful and move forward. All right. All right, let's start with put that in a context. And the reason I'm doing that, and for those who actually watch the videos from the conference last year, might remember that we start with a conversation with a scientist who had his research shut off because of the lack of interaction with the community of autistic individuals in his surroundings. So they didn't understand why he was studying the genetics of autism, and the work was temporarily shut off. And thankfully, I mean, everything was clarified and he's back on track. But we took that opportunity to have him talk and discuss here with our community, I mean, about that experience. So I'm good on our community because we have been doing this for several years. So I think we interact really well with each other. So I'm not too worried. But because we have a hybrid event now, and this is being broadcast worldwide, I'd rather, again, I mean, set the stage for what I'm going to say or talk about gene therapy and autism. And first, by contextualize autism spectrum disorders, this is what most, I would say, neuroscientists and medical doctors will characterize as autism spectrum disorders. You have different levels of autism. High-functioning autism would be what we call level 1, where these individuals will need some assistance. Some are already fully independent, so they don't really need too much more than that. And then you have in the middle, which would be level 2, where they might suffer from some comorbidities or some consequences of autism and might require a little bit more of support. And then finally, we have level 3, where these individuals will definitely need some support, sometimes one-on-one, sometimes 2-to-1, to have that individual functioning. So yeah, it's a large spectrum, and over the years, the spectrum has grown a lot. And I honestly think that in the beginning, I thought that this would be good because we have more representation. Now I think that we need to divide it again, and I think there are discussions about how to do that, but that's not the topic here. So we have to work with these right now. So again, I mean, this is what most medical doctors and scientists will see. What we see in societies is slightly different. Would be something like that. You have perhaps in one extreme of the spectrum, what we call the high functioning would be the Asperger's, and these are often what you see in movies, in TV shows. They're often called smart kids, genius, or they're just different. Some of them are completely independent. Some of them still requires like some help. Most of those are looking for a job, or they already have a job. They just don't function well on that job. And there are a vocal part of that fights for neurodiversity, and that's amazing. It is actually amazing to see how that community has grown up to fight for what they really want. And we've seen here at the TPF, and we help that voice to elevate as well. But on the other side, you have what we are now calling, or some people are advocating to call what would be profound autism. And that's highly different. That's invisible to the mainstream society. So you don't see those kids most of the time because they don't go out. They stay at home. It's very hard to leave the home with someone who is severely affected. So these are sometimes syndromic. They might have like a syndrome that are under the umbrella of autism. They require assistance 24/7. They will die if you leave them alone for 1 hour. So they have no idea what's a job. They have no idea what's diversity. They're not part of any conversation. So, and there are some data saying that these are like 50 and 50% of the whole spectrum. Debatable if those numbers are right, but nonetheless, what's important here is that the extremes are getting very opposed to each other to the point that what one end of the spectrum is fighting for example, the Asperger's, inclusion, diversity, acceptance, they would even disapprove words like patient, disability, intervention, cure. Come on, I don't need that. And again, I mean, it was beautiful to see how this was growing over the years, and the rise of the neurodiversity among autism, something that we've been talking on these conferences for a long time. However, we need to be aware that the profound autism population cannot fight for anything because they cannot even talk. So most of those kids are nonverbal. They cannot, they have like severe neurodevelopmental disorder. So most of the fights are actually done by the families or the parents who are, again, I mean, supporting them 24/7. So the time that they have to fight for something is really minor. So it's a very sensitive population that it's perhaps the most vulnerable among the spectrum are the ones with profound autism. And of course, I mean, these are actually patients. They do have these abilities. They need intervention, and their families are actually hoping for a cure. So there is nothing wrong about what the two extremes want to fight for, the two extremes want for. We just need to understand that they are actually two extremes of the same spectrum, something that over the years, it seems that we are losing the sense of what are the needs for those two, which, to be honest, they are not mutually exclusive, because the profound autism will like to have inclusion, diversity, and acceptance at the same time. So not mutually exclusive, so we should not put them against each other, but we should converge them, I mean, making them work together, which would be even better. So what I want to point it out is that for the topic of what I'm proposing here, which is gene therapy, I'm not talking about Asperger's, I'm not talking about high-functioning autism. I'm talking about profound autism, people that will die in 1 hour without supervision. So these are the ones that I'm talking about. So gene therapy, it's for those. So if you are not in that part of the spectrum, you don't need to worry about that, because we're not studying you, we're not providing any therapy for you. We are doing for the other ones. So that's the contextualization. So let's move on to what is then gene therapy, why we are doing that. So this is my definition of gene therapy: a way to overcome the consequences of a genetic mutation in a cell, hopefully restoring the proper function of the cell, tissue, and or behavior. Basically, we're trying to correct something that you are born with, some genetic mutations, in hoping to restore the proper function of the cell, tissue, or the brain. So how does gene therapy work? So for those who are not scientists, you might ask, I mean, how do you do that? I mean, you have about 86 billions of neurons in your brain that controls all your emotions, your behaviors, the way you talk, the way you present yourself. These are all orchestrated by 86 billions of neurons, and about the same number of non-neuronal cells that helps the brain cells to orchestrate as well. So how do we fix a mutation in each one of these? So that's not an easy task. And this is how gene therapy works. So virtually what scientists can do is to pack the right version of the gene or enzymes that can fix the mutation of a specific gene inside a virus. And we use virus because viruses have a good way, they're very efficient on penetrating human cells. I mean, after the pandemic, we all know that this is true now. And we use a virus that does not cause any harm to the person, and it's called AAV, an adeno-associated virus. So this is like a very mild flu virus. So we remove all the genome of that virus and we place these enzymes or the correct version of the gene inside that virus. And then we use the virus to deliver the correct gene or the enzymes that will fix the mutation back into the person. Those viruses would migrate to the nervous system and will deliver the correct copy of the gene to your brain cells, right? So in concept, that's amazing. That's perfect. But practically, we still have like a long way to go, because there are several problems with this idea. Your body might react to the virus. Your body might react to the correct copy of the gene, because remember, if you have a mutation, maybe your cells, your immune cells, never seen the correct version of the gene, so they might fight it back. And the delivery part is the most problematic. The virus does not fully penetrate the human brain. It would infect only a few cells. Is not 100% correction. And sometimes there are doses problems. Some genes cannot be, cannot have like too much of the gene in there. You cannot have too low of the gene in there. So you have to control all of those aspects. So there's still like a fine-tuning problem with this technology before it becomes like something that you can move on into clinics. Although there are several clinical trials and protocols already moving on. I mean, if you go to clinicaltrials.gov and just place in a search like gene therapy, you're going to see hundreds of gene therapy clinical trials going on. So this is definitely moving to mainstream. It's not perfect, but it's the best what we can do right now. And so, I mean, it's a good tool. All right. Okay. So yeah, why we're not doing that for autism? We should be doing this for profound autism or the ones that we know the genetic mutation right away. Well, there's a problem that scientists have when we are trying to study autism, which is the lack of a model. And especially if you take like syndromic autism, profound autism, these are mutations in genes that affect your neurodevelopment. And meaning that you are actually born with autism. You don't acquire autism through your life. So your autism actually starts in utero. So how can we actually study the brain of autistic individuals when they are forming in uterus without use of non-invasive technology? So we cannot do that. Instead, what scientists have been relied upon was in different models, such as postmortem tissues. I mean, people with autism eventually die. Some families donate the brains to research, and we have a couple of those, so we can study. But the person is already dead. So the tissue is not alive, and the damage or the alterations is already formed. I mean, we don't capture what happens before, during the formation of the brain. Another way to study autism is to collect blood from people. But blood cells are not brain cells. Blood cells does not make connections, does not have synapses, does not influences your behavior. So there is so much you can do with blood. And most of the genetics of autism was done in blood. So it's still a very useful model, but doesn't get to the brain. And then finally, we have animal models. Animal models are tricky because you can create mutations similar to the ones that you see in patients in a mouse model. And the mouse model might seem autistic, but sometimes it's tricky because the mouse brain and the human brain, they evolve in different ways. What a mouse might be doing that seems like autistic, for example, having like a repetitive behavior, might have nothing to do with the circuitry or the brain cells that actually do the same thing in humans. So most of the times, especially when we are moving into treatment, mouse models have failed, miserable, especially for the brain. So you can see as a scientist, we don't have too much options here. And that's why I decided to bring, or me and others in this field, to take advantage of stem cells, human stem cells, because with human stem cells, I can actually make brain stem cells. Stem cells are the ones that can differentiate or specialize in different brain tissues, including brain cells. And it was in 2008 when a researcher called Yoshiki Sasai from Japan showed us that if you use a specific combination of factors, you can guide these cells to form brain tissues in 3 dimensions. And this was the first publication. 2008 was when I was starting my lab, and I saw that. I said, oh my gosh, that's the model I need for autism. So I started following his path and improving protocols that now we have way more robust than what he previously did. And so we could derive stem cells from autistic individuals and controls and compare them. So I can compare directly their brain cells in a dish, in a controlled situation of the lab. So there are different ways of doing that. These are the different protocols that we have for the stem cells. You can create brain organoids that have multiple brain regions, or you can guide them to specific brain regions if you know where are the brain regions. For example, in autism, we focus a lot of attention in the prefrontal cortex because we know it's a part that is affected in autism, and it's a part related to human cognition, and lots of processing of information happens in the cortex. So we now have tools to guide the stem cells to become cortex, a brain organoid, cortical brain organoid with the right proper cells that we want to study in the lab in a dish. So before I move on, let me just say that if you search now this technology in the media, you're going to end up with names like, oh, they created a mini brain in a dish. So you give the impression that you have like a fully formed brain in a petri dish, which is not true. I'll show you how they look like in a moment. But this is enough for me to highlight the limitations of this technology that I want you to keep in mind. Because we are starting from the stem cells, these neurons are still very mature. They are not vascularized. We don't have vases passing through it. So that's a problem. We might not have all the cell types because as an organoid, it means like a small size tissue. So it's a tiny thing. It's not the size of the brain. And we still have problems with reproducibility and translatability of this technology. So lots of things that we need to overcome. And I think most of them, to be honest, we're going to overcome in the next 3 to 5 years. For example, vascularization. I think in the next 3 years, we're going to have a brain organoid that is vascularized. So we can grow them bigger. We can add other pieces of the brain there. All right. So this is our protocol. We have a very nice recipe to make brain organoids. We start with pluripotent stem cells. We neuralize them. We induce their proliferation so they can grow bigger. And then we end up with spheres that are about 0.5 centimeter, and they contain about 5 million neurons in there. This is about the size of a bee brain. And a bee can do lots of things. So you can say that even though it's a very reductionist approach, we have something that might be useful. And this is a postdoc in the lab, and she's holding a plate which contains about 3,000 organoids. Each one of these white dot is one brain organoid with around 5 million cells in there. So we can use this to perform several experiments, and I'll highlight some of these experiments for you in a minute. But just to say that the anatomy is really good. It resembles something that is similar to a fetal human brain. There is a ventricular zone. You have progenitor cells surrounding the ventricle. These progenitor cells migrate and form the cortical plate, which is how the human brain is formed in utero. So we recapitulate all these stages inside the brain. Some people ask me what kind of cells you have. Well, it really depends on what stage you're looking at. I start with single cells. I end up with 5 million cells because the cells replicate. This is a snapshot at 4 months. You have the pool of progenitor cells on top. You have most of neurons being formed. These are excitatory neurons. This is one of the most affected brain type in autism. You have your astrocytes being formed at that stage, and some of the inhibitory neurons start to emerge as well. So as we age the organoids, the diversity of cell populations will actually change as well, making the model closer and closer to the human brain. That's why we like it, because we can recapitulate very nicely all these stages. And I'll show you one example where this technology was very powerful. And it was another virus outbreak, but this time happens in 2015, 2016, back in Brazil. You might remember the cases of macrocephalic babies that were born in the northeast of the country. So at the time, there was no link between the virus and the defects in the brains of the babies. So to prove causation, what was done was to actually expose the Zika virus to the brain organoid, and we showed that the Zika virus could actually kill the progenitor cells. And so during the migration, they would fail, and you have like a very small cortical thickness, which was mimicking exactly what was happening in uterus in those babies. So most importantly than just proving the causation, just 2 years after the outbreak, we're able to use that tool to screen for antiviral drugs that could block the viral replication. And we find one that's called fosbuvir that was initially developed by hepatitis. And so fosbuvir can even actually block what we call vertical transmission from mom to the fetus. If you have a mom that is positive for the virus and takes the drug, the fetus would born without the virus. So this is from causation to treatment in 2 years, which shows that if you have the right tool, you can actually really advance sciences. And this was something that was missing in the hands of the neuroscientists, a really good human model. But autism is not about brain malformations. Actually, very small percentage of autistic individuals have alterations in their brains. Most of the alterations that we see is about networks. And we always wanted to use the organoids to actually measure the networks that was formed during these stages. And the way we do that is by placing the organoids on top of a multi-electrode arrays. So these are electrodes that are just capturing the electrical activity of the organoid, of the tissue over time. So we can measure that over time and see how it looks like. Scientists were completely skeptical that this would work, and I'll tell you why. Most of the tissues that we had, or the cultures that we had in the past, they were always below what we call the 5 Hertz threshold. This is overall brain activity. So you can see 2D mouse, 2D human cells or mouse neurons, they are always below the 5 Hertz threshold. And a primate brain fires at about 20 Hertz. The mouse brain fires on 18 Hertz. So we never could get there. Even if you wait longer, someone waited for 32 weeks, couldn't cross the 5 Hertz threshold. So with our protocol, this was the big surprise of the lab. We see that if you wait for 40 weeks or about 9 months of age, which is similar to the human gestation, you actually achieve that 40 Hertz with the organoids that we were able to produce in the lab. So that was a big news, and now we can actually study or use that as a model for conditions that affects neurodevelopment, such as neurodevelopment of the network, such as autism. Most importantly, they do that in a very similar way to the human brain. We did that by looking at EEGs, which is the same thing. These are electrodes that you place in your skull, and you can capture the different oscillations. The reason why this was important is that because labs like mine, we are too focused on the basic biology. We like to study genes, we like to study synapses, cells, but it's hard to bridge human cognition. So we'll never be able to do that. But this is something that cognitive neurosciences has been doing for a long time. They place the EEG on people's head, and they correlate the different oscillations that they can get with different human behaviors, including autism. So if you have autism, if you have seizures, your EEGs would be different. And that's what we want to do with the brain organoids. Can we bridge that? And again, I mean, before we start doing that, the only thing that we could do is to look for individual neurons. And I mean, individual neurons will not move you too far. And here's the reason. This is the analogy of a stadium. If I just look for one person, I don't understand what's going on in the entire stadium. Some people are quiet and then they become active. Some neurons are quiet and then they become active. That's cool, but what does it mean? So it means that someone scored a goal, or it means that there is a ola going on in the stadium, right? I only know that if I capture all the electrical activity at the same time, and that's what we're trying to do with these multi-electrode arrays. This is too complex, but just to say that there is a way to do that, and we team up with a colleague of mine here at UCSD, Brad Wojtek, for this study. And long story short, I mean, together with him, we showed that the organoids start to show these oscillations when they are about 4 months of age. By 6 months, they are very stereotyped. And by 8 months, they become highly complex. The activity of the organoids correlates really well with the diversification of cell types that you have in there. All right. So we also showed that this is similar to the human neurodevelopment by creating a machine learning algorithm where we train the machine using the EEGs of the preterm baby brains, because we cannot do that in uterus. And then we feed the organoid with, we feed the machine with the data from the organoid. And what we see is a very nice correlation between the trajectory of the human EEG compared to the network of the brain organoid, which again suggests or confirms that the model we have is good enough for us to mimic the oranges or the alterations that we see in uterus, including from people who has mutations in those neurodevelopmental genes, which includes the population of syndromic profound autism. All right, having that model allow us to move forward and now try if the gene therapy approach will actually function in the way that we would expect. We don't want to do that before testing in the lab. I mean, that might have like some side effect, might not work, might not be safe. So the first thing that we want to do is do no harm. So we want to make sure that it's working properly as we had expected before we enroll our kids in this kind of clinical trials. So the first thing that we did was, okay, let's find a cohort of individuals or patients that are very similar to each other, that all have the same mutation, so we can test it. And we started with our friends at the Pitt Hopkins Foundation, and we collected fibroblasts from different individuals carrying mutations in a gene called TCF4. And you can know the gene is mutation by doing genome sequencing. So they all have genome sequencing. They all figured out that they have alterations in TCF4. So they are now classified as Pitt Hopkins kids. And we reprogram these cells and we start the work. So this was all performed with Fabio Papes, who was a visiting scholar in the lab, and he was the one who led the study. So Fabio collected the cells, reprogrammed the cells. These are some of the characteristics that we keep in mind when we try to model the conditions, try to see if we see something similar in our brain organoids. Of course, I mean, there are situations that requires other cell types that we don't have in organoid. For example, constipation. We cannot model yet constipation in a dish. We might be able to do that in the future. So we were looking for more of the intellectual disability alterations in the networks, something like that. And so Fabio was able to create these brain organoids. And one thing that he immediately saw it is that the size of the organoids compared to the controls here are the parents of those kids, they are smaller. We can see that they do not grow as you would expect for neurotypical individuals. So the size is about 50% reduced compared to controls, just looking for the diameter. And the expression of TCF4 is also reduced in the patients, as we would expect, because they have mutations, so they're not expressing the full level of TCF4 in there. Also intriguing is that we do see some malformations of the organization of these neurons inside these organoids. For example, the organoids, the normal organoids or the control organoids, they will form rosettes. These are early stages of neurotube formation. You can see those rosettes or these morphologies on the parental organoids, but we barely see that in the Pitt Hopkins derived organoids. So once we have, and we quantify, for example, SOX2 is a cell type that proliferates in the brain. So you can quantify that and you show that there is a reduction in the number of SOX2 proliferating cells in these organoids, explaining why they are so tiny, they are so small. All right. So what else can we find out using these organoids? Well, we can look around all the genes that TCF4 might be regulating. TCF4 is a master regulatory gene that controls the expression of downstream genes. We do that by doing like a gene expression profiling. For those who are not neuroscientists, this is just a way to analyze thousands of genes at the same time. And the different colors means that we have more of these genes or less of these genes compared to controls. And by looking at these genes, we can guess what kind of a molecular pathways are affected as well. And we are able to create insights about how this gene is actually reducing the number of SOX2 positive cells inside these organoids. And we learned that some of these progenitor cells are actually controlling the identity of cortical neurons that would appear later, a little bit later in life. And these cortical neurons are important for the proper network to be formed. And one of the markers for that is called CTP2. So this is a specific cortical neuron. And you see, again, because we don't have the right proliferation, you end up with a cortical population that is lower on these CTP2 neurons. These neurons are involved on the microcircuitry of the cortex, but also between the communication of the two hemispheres, between the communication of the cortex and other cortical regions. So it's a very important population that might explain several of the clinical symptoms of these patients. So it's very important that we are recapitulating that in a dish. We also confirm these findings in postmortem tissue. We were able to perform the same analysis in one of the postmortem tissues that we had. And we validate that not only the CTP2 population was reduced, but also several of the genes that we were expected to be affected by the lack of TCF4, they were affected as well as in the postmortem tissue. So that give us like a confidence that what we have is really something that mimics the TCF4 deficiency, or as we call haploinsufficiency, because one copy is functioning, the other one is not in the brain cells of those individuals. All right, so we have a model, the model is working, the model now, he recapitulates some of the key parameters that we think is important for TCF4, what we can do. So one strategy, and you probably hear about the CRISPR enzymes. So these are enzymes that you can use to correct genetic mutations, but you can also use these enzymes to stimulate the expression of genes. And I told you that those individuals have one of the normal allele, the normal copy of the gene is still functioning. So what Fabio decided to do was, okay, let's use CRISPR to activate or to overproduce the right copy of the gene. So maybe by doing that, you have more protein inside the cells and we can rescue that. So he designed the strategy. It requires like some specific guided RNA to the specific regions of the TCF4, and he tested in the protocol of brain organoids. And to our biggest surprise, if you add the CRISPR early on in the development of the brain organoids, you no longer see the macrocephalic phenotype. You basically rescue the population. You rescue the expression of TCF4, but as well as some of the downstream genes. But one of the most, I would say, surprising findings that he shows, it is that something that we're not expecting to be rescued, which was some of the morphological alterations that he saw in these organoids. For example, here I'm highlighting in magenta, there are some of the what we call cortical scars. So these are neurons that are just not formed properly in the cortex. But upon the treatment with the CRISPR enzymes, he was able to see that this was gone. So he no longer has these structural defects in these organoids. So these, again, all very good news. The only bad news here is that the CRISPRs are not yet ready for clinical trial. These enzymes are still under development. They create what we call off-targets. So they might activate other genes than the ones that we want. So it's not quite ready yet. And then we thought, what about if we create a second strategy where we just put the correct gene inside the cells without trying to regulate the endogenous one? So it was a little bit tricky to do that because the expression of TCF4 needs to be regulated in the precise amount inside the cell. So he created a very nice strategy that I'll spare the details now, But basically, by manipulating the repeats of these micro E5 box upstream to the promoter region, he was able to get the right amount of TCF4 inside the cells. And then he shows that the virus, he built the virus carrying that gene, and shows that the virus was enough to restore the expression in the brain cells and all the downstream genes that regulates TCF4. More importantly, he then used the virus to infect brain organoids already formed from Pitt Hopkins patients, and he started to see back all the alterations that I previously described. First of all, he restored the expression of TCF4 inside the neurons. He was able to restore the structural defects caused by the lack of SOX2 positive cells in there, and the restoration of the CTP2 cortical neuronal population responsible for all this cortical communication. So that was great news. And then we said, all right, what about the function analysis? So he placed the organoids on top of these multi-electrode arrays, and he recorded over time. This is the behavior of the networks of the controls in the first graphics. And in dark blue is the patients, and in light blue is the patients after the viral correction. And you see that we're able to restore some of this network activity. Again, very nicely done. And so I'm glad that this program was picked up by Mazi, which is a company that does clinical trials. And these vectors are now being tested in monkeys for toxicity. Once we clear the toxicity, this would be moved into clinical trials. So we're not expecting any side effects, but the FDA will require that we test those vectors in primates' brains just to avoid any surprise when you're doing the clinical trial. So this is a proof of principle that you can actually correct the gene inside brain organoids and rescue not only the cellular defects, the molecular defects, but also the network defects, giving us confidence that this might actually work in vivo as well in the patient's brain. So we, in my lab, we're expanding these ideas to other genes. This is another program that we have for CDKL5 deficient syndrome. If you have mutations in CDKL5, you have a very early onset of seizures. When you are like a newborn with 3 to 4 months of age, you start having seizures. So that's dramatically affect your neurodevelopment. And this is due to the hyperexcitability of these networks. The bar in the middle shows how hyperexcitable is the networks compared to the black bar, which is the control in the organoids. Now, if we just replace the gene back into the organoids, you see how can we reduce this hyperexcitability, most likely eliminating the susceptibility to seizures. So this is another program that is moving on for a potential clinical trial. And we think that the earlier we act, the better for these patients. And finally, we have another one for Rett syndrome. This is a gene called MECP2, is a epigenetic gene. We showed that we showed that there are many defects associated with alterations in brain organoids that carry that mutation. Most importantly, if you place the organoids from Rett syndrome in these multi-electrode arrays, they will show a deficiency as well. And now if you add the virus, the little bit of green that you have in that organoid in there is the small number of cells that we are able to infect with the AAV. And this is a nice curve showing that as you increase the dose of the virus, you restore the electrical activity of the population. And what I think it's quite important, and we still don't know yet how that works, is that the number of infected cells here is less than 5%. I mean, we are getting like a very low infection, but that's enough to correct the entire network. So that gives me hope that the brain is so plastic that even with just 5% of functional cells, you are able to overcome all the genetic defects. So genetic is not deterministic if you have the proper environment, for the neurons to thrive. So that's quite important. So there are many other alterations related to autism. This is just like a sample of the conditions or the genetic alterations that we're doing in the lab. Most of these conditions, we don't have even animal models to do that. So we generate brain organoids. We test for therapeutics. We test if we can find drugs. We test if we can find a gene therapy approach. There are now antisense oligonucleotides, ASOs. You might hear about that. So that's another possibility. And the goal is really to show proof of principle and accelerate, speed up all the clinical trials. And I'm glad to say that over the years, the FDA is being more and more sympathetic to this model, understanding the needs to really push this type of therapies further on. All right. So proof of principle, we have it in our hands in different stages of development now. What are the next steps? There is one major problem with the gene therapy, especially in autism, especially for the syndromic profound autism. It is that not everybody does genome sequencing. This is something that is not like mainstream. Most of the doctors don't even prescribe that because they don't even understand the value of that. To be honest, I mean, even if you have the gene right now, there is not much you can do, but you can start already personalizing the treatment. Even if you cannot do that, I mean, you might be able to participate in these future clinical trials. And the problem that we are having is to recruit enough patients for these clinical trials because we don't know where they are. I mean, we might have good foundations, such as the Rett syndrome, the CDKL5, the Pitt-Hopkins, that help us to map all the patients in the world. But what about all the other 100 genes related to autism? We still don't have foundations being formed. We still don't know how many of those are affected. We don't know where they are. But the good news is that genome sequencing is getting cheaper. People are having access to that. There are more conscious about the value of that, so people are doing it. So what we decided to do is to create an app called Autism Me. On that app, you just plug your information and you add the genetic alteration that you find in your report. And researchers like me or doctors or even family members, they can search by the characteristics, by the gene mutations, or by the profile, and be able to connect to each other. So we initially thought about doing that for the science, but we realized that there is way more benefits on creating the first social media for autism. Actually, that's what it becomes, because there are several stakeholders that might take advantage of a social network like that. So we are seeing like families connecting and exchanging information among them. Some have kids that are older, some had kids that are younger, and being able to connect with the families, what they try, what kind of drugs it work, what kind of treatments they're doing, how they're progressing in life. It's really value for other families. So that's a plus. But also, I mean, all doctors are now finding ways to find other families to point or to ask questions about all these specific genetic alterations, because honestly, we don't have natural history for most of them. So we don't know how they progress, what are the clinical symptoms associated with specific mutations. So that's the value of that. And we created in a way that you can opt in for research. So it's not automatic. You actually see a value on that, and you decided to participate, and the data is yours and it's fully protected. So we are running like a pilot experiment in Brazil with my colleagues over there. I think we have about 5,000 people already connected and using the network. The next phase is really the expansion to other countries. And we really hope that this tool will facilitate not only the research, but the interaction among families, among therapeutics, among medical doctors. So we'll see how this goes. I mean, the network would be shaped by us. So we create the network, we can shape it, and we can evolve the way we want. So it's really something that we should take advantage of. All right. So I'm stopping soon just to take comments and questions, but these are the major take-home messages that I hope you keep in mind. Profound autistic individuals are likely the most vulnerable population and might benefit from gene therapies. Again, I mean, in the context of the whole spectrum, this is not for everyone. This is for the ones who actually need it. So the science is working towards that, towards improve the conditions, improve of the symptoms. We might not be able to get to a full cure, but we might improve some of the symptoms. We might eliminate some of the comorbidities with a gene therapy approach. So it acts on the root of the problem. So human brain organoids can be derived from autistic individuals, and they function as a gene therapy approach or as a proof of principle, and might be useful tools for preclinical assays. So we can already test the vector toxicity, some of those fundamental issues that the FDA might get back to us before we move into proper clinical trial. And then finding these individuals with profound autism, with specific mutations in the same genes, it is and continues to be a major challenge. We have some programs for specific genes, and when we ask the help for some of the pharmaceutics, they are refusing because the patients are not organized. They don't know how many patients are there. They don't have a foundation. They don't have natural history. So it's sitting in my lab and is not moving forward because we don't have that. So we hope that a network like TismoMe might actually help to speed up all these challenges that we have right now. All right. I would like to thank all the members of my lab. Full disclosure, I'm a co-founder of Tismo. Tismo is a genetic company that actually founded TismoMe. And these are members of my lab. I highlight mostly the work from Fabio here, but there are so many people working on these brain organoids right now. And we are in debt to all the families that actually contribute to our research, that donates their time, that comes to visit the lab, donate their fibroblasts, and actually help us to shape the way we are moving with our science here. So thank you very much. I'll be happy to take questions and comments. I guess Roger would moderate that. I think we need to speak on the mic so people that are online can actually hear you.
Does this work? You can hear me. Okay. Can I ask a couple of questions to begin with? First of all, this has all happened really in sort of 20 years. I mean, the stem cells, CRISPR technologies, and so on and so forth. So for the first time, we're now in a situation where autism researchers are actually having to deal with these issues, and you dealt with it at the beginning. I don't like to be negative, but let's really hone in on this and get it done. I've got several papers here. Why autism eugenics is everybody's business. Eugenics today, autism enableists, and so on. So the same sorts of things that are coming up, certainly on Twitter all the time, about sex, trans, and racism. Autism joins those things because of the whole notion of eugenics.
Yeah.
Could you just do a little extra on that, just to clarify what's going on?
I think there is a confusion about eugenics and gene therapy. First of all, these are completely different concepts. And we scientists laugh about it, but people don't know that. That's why we need to be very clear. I mean, eugenics is something that scientists are not doing it. I mean, nobody's doing it, to be honest. This is selecting a specific race, population, or genetic advantage in the embryos as you selected the population. So that's illegal, to be honest. That's immoral. So nobody's doing that. So even the word eugenics doesn't go along with science, because science is actually debunk eugenics. So we should not talk about that. So if you have any misconception, just Google the definitions. But I presented the definition of a gene therapy. So the use is is for a treatment, is to help and not to unselect anything. Well, yeah, I think that's it. I mean, is that what you want to hear?
Yeah, I mean, incidentally, the other comment that was, now you will see why it also said in this piece about Alison that I was quoting from before, bold plans, while others debate the merits of his scientific ambitions, Alison Motri likes to be out on the edge. And it's very wonderful to see how far you are doing this. Let me just do another thing, because when you got to the end there talking about the equation, there's an author called Yuval Noah Harari, pretty well known for *Sapiens* and other books, and Noah has a what he calls a danger equation, which is that B times C times D equals AHH, which means biological knowledge times computing power times data equals ability to hack humans. Are you aware of this?
Oh yeah. So all the technology that I described to you guys right now, the creation of human brain organoids, the use of gene therapy, the use of CRISPR enzymes, all this technology can be used for good or for bad. And we actually seen that over and over during history and during biology.
Yeah.
So they are not, let's say, neutral regarding the ethical discussions. So right now, for example, there are some people worry about how far we are taking the brain organoids because they're getting super close to the human brain. And if you are to create a network of neurons or brain cells, they might do what the brain does, which is to become self-aware and conscious. So then what happens? I mean, are they part of the person that they were derived from? Do they deserve a specific right, like a newborn? So these are questions that we have to kind of create at the same time. And sometimes scientists put together a set of rules or even a moratorium on what we think is doable with the technology at the time. For example, CRISPRs in babies, there's a moratorium saying that we should not do it for now because the tools are not, we don't fully understand how the tools work.
Right.
And brain organoids might reach the same level. We might have to stop the technology at a certain point or not if we decided to. So that's why the dialogue with all these communities is important. I keep saying that we should develop these technologies for the good and for the most vulnerable ones. But there is always a risk that someone might take this technology to improve brain function, to augment the human brain, or God knows. I mean, people are creative, but
the debate is good. I mean,
the debate is good, and this is true for all the technology. I remember, for example, blood transfusion, organ transplantations. We passed to that, so we are used to that. And as soon as people start to see the good on it, I think the humanity embrace it. And I think that's the same what's going to happen with CRISPR, brain organoids, and gene therapy.
Some questions came in. One from Claudia Villarreal. Will this therapy help a teenager 16 years old who suffered from severe hypoxia at birth, as a result at present time has severe speech delay, intellectual disability and autism? ? Genetical study was done. No other reason for his delay was found.
Yeah, so there is several conditions where, first of all, it might not be caused by a genetic alteration.
Right.
And the question is, can we do something about it? Well, I mean, the traditional gene therapy might not work, but again, we might hack some other genes that facilitate the plasticity of the brain to help those that have hypoxia early in life. What I'm learning is that the human brain is way more plastic than we thought before. There are people walking around with just half of the brain and we don't even know because we don't study them, because they don't go to the doctor because they're not complaining. But the brain can do things that we are very surprised. So I trust the plasticity of the human brain, and I think we're going to find how to modulate such a plasticity even later in life. So I truly believe that that's possible.
There's another question. An anonymous attendee, going back to the beginning of your presentation and from your perspective, is the activism from highly functional Asperger's autistic individuals hurting the profound autistic individuals? By the way, that word that you used up there, profound, which is obviously very, well known to this community and so on. I mean, I was these words are turning up in articles now.
Yeah.
First of all, what do we mean by profound autism?
Yeah, I think it is a tentative or a way to really characterize that population that is most vulnerable. Going back to your first question, and by the way, not everybody agrees on the term profound autism. I think scientists are slowly embracing it because it helps to describe. When I say profound, everybody gets, all right, we know what you're talking about, are the most vulnerable ones. But this is true with all the autism terminology. It doesn't make 100% of the people happy. So some people don't like this term. I think the community is slowly embracing it. We'll see. And if the activism of the high functioning autism is hurting the ones on the profound autism, and the answer is yes, unfortunately. And that's why I think it's important to contextualize. We already had instances where scientists had been attacked in conferences or bashing social media because of such terms. And one example is the use of patient as a terminology. These are patient. And then, I mean, if you say that in social media, oh my gosh, people will go after you. And I think it's just, it's either lack of knowledge or lack of contextualization, because I don't think this population, the high functioning, are lack empathy. I think maybe they are just not aware of what's going on. So I think, again, I mean, that's why I encourage all the scientists to contextualize their research and explaining exactly what you're doing. Because once you explain, I mean, and we try with this community, in ATPF, we have several high-functioning autistic individuals, and we talk equally. They fully understand what's going on. I mean, they're not against a treatment for the profound autism, but because we have been working with them for a long time. This is not true in other parts of the country, in other parts of the world, but we need to catch up.
Yeah. Just for anybody who didn't know, or anybody outside this community who's watching, there was a piece called "It's Time to Embrace Profound Autism" by Alison Singer, as recently as the 27 October 2022 issue of Spectrum News. So that's probably worth a look for those who need a better explanation of it. Last question just came in from Michelangelo. Huh.
So must be a good one.
Great guests. The brain is a complex system, in quotes. Dynamics at neuron or network of neurons, micro meso scale, does not correspond to dynamic of brain network macro scale. You are studying networks of neurons in MEA generated by iPSC. It's all initials, isn't it, in capital letters, and how they change their dynamics using drugs or gene therapy. How do you deem to bridge the gap between what you see happening in the network of neurons of your petri dish and the brain dynamics? I mean, how do you think to understand if that drug gene therapy is actually working on the whole brain dynamics, which is what actually cares to us?
That's a great question. The model we have is very reductionist. It doesn't is not yet mimicking the entire brain. It's not a mini brain. It's a brain organoid. So that's part of the confusion that we see in lay terms. So and it's true, we're not quite there yet. So we don't know if everything that we see in a dish actually represents what we're going to see in the more adult brain, or when we do the real clinical trial. We will know as the clinical trial progresses. For example, if we now, let's suppose that everything goes well and we have a clinical trial for Pitt Hopkins, and that goes into the patient's brain, the virus goes there, restore the expression of TCF4 in a reasonable number of neurons. Do we see any improvement on the condition? So if you don't see any, it's a negative data. So it means that what I have in my lab, in my dish, is not predictable of what's going to happen in the clinical trial. So that would be a negative result. So we are prepared for both positive and negative results. So it means that we have to go back and revisit our model, or maybe improving our model. And that's why I continue to improve the model to make that organoid getting closer and closer to the human brain by adding vascularization, by growing them bigger, by adding more cell types. So the more I move towards a better model, the more predictable the organoid or the model will be. So that's where we are.
All right. We're going to have to move on now.
Should we take questions from the audience?
We've been taking questions from.
Oh, yeah? Oh,
they've
they're
they've
online?
been coming
All right,
in.
okay.
I mean, does anybody want to raise their hand? So, no.
Hi, my name is Kyle Cabrera. I'm a student at UCSD. I wanted to ask, in going beyond organoids, as far as what you believe the next steps are for this gene therapy treatment, do you personally believe that the cell type, the cell species matters in terms of different regions of the brain? And I ask because in learning about genetics thus far, in my major, I have learned that there is evidence suggesting that certain regions of the brain develop slightly differently in utero for people that have autism spectrum disorders, the cuneus, for instance, and that may not necessarily be the case for other species of cells in different regions. Do you feel like the focal targeting of these specific areas and the development of these areas is going to be important going forward?
Yeah, absolutely. And that's why I think we need a better representation of all the cell types in the organoids that we currently don't have at all. But we have the major one, so it's very reductionist, but I think it's a good starting point. And I do think that the species matter. For example, the most used animal model in biomedical research is the mouse. And you have a fully formed mouse brain 20 days. So the human brain takes like 9 months and is still immature. So the species and the cell type and the background, the genetic background do matters. And I think that's why we are failing miserable on developing drugs for the brain, because we never had like a good model system. So I have high hopes for the tiny organoids. Yeah.
Okay. Doris, you ready to go? Oh, you're there, right. Was that a hand wave or? Yeah. One quick. This is the last question.
So as a parent of an autistic child, I believe some of the issues came in utero, and there's no history of autism on either side of the family that we can tell for generations. So I'm wondering, so I understand you're coming from the gene aspect of it. What if it's environmental aspect? Or how do you address that versus the genes?
Yeah. So first of all, by not having a history of autism in the family doesn't mean that it's not genetic, because there are mutations that acquire during development. The person, we call them private mutations or de novo mutations. So it's still worth investigating if they don't have like private mutations. But nonetheless, there are certain types of autism or conditions that resemble autism that are caused by environmental factors, and we know a few of them. It's still, I mean, gene therapy might not work because there is no gene involved, but we in the lab are investigating potential drug treatments. So there are maybe some drugs that might actually help the networks to reestablish, help the formation of synapses. One of the first work that we had for Rett syndrome was not on gene therapy, it was on finding a drug, and that was IGF-1. And in our tissue culture, IGF-1 works beautifully. So we restore all the synapses that were missing in Rett syndrome. The problem with the clinical trials was that IGF-1, insulin growth factor 1, was a long molecule, was not penetrating the brain. So it failed until a company called Neuroscience create like a short version of that molecule that now penetrates much better in the brain. And I think it's probably the first drug to be approved for Rett syndrome. So there are hope for drug treatment as well.
Round of applause for Alan. Thank you.
Thank you.
Did you all get one of these when you came in? Because these, as usual, are extraordinary books full of wonderful information and so on. You can read more about Doris Trauner in here. As it says, Distinguished Professor Emerita of Neurosciences, Research Professor at UCSD School of Medicine, attending physician at Rady Children's Hospital. But the kind of thing that we're really interested in at the moment is these kind of articles that have come out. The UC San Diego team sets out to study the effects of cannabis component CBD on severe autism in children. Cannabis? Children? Autism? What is this? Doris will explain.
Thank you and good morning. Is that good? Can everybody hear me? I'm going to talk this morning about a problem that Dr. Motri alluded to very nicely, and that is the problem of severe autism. And I'm sorry, I'm having trouble figuring this out. Severe autism and behaviors associated with it. And there are a lot of behaviors that are problematic that are associated with in particular with children with severe autism. And those include aggressive and self-injurious behaviors, severe repetitive and stereotype behaviors, and severe hyperactivity, as well as some other problems that are common in autism across the spectrum, sleep disturbances, anxiety, and things like that. But some of the problems that children have with aggression and self-injurious behaviors and extreme hyperactivity and repetitive behaviors are so severe that they interfere with their lives. They interfere with all aspects of their lives, including home life, social life, academic life. And there are very few treatment options for those kinds of behaviors. The mainstay of treatment is behavioral therapy, which certainly helps to an extent, educational intervention, and then medications. And there are only a couple of medications that are approved for the treatment of these behaviors. And they do not work with every child, and they often have somewhat unpleasant side effects. So people have in recent years been very interested in the role of cannabinoids in the treatment of some of these behaviors. And cannabidiol, CBD, is the one that has really been focused on the most, although there are other cannabinoids that have been and are being looked at as well. Phytocannabinol. Okay, I'm sorry, I'm looking at the wrong thing here. Phytocannabinoids are cannabinoids. They're substances that come from the cannabis plant. And the most well-known is THC, delta-9 THC, or cannabis. And this is the main psychoactive cannabinoid. And cannabidiol, which is non-impairing cannabinoid. It's we used to say not psychoactive, but it does have psychoactive properties. They're just beneficial ones, at least people think. So they're non-impairing cannabinoids. And cannabidiol makes up about 40% of the cannabis plant. So it's present in good quantities. And we don't know as much about these drugs as would be nice to know because of legal restrictions until relatively recently. But the mechanisms for CBD are really multifactorial and complex. They're not just they work on one receptor or something like that. They have a lot of mechanisms with possible effects on various neuroreceptors in the brain, various neurotransmitters, and possibly and mitochondrial function. There is a recent study in a mouse model of autism looking at CBD effects and showed benefits in on repetitive behaviors and anxiety, but not any improvement in social deficits in that mouse model. This study showed that another role of CBD or another mechanism was that there were changes in mRNA expression in several neurotransmitter related genes post-treatment. So there's really a lot of effects on the brain chemicals that carry messages around the brain and also modulate brain function and also work on neural circuits, which are important problems in autism. We know that cannabinoids do have some therapeutic roles that have been documented, most importantly in the treatment of intractable epilepsy, and also have been beneficial in treating nausea and vomiting associated with chemotherapy, and to some extent in treating weight loss associated with HIV infections. There are other areas that it doesn't seem to be effective, and we don't know whether it's effective in autism. But there's been a lot of interest in it because of its reported effect on anxiety and possible social improvement. There have been some adverse events of CBD identified primarily from epilepsy studies, and these include insomnia, nausea, diarrhea, vomiting, dry mouth, a lot of milder problems, and then more serious are elevation of liver function, liver enzymes, which could lead to chemically induced liver injury. So it's not benign. And it really is important to see whether it has any effect. And there have been a number of studies published over the last 3 years or so that suggest that there is an effect, positive effect on behaviors in autism. So there have been a number of open label, uncontrolled studies, meaning that excuse me, the family and the patient knew what they were getting. They were all getting CBD of some sort. Most of these were observational studies. So the family wanted to use CBD. They went out and bought CBD and they and they used it. And then the physician or clinician who was studying what they were doing asked them to complete questionnaires after they had been given the CBD that they were taking. So really uncontrolled studies, because you can buy, 50 different brands of CBD and have 50 different things in those bottles. And so there was really no control, but based on those studies, they did feel that there was an improvement in anxiety, communication, and disruptive behaviors in about almost 2/3 of children with autism treated with CBD. And most of those were CBD to a THC ratio of 10:1, which is relatively high, and in some instances was associated with the children having a bit of a high from the THC component. In more recent studies, there have been several, again, a prospective uncontrolled study using parent questionnaires showing that 30% had a significant improvement and 54% a moderate improvement in restlessness, rage attacks, concentration, and mood. And interestingly, the reported side effects in this study were primarily sleepiness, lack of appetite, and restlessness. Excuse me. And there have been a couple of more recent studies, but they're basically the same. That there were some, now this more recent one sorry, I covered that. In 2021, I'm sorry, excuse me 1 minute. It showed a different effect. This was using 2 different formulations, and they did not find good evidence, thank you, evidence of efficacy in treating the targeted behaviors. They did not find a significant difference between the groups in most of the behaviors that they were looking at. So mixed results with studies that were not well controlled or not controlled at all. Now, I told you a couple years ago at this meeting that we were starting the study, double-blind, placebo-controlled study, a crossover study of severe problem behaviors in children with autism. And we've just finished the study. We started it at the beginning of the COVID pandemic, finished it as masks were starting to come off in July this year. So went through a lot of challenging times with the study, and we are just starting to analyze the data. So that's still all in various levels of analysis. But the study was aimed at targeting the most severe and pervasive behaviors. And we used a highly purified plant-based cannabidiol with a very low concentration of of THC, and this is Epidiolex, which is the only approved, FDA approved CBD preparation. And that's the one that's used for treatment of epilepsy. And the goal was to achieve or obtain objective indicators of improvement, as well as subjective indicators of improvement. We used a multimodal assessment approach with parent observation, clinician observation, and then objective objective measures, including neuropsych testing, neuroimaging, and electroencephalography. Our primary outcome measures were looking at aggressive behavior, self-injurious behaviors, severe, pervasive, repetitive, and perseverative behaviors, and stereotypies, and extreme hyperactivity. And by extreme, I mean this is the level where a child cannot sit still and focus for more than seconds, a few seconds, all day long. These are not mildly hyperactive children. And we had some secondary outcome measures as well, looking at anxiety, sleep disturbance, social interaction, eye contact, social communication, and language. So as I said, we did use direct neuropsychological testing and parent questionnaires, and did eye tracking, EEG, and neuroimaging. And this is so this is some demographics and information about the participants in the study. We screened, well, actually, we screened initially about 200 children, but a detailed screening in 57. We ended up randomizing 44, and 30 completed the 20-week study. It was 2 phases. The children were tested at baseline. They then were randomized to either placebo or CBD. They took that for 8 weeks. They were retested at 8 weeks, and then taken off whatever they were on. There was a 4-week washout period, and they crossed over to whatever they hadn't been on the first time, and then took that for 8 weeks, were tested again, and then came off of that. So there were some children who were ineligible for the study after we had screened them and thought they were. Nothing exciting about the reasons. And then 4 who decided they weren't interested in the study. We excluded any children who had a known genetic disorder that accounted for the autism. We excluded children with epilepsy, children with any other health issues that might be significant or might interfere with the study. We could not use children where we couldn't get any blood draw. One of our children that we had randomized initially went on an anticonvulsant medication by their primary doctor, and so they could not be in the study. This study was all male, and there was a specific reason for that. The plan was to do an all-male study and an all-female study separately, and not to combine the data from males and females, because the presentation of severe autism is different in males and females. Not just severe autism, the presentation of autism is different in males and females, and that's just starting to be better recognized. And there are a couple publications now on that, but we felt that it might dilute our results if we tried to look at the same parameters in males and females together. So the plan was, as I said, to do separate studies. This will report on the study in males, which has been completed. The age range was 7 to 15. And as you can see, ethnicity, we had a good mix of Hispanic and non-Hispanic, and a good diversity in racial representation. We had a number of adverse events, 44 while on CBD and 33 while on placebo, but importantly, no serious adverse events. We had, these are some of the adverse events. Insomnia and difficulty sleeping was the most commonly reported side effect, more so in CBD than in placebo. Increased appetite was also common and present in both groups. Diarrhea and loose stools was really appeared to be a CBD related side effect primarily. Increased self-injurious behaviors. I cannot see the numbers, but I believe it's only the placebo group that had an increase in self-injurious behaviors. And then elevated liver function tests, not surprisingly, we did find in a few patients. And then in very small numbers, other problems. Several of our patients developed COVID while in the study. And then we had just single, one child in each group who complained or who had increased appetite, sleepiness, itchiness, rash, and constipation. And then clearly unrelated, well, at least we think they're unrelated, adverse events, a dog bite, a piece of glass stuck in the foot, and then a sponge stuck in the nose, and a seizure, a new onset seizure, interestingly, was in the placebo group, and one human bite. So even though those are unrelated to the drug, we do report these as adverse events because they happened during the study. So as I said, our data are still being analyzed, and we really don't have a lot of numbers to report, but just to give you a hint of what's to come or what we're finding. So the study participants tolerated the oral CBD very, very well. They all were able to take it. That was one of the concerns. A lot of the children are, well, all of them were extremely picky eaters. A lot of them were were averse to certain tastes and textures, and so there was a concern among some of the parents that maybe they wouldn't take this medication, but they did. They all did, and we got measurable blood levels in all of them, and I just showed you the side effects. These are but this is the mean blood level. It doesn't tell you anything except to show that, I don't think I have a pointer, but the first peak is the group in the first 8 weeks who was on CBD. And what you can see is that the blood levels rose pretty quickly and then stayed stable for the 8 weeks, and then dropped down over the course of the washout period but they didn't drop down completely to zero. And what we found was that there was a little bit of CBD still present in the blood at 12 weeks after 4 weeks of washout in some of the children. It was a very low amount, but still it was measurable. And this is interesting because and important because it may affect our results of our crossover study, because some of the children they still have CBD in the system during the second 8 weeks. The second peak to the right shows the second 8 weeks of the children who were on CBD. They had no CBD in their blood from the beginning till 12 weeks, and then they peaked and stayed pretty stable until the 20 weeks. So we were able to measure that. And then one of the things that we did was clinical impressions. So these are clinicians who were seeing the children. We saw them every week to every other week during the study. So this is like a clinical global impression of change, basically. And so what we noticed was that there were two-thirds of the children who completed the study, the 20 of the 30, showed measurable or notable improvements based on clinician ratings. And the remaining third either showed no change, no detectable change that we could see, or in one or two cases were a little bit worse. Now, the problem with that is that there were really a lot of variables that may have affected these changes. Some of the children had COVID infections during the course of the study, so we're going to be looking at that, whether that made any difference in in terms of their behavior changes. And some were going back to school at the end of the study after having been home for a year, being homeschooled because of COVID. So it really complicates the picture some. But again, because of the design of the study, we had both placebo and active drug treated children at the same time, in the same time frame. So if going back to school made a difference, we should see it in both the placebo and the CBD groups. We had actually more kids in the placebo group had COVID infections than in the CBD group, so it's hard to compare that, but at least our CBD group didn't have very many COVID infections, so we were able to look at their within group differences. So for the primary outcome measures, where we used questionnaires or tester administered tests, our statistical analyses are really detailed and complex, and currently using mainly mixed effects models with subject-specific random effects and fixed effects for treatment, CBD or placebo, treatment duration, their interaction and the time of treatment, a number of items. So it makes it very difficult to explain or to show you a graph of the of some of the data, but I'm going to show you an example. It's not to be interpreted as final data. It is an example based on that model. But this is from the data. It's real. It's just that it's not just the data. So what this is the repetitive behavior scale, which is a parent administered questionnaire, total score, The orange bar, I guess it is the placebo group, and the green bar is the CBD group. The left-hand graph is weeks 1 to 8, and the right-hand graph is weeks 12 to 20. And what you can see is that between week 0 and week 8, or baseline in week 8, there was a big drop, and that means an improvement actually in the scale, a big improvement in both groups, in the placebo group and in the CBD group, meaning there's a big, a pretty impressive placebo effect. So we see it in the first 8 weeks, we see it in the second 8 weeks, a big placebo effect, but over and above that, there is a bigger CBD effect. So actually CBD did cause a significant reduction in in repetitive behaviors and these problem behaviors in both the first 8 weeks and the second 8 weeks of treatment, above and beyond what you would see with the placebo. So there is an effect. It is about for this test, for this particular graph, it's about twice as big as an improvement with the CBD as with placebo. And then I wish I had a pointer. This is looking at CBD levels and the score on child behavior checklist. Or I'm sorry, is this the repetitive behavior scale? Sorry. And showing that with the children who had the lowest amount of CBD in their blood on the left, there was no difference between scores at 0 and 8 weeks. Children who had intermediate levels had a difference, significant difference. The p-values, I hope you can see because I can't. And then on the right, there was also a bigger difference after treatment with the highest levels of CBD. So the data analyzed so far do indicate a large placebo effect. And this, I only showed you one test because, as I said, these are all being analyzed now, but we're seeing a large placebo effect in caregiver reports. And we're seeing improvement in behavior problems, including self-injurious behaviors, repetitive behaviors, and stereotypic behaviors, but a larger effect with CBD group than with placebo. And possibly the effectiveness may, at least in part, be depend on the dose and how much actually gets into the blood, because the absorption rates can be different depending on what depending on a lot of things. And then one final interesting little bit of information related to possible mechanism of action of CBD. And this is from our MR spectroscopy study. So we did neuroimaging, or we attempted neuroimaging on all of the children. It's very difficult to do neuroimaging, as you can imagine, on children with significant behavior problems. And they're not sedated, of course. And so we were able to get useful images in about half. We did structural imaging looking at what the brain looks like. And we also did spectroscopy looking at different brain chemicals. And this is just one of them. myoinositol, and myoinositol is a chemical in the brain that's thought to be a marker of neuroinflammation. And what we saw when we looked at CBD, the CBD level versus the amount of myoinositol, the level of myoinositol, was that in the higher the higher the CBD level, I'm sorry, I'm going to just step away just 1 minute. Wrong slide. This is no CBD on the left and CBD on the right, so placebo and CBD. And in the presence of CBD, the myoinositol levels are lower, suggesting that there may be less neuroinflammation, and that is may be relevant to the brain, the underlying mechanism by which CBD may help. So this is just the beginning. There's tons of data to analyze, and we are already excited about the findings, and hope we continue to be excited the more we do. Lots of people to acknowledge. Oh my goodness, that's so small. But the research staff, the imaging people, statistician, the CMCR, Center for Medical Cannabis Research, in which the study is housed at UCSD. And the funding was from the Norda Foundation and Holistic Foundation, Greenwich Biosciences, GW Pharmaceuticals, and Jazz Pharmaceuticals, all the same group. Provided Epidiolex and placebo, no other funding, but just the drug and the placebo. And I'm really appreciate the families who participated in the study. It was a lot of work for them. Thank you. Any questions?
We're sort of running over a bit, so if you got a quick question. Okay.
Because it's so exciting, Doris, and thank you so much for doing that. I know how hard it is to work with severe type of autism. We knew that the placebo effect is going to have an impact, but I think we're all surprised by how much is that. It's amazing that we are seeing such a dramatic impact on placebo. So do you have any explanation for that? Why this is happening?
Yeah, so that's a really good question. And I would start, talking about how, maybe it's because of something in the mix of, the sesame oil or whatever, but there's a huge placebo effect with other things that have been studied in autism. Oxytocin, the most recent one, paper just came out, negative effect of oxytocin on social affect in autism, but high placebo effect. Another drug that was studied a few years ago, and I'm blocking on the name of, same thing. I don't know what it is. My group and I have discussed this a lot, because we're trying to figure out whether, I think it's maybe to a small extent expectation, but that doesn't change the behavior of a child with really severe behavior problems.
So that actually leads to a question from Claudia Villarreal. So would you recommend to use CBD for severe behavioral issues, which you just mentioned?
So based on our data yet, I can't say, because we haven't finished analyzing yet. All I can say is that it looks very encouraging. And I think if it were able to be prescribed, I would prescribe it. I mean, it is, so Epidiolex is the drug that's approved. It's approved for severe epilepsy. We can prescribe it off label, but in general, the insurance companies don't pay for it. So even if we wanted to prescribe it, it's not really doable now. And if somebody decides to go and get it from a dispensary, the doses that we use are so much higher than the doses that are usually gotten in a dispensary that there's probably no overlap, and it would cost a lot to go up to the doses that we used.
Sure. Actually, speaking of dosage, that was going to be my question. I know that there's not enough data to support the benefits of it. However, I want to try it. I think it's worth trying. But there is no information online on how to dose it for autism. So is there like any information we can get?
Right. So there is
Yes,
no
ma'am.
information because it's not regulated at all. And like I said, it's not approved by the FDA. So I can only tell you the doses we used. And I can tell you the doses that in the one other study that used a drug where they actually knew what the dose was, they went up to 10 milligrams per kilogram per day. We went up to 20 milligrams per kilogram per day. What you get in a dispensary is maybe a milligram total, maybe 20 milligrams total, but we're talking about if you have a 30 kilo child, you're talking about 300 milligrams a day, as for 10 per kilo and 600 for 20 per kilo. It's a lot. And it should be monitored for liver dysfunction to make sure there's no problem there.
I think we're going to have to break at this point. So thanks very much, Doris. It's now 10:47. So can we come back? 11:02. 15 minutes for a quick coffee. It's not much, I know, but you're all stars.
Autism Tree has been serving one family at a time for 20 years. And now we have focused our process into 4 core values: embrace, connect, empower, and create. And we embrace every family with love and acceptance.
Before Autism Tree, we felt lost. We kind of felt on an island of our own, and we didn't have anyone that really understood us. Whenever we've gone to events, he feels embraced. He feels like he's at home, and his guard goes down and he's himself.
We connect them to a community of resources.
I can remember how confusing it can feel and overwhelming. And to be able to say to a parent, you know, I've been there, you're not alone, and help a parent see the value of who their kid is beyond the word autism.
We empower them to create the best version of a future for their child.
Music really does help me to express myself. Felt empowered when I was a liaison in the San Diego Symphony.
And create organic relationships, meaningful opportunities, and lasting friendships.
Part of my love and excitement over, Autism Tree is, you create this massive umbrella where all these like-minded people, all these wonderful kids who all kind of have a similar but different approach, and give them an opportunity to, explore and enjoy.
The idea of the navigating neurodiversity is exactly for those families that are in crisis position, is to have someone say and help prioritize and say, to help your child, do this first, because this will unlock these resources.
And we connect them with ABA providers, mental health providers, family counseling that might help them as a family. We also look at their IEP and the services that they're getting from the school to ensure that they're adequate. IHSS that helps families who a parent has to stay home and give up their career.
Our goal is to go big and, with your help, serve 200 families who deserve to create a brighter future for their child with autism.
Are you turning these cats back in? Whoa. I felt that was important to contextualize before I start using therapy. Yeah, I think that equation around is fine. Yeah. Oh yeah. Let's get inside. Oh, yeah. Well, sorry again.
What is it?
Let's go on. Next speaker is Leanne Tchaikovsky, co-founder and chief science officer at Brain Leap Technologies. And she's also at the Department of Physical Therapy, Movement, Rehabilitation Science at Bouvet College of Health and Game Design and the College of Arts, Media and Design at Northeastern University. What a long sentence. What a long sentence that was. But I was saying earlier how much this is, for me particularly, a kind of a family thing, going back to when I first met Alison. I also remember being in my office at the Salk Institute in the 2000 or somewhere in the very early 2000. And this member of the lab, Terry Sinofsky's lab, came and knocked at the door, wanted to have a chat and see what on earth I was and what I was doing. So she sat there, and so then I asked her a couple of questions about what she was doing. And if you ever get a chance to see this, There's an amazing painting. This is well known to students in the field. Amazing painting. Oh, by Ilya Repin called Unexpected Visitor. And this was used, as you can see, by the wonderful Scottish scientist called Yarbus, to look at the shifting gaze. I mean, who's looking at what? And so it's funny for me that 20 years on, what Leanne is doing is taken that gaze and is very much one of our experts on gaze and games. So I will let you speak. Lovely to see you again.
Lovely to see you too. I'm delighted to have been invited back again because coming back here to the Autism Tree Project Foundation annual meeting is like having a warm hug, and it is so needed today. I don't know what you're doing. Alice is wearing a puffer jacket. It is colder in Boston today, my new home, than it is in San Diego. I don't know what you're doing, but it's a little scary. So hopefully it gets warmer in the next days. So yeah, I am here to tell you about movement. I trained as a neuroscientist, but I got really obsessed with behavior and particularly eye movement. And I'm going to particularly tell you about a set of studies that I conducted here with my colleague Jean Townsend in the Department of Neuroscience. That was a clinical trial that was particularly designed to address one aspect of attention in children with autism. But it actually redirected my studies into the importance of play. And so Roger very kindly brought me back to some of the work that I did with Terry Sinofsky as a postdoc. And during that time, I was really taken by how people learn super early, and by people I mean young children learn where to look in our environment. And this picture illustrates it super clearly. So this is an example where somebody's just looking around this image to see what's going on. But if I were to ask people to look at the picture and estimate the ages of people here, they would look at the face, the face, the face, the face, the face. And this happens even with young children because we know where to find information in our environment. We learn it very quickly and implicitly. And this is something that can differ in children with autism. It's one of the many things that can differ. And it's one of the things that develops very early on through play-based relationships. So in the 20 years that Roger was discussing, I got very interested in using games and game-based technologies in order to study behavior, in order to make assessments and make some interventions. And of course, this picture here shows us what many of us think of as play, physically moving, running, sliding, swinging in our environment. But this is also play, right? And a lot of the same things that make play exciting in this environment also make it exciting in this environment. And those things include autonomy, right? You get to pick generally what you want to do when you're playing. You have a sense of purpose. I want to go down that slide. I want to capture the treasure in the video game. There's a sense of mastery. You can engage things again and again and again. You can try to succeed on your quest. There is usually a relatively safe and explorable amount of uncertainty in games. Some games, and particularly I think all of us know many people for whom games can be very immersive, super, super focused. There's a sense of competition at times, but also social interaction, peer reinforcement. These things I've seen time and time again in working with, and I typically work with teens on the autism spectrum, teens and young adults, most of whom, thank you very much, Alison, for giving us that wonderful talk this morning about this broad spectrum. Most of the people I work with have relatively minimal support needs, and I'll explain in part why that is, but we're moving to other parts of the spectrum as well. But the idea is that these are powerful ways to play and to learn in the autism community as well as in the broader community. And so why don't we use them in order to make better assessments, put people in the best place we possibly can to measure something and help people want to engage with their therapies? So I told you that I'd tell you a little story about some interventions that we built we're starting around 2012, around the time when this paper came out from Sean Green and Daphne Bolivier that was talking about serious games, because it was found that individuals who played a genre of games called action video games, the ones that you tend to think of as the shoot 'em up, the really fast movement, so games that demanded integration over a broad area, fast shifts of attention, people who played those games tended to have improvements over those who didn't in perception, visual spatial attention, movement skill, and also working memory. So there's a bunch of issues with this literature, and this was very early days, but there are some self-selections in this population. Of course, if you already have better skills in those domains, you'll find games more appealing, but it was also found in some studies that for people who didn't play and weren't necessarily good at those things, playing these types of games actually did build skills. Interesting. We wanted to use them for a very specific purpose. We wanted to make video games to improve specific attention skills in teens. It was mostly teens through young adults with autism. And the specific type of attention, because attention is a very broad construct, is a type of orienting attention. I'm going to tell you a little bit about the data that led us to that. But importantly, we really wanted to do this in a way that was different from some prior intervention studies, because we knew from the literature that doing this in a way that required people to come to our lab multiple times per week, like you have to go to the gym in order to exercise, wasn't necessarily going to work. We needed to make it accessible in their home so they can do it 3 to 5 times a week, right? I get on my rowing machine much more because it's in my basement than I would if I had to get in my car and drive, because that just doesn't happen as often, right? Same thing here. We wanted to make it very accessible. We wanted to make it home-based on a computer that anyone could use. We specifically, and I'll talk a little bit more about this, we make these games, they're not using a mouse, not using a touchscreen, but using their eyes, because we believe that gaze opens a back door to the neural circuitry underlying attention. Okay, so why did we do this? And that's my colleague, Jean Townsend. She had shown back in 1995 and 99, some studies using, I'm just going to call it a Posner-like queuing task. So it's a task where there's a queue and then later a target that appears either in the same location of the queue or on the other side. A very, nerdy, controlled type of experiment that we do in our lab. But the special thing was that the time between the queue and the target could be varied. It could be very short, 100 milliseconds, or it could be long, 800 milliseconds. And in the span, if we're talking about eye movement behavior, 800 milliseconds is kind of an eternity, because we make approximately 3 to 4 saccadic eye movements. These are the very fast eye movements you use to explore your world, use to read a book. These are the ones we make all the time, 3 to 4 every second. Okay, so you can make a lot of them in 800 milliseconds. It's a long time in terms of neural processing, in terms of this type of behavior. So what we're seeing is that typical kids of the same age as our autism population, regardless of whether the queue appeared 100 milliseconds or 800 milliseconds before the target, they did equivalently well, hovering up here a little above 90%. The ASD group, when given 800 milliseconds to process the queue, did pretty well, comparably, honestly, with the other group. But 100 milliseconds was not enough time to orient their attention. And this is not a movement. We had them fixating in the center. This is to send their covert orienting, their mind's eye, if you will, to the spot where the target was about to appear so that they can discriminate it and give the right answer. It was too fast. And this is something that we thought was perhaps very core, perhaps the sort of thing that actually could, if detected early, could build into something that is a skill that helps people pick up subtle social signals. I am fond of saying that if you're not fast with your eye movements in a dynamic social setting, you'll miss that cue that tells you that mom is a little annoyed with you, right? If you didn't see my eyebrow raise, then you missed a very important cue. And there are lots of these subtle social cues that are part of our everyday dynamics. And our kids on the autism spectrum may very well be missing them. How can we try to ameliorate this? And this is where we get to why in the world would you want to play video games with your eyes versus with all the other ways we have to play video games? And it's because the saccadic eye movement circuit, again, saccades being those really fast eye movements that we make 3 to 4 times a second, is widely distributed throughout the brain. It is very, they involve a lot of processing, a lot of visual processing, a lot of auditory processing. We don't just make eye movements to things we see, but to things we hear. So you can very well say that the voluntary saccade circuit is equivalent to the voluntary attention circuit, how we shift our gaze, right? And I showed you data about attention previously, but it could just as well be about saccadic eye movements, right? If I'm showing you in a very simple task, I present a single big old target on a screen, and I ask someone to align their gaze with it, make a saccade to it. The kids on the autism spectrum in red, it takes them more eye movements to align. Every one of those takes longer, meaning you're missing information. The very first one they launch takes longer, and the first one they launch is less accurate, which is, of course, related to the fact that it takes longer overall to orient. So how can we use what we know about this circuitry in order to ameliorate these kinds of effects? So together with Jean, we designed, so that she's been studying attention. She's a neuropsychologist, has been in this for a much longer time. And I came from a very different background studying eye movements. Together, we decided to make a suite of games that you play with your eyes, to use this circuitry kind of as a harness to try to train attention. And we made a suite of games with a lot of different kinds of training principles in them. And I'm going to show you them. And I think that they're not, I think that these are not, you won't hear the sound, which is probably for the best. Although I hope you can see them. There we are. Okay, so here is you can hear the sound, okay, but you can still hear me, I hope. Okay, so here we're hitting the moles that are popping out of the ground, except for that special mole. Okay, so these guys that have they're the bandit moles, we hit them to pop them back into the ground, but the mole that has the glasses on, we call it the professor mole, and we don't hit professors here at UCSD or at Northeastern. So, and the kids get it. They get it really quickly. So this is a task that requires them to switch fast between go to it, take a break from it. This is training a different kind of principle. This is training steady fixation, which is needed because if you don't have steady fixation, then you're accumulating time before you can make the next movement. This is a really fast game that requires you to get through that green gate. It doesn't start out this fast, and make a decision about whether or not you can pick up that star along the way. That's an executive decision you have to make super fast, given the temporal demand of your eye movement in order to guide your eye. So this is actually, at the time, was my 8-year-old playing that game, and he loved that game. This is a game that actually one of our then high school interns made, which is a beautiful game, but it's much harder. Your job is to actually avoid the heads of all the little comets coming at you, and you have to stay in this aperture. It's extremely hard because we don't typically look into empty space. It is very much a game about avoiding looking at things and looking into empty space. And so we unlock that game in training after you've done a bunch of other things. These are just a sample of some of the games that we have. I just wanted to show you some effects because we saw some, and this was a small clinical trial, but it generated a lot of excitement. And we of course saw benefits, we of course saw improvements in the game. Of course you did, right? The kids play the games a lot. You saw improvements in the game, but these were things that we observed outside gameplay. So this was in being able to look more steadily at targets in terms of fixation, deviation, improvements in post versus pre, that the accuracy of something called an anti-saccade. So instead of making a movement to a target, you actually suppress that movement and make it in the opposite direction. That requires an incredible amount of inhibitory control. And during our training, the kids got much better, so their accuracy improved after training. So we also engaged kids in simulating driving tasks. And here I learned that 9-year-olds shouldn't be put in a driving simulator because they like to crash. that was a good learning. But generally we're trying to get to something that was more a real world transfer. So a little bit less like the kind of task we typically use in the lab. And so we did see some benefits in divided attention in this task, but not necessarily in road excursions, partly because they were all really bad drivers, so particularly the young ones. But these were some nice effects. We also saw some effects in the kids who, so all of these kids had a ADOS, a community diagnosis, which was verified with the ADOS-2. So that meant that they were, primarily diagnosed with autism, but several of them also had ADHD. The kids who had ADHD, when we actually learned later to just give this tool to all of our kids, they actually showed on the screener tool that we gave to the parents a lower rate of endorsing ADHD symptoms after training. So although these were made specifically to target that orienting aspect of attention, the kind of attention that kids with ADHD tend to have a problem with, which is more of focus, also seems to be improved. Along the way, we generated a bunch of assessments because we realized that in order to assess things out in the field, we would need to actually translate those from the kinds of tasks that we used in the lab. And so I can show you these really quickly. This is basically like a Ghostbuster, pull the ghost in. That is a basic SICAD center out task, kind of like I showed you the data for, but it is more fun. I think this is a really excellent task, and this one has legs. This one is basically a continuous performance task. For those of you who know what that means, your job is to just look at the fire, keep the soup going, and ignore everything else that's going on in the campground, which is hard because there's eyes popping up. There will be some animals popping up. And so this is a very sensitive test, and it compares nicely with standardized tests of continuous performance that tend to make kids cry. No joke, like they tend to make kids cry. So nobody wants to do a continuous performance test. That's very palatable. This is a test that's very much like the one where I showed you with Jean's picture on it, right? This is a kind of covert attention orienting task where the cue and target, so the cue is the brightening of the box, and the target, believe it or not, one of those paws pops up first before the other two, and you have to detect that, and kids do. This is an anti-SICAD assessment. So basically your job is to find the fish and ignore the turtle, but the fish isn't always there. The turtle pops up first, and when he does, you have to look in the opposite side. Don't look at the turtle. Bummer, missed it. So again, don't look at the turtle there. So that was the fish came up first, and when the last one will actually get it right. It takes a bit, but he gets it right. So his first movement wasn't correct, and we can collect all of these movements and try to analyze what is going on with their behavior. I told you that this correlates really nicely with a test that's out there, the TOAV. And this is some nice behavior. We need to do a little more work on this. However, my colleague Joe Snyder and I did publish on some early results. You'll see that there is, I'm the acting chief science officer of Brainleap Technologies, because these games were very exciting to some of the people in our community, and UCSD helped us start a company so that we can make them more available. The pandemic made that kind of hard, but we do actually have some things that individuals can use, but we're mostly interested in getting them into schools where they could be accessible to all. We did a in-school study with the University of Florida, and we saw some nice effects. This is just very generic. Improvements in fast gaze shifts, improvements in inhibitory control and fast attention shifts. But in those tests that I was just showing you, we see nice improvements over time, right? And this is over many, many kids, as opposed to the previous plot that I just whizzed by. It was only one kid. Okay, so I've been talking to you for a couple minutes here about these games, and I think that games are really, really powerful for assessment and potential interventions, and we're continuing down that line. On this path, while I was doing this work, and I was a lot younger then, we were also a poor lab. So even though we got a clinical trial, we were using a first-in-class eye tracker that could actually attach to a laptop computer, that could actually go home, and it broke a lot. This is what happens with first-in-class things. But I'm glad it did, because I got to meet a lot more people in our community because it broke. I got to go out to folks' homes and go give them another one, go figure out what's going on. And in that time, I got to see the reason why these kids were so excited about our study. Because they're writing game narratives in their home, because they're drawing detailed storylines about the video game they have in their head, because they're making their own games and it's all stuck at home. And so this was the first time that I was like, this is nuts. We can't let this happen, because this is some amazing talent that's stuck in our homes and it's not getting to the light of day. So in 2018, I applied to the San Diego Foundation and they approved some funding for our first internship, where we had 25 autistic young people come and work with us to make video games in teams. And it was beautiful. And we had a fantastic showcase event here at San Diego. Fortunately, the National Science Foundation liked the idea and the idea of using game-based tech for assessments in order to do some workforce preparation. And so we got another 4 years of funding. So we were doing that work here at the Power of Neurogaming Center, now also collaborating with my colleague Pam Cosman here. So we have spread, because I'm now the Regame XR lab for rehabilitation game at Northeastern. And we were really interested, and this is a picture from that 2018 in-person program. And we were able to really put to test this idea that when we brought people together who were passionate about games, they really showed us what they could do, the motivation, just like I showed in that first slide, the power, what's special about video games. They used it to build job skills. Okay. And we went on to do this for several more years. You can learn a little bit more about this at diverseinternship.com. I'm really pleased to say that we made it through the pandemic, where we had to have 2 remote internship programs. That was tough, but we did it. We figured out how to have groups of people working together online, supported by coaches. I think that's important, coach support. Through these years, we've had people help us out. So we've had clients in the community host projects, video games. We had a video game from Sandag. How do you get from here to there in the most efficient way? And have a group of 5 working on it. It was fantastic. And so we're now doing this in 2 sites. And I want to share with you, lest you think that this is incredibly Pollyanna-ish, I'm not the only one. I started this out thinking this is a great way to learn tech skills to get a job. I now am very happy to stand here saying this is a great way to, if you want tech skills to get a job, sure. If you want to be in the game industry, they actually very well may want you, because the game industry has recognized, and you can look up something called the Yuki survey. It's the UK based survey of the game industry, showing they actually track what proportion of people that are in their field that identify as neurodivergent. And it's about 18%. That's 18% who say yes, who are willing to raise their hand and that know it and that's identify, right? Interesting. So the gaming industry has recognized the need to attract talent from this pool. And we've partnered in particular with Ubisoft. And so I want to show you just a really quick clip from my colleague, Pierre Esketch, welcoming the interns at this year's in-person internship program. So this is a showcase event from the in-person program. We made it. And how he talks about this. And are we going to go? Oh, do we not have Pierre? I hope we have Pierre. Well, what I could tell you is that Pierre is the head of neurodiversity at Ubisoft. He works at a studio. There's something wrong. You know what I think it is? It might just take a minute because this is a very big video. Yeah, we might need to just go on. I'm sad about that. Perhaps I can send it to you separately because it's part of like the 1.5 hour long showcase event. But I think because it's part of this massive video, I should have cut out the 4 minutes of Pierre talking. But Pierre Esketch is the head of neurodiversity at Ubisoft. Let me say that again. At Ubisoft, this global AAA studio has a head of neurodiversity, and he and his team have been going around talking about how you should start employee research groups at your company. I also work with a wonderful person named Eris Bricker, who actually came out of the Game Science and Design Master's program at Northeastern. who is now at Red Storm, an Ubisoft studio at North Carolina. They are inspiring. They talk at global game jams. They've talked at a recent neurodiversity event all about the things that you can do in your workplace to better support and attract neurodivergent talent. This is coming from the games industry, about which you've heard many terrible things, but there's rightly so. But there's a change afoot, and it's very exciting. So keep an eye on Pierre. He's actually going to be presenting at the Game Development Conference, and he's been in touch with our colleagues at Stanford who host a neurodiversity event every year. So I wanted to just end by saying that we don't just host internships and you know, make video games for the purposes of for training specific cognitive skills, but we're using video game tech. This is a VR system that one of my graduate students, Trent Simmons, has been working on in order for us to study social interactions in a controlled manner. So the point is that two people are actually in this space. They're across from each other. And what they're doing is they're looking at this puzzle over here, and they're trying to recreate it here by grabbing blocks that fall down. And so we can actually have a very, very sort of locked down sequence of movements that we can compare across subjects and in conditions where one person is playing and when two people are playing. And the kind of thing we're observing, which is exciting to us, is that even in this environment where you don't really see a person fully embodied, you take away a lot of the social signals, the face. I mean, this is just a ping pong ball that they see effectively, right? That our autistic participants are slower in the social condition, in any of the movements that require fast temporal integration. In some of the other movements, no difference at all. It seems to have to do with that fast integration and movement that presents a challenge, and it's more evident in this social environment. When again, and here's a video of that, there's really nothing social about it. Why are my videos not working? Well, I guess I can't show you. Well, what's happening is that these puzzled pieces are dropping. And so in particular, that the fast piece is when the blocks are dropping versus when they're already still and you're moving them. So it's the same kind of movement, the same orienting behavior, except in one case, something is falling and you need to integrate that over time. That's the condition in which having another person across from you, even virtually, takes more time in people with autism. Honestly, I just showed, there's something, I think it's Pierre who's messing with me. I'll have to tell him that, because I just gave a talk on Wednesday to my tough CTSI colleagues, and this worked just fine. All right. So I want to thank some folks some of whom moved with me. Ara here on the right, Trent and Sundar were here in San Diego, and Minchin joined us when we moved. But this was very early days. This was October 2021. We were at an event that Northeastern holds in order to attract undergraduates to the lab. And we've been very successful. We've been doing a bunch of different things. And I highlight this picture in particular because we now have a first-year PhD student named Uri Seitz, who is interested in using games and game-based technology in order to change the way we offer inputs for AAC devices. So we are mostly working with individuals who have minimal support needs, but we're moving in that direction because it absolutely needs to be done. And so I'm thrilled that Uri wants to do that, and we have the support and colleagues to do that. He comes from a computer background. The human-computer interaction community is very behind this. So I'm super excited about the things he's trying. And I do want to end, though, with a small note of caution that using games and engaging games can have its effects, because this is our lab right now, and dinners have gotten very expensive. So it's wonderful that so many people are excited about what we're doing, but it's a little nuts, and management has become a deep and dire need. So we have a bunch of really exciting projects, and I hope that the next time I come visit you, it will be warmer, but I will be able to tell you more about the great work that these fantastic young people are engaging for our community. Thank you very much.
So a quick question before Alison. wasn't something about counterintuitive about this when you first started thinking about it? I mean, you know, the attention economy, buzzing brains. I mean, just going to make it worse, surely, with all the attention now ticked up, the speed of everything, and yet it turns out to be very positive.
So I think that there's a double-edged sword there, and thank you for bringing that up. I mean, I talked about in my job talk for Northeastern that we're in the attention economy, and we should all really be taking care of our own attention. It's very important. We're not necessarily doing a great job. However, when I talk about using video games, played with your eyes or not, for potentially young children with autism, I'm met with a good amount of skepticism, right? And when I gave a talk to Helen Tager-Flussberg's group at BU, you know, they got it immediately. They got what I was talking about these, you know, differences in orienting behavior and how we think we can address them with this kind of game. And so they're like, what's the end game, Leanne? Are you going to be putting little babies in front of your games and playing games with their eyes? And it's not a good look, quite frankly. But that's not how we see things. We expect that children should be in naturalistic social scenarios.
Right.
I don't have time to talk about it today. I don't disagree. We don't know that space. We certainly don't want to do anything harmful. However, there are things that we can do using like glasses-based eye tracking that we can turn the real world into a video game. So I'm working on technology with that can do like joint attention work. So two people are wearing glasses and you have a bunch of special objects that have been trained with machine learning so that you can identify looks to objects in the real world, right? That I think has some promise. You can do the same kinds of things, but you're not necessarily forcing children to look at a screen. So
yeah, here's a question. How could my child get evaluated or get involved in related studies in San Diego? How could a parent be able to download those games?
So I can definitely help you. So I'm no longer in San Diego, but I can certainly help you get access to those games. You have to mail me at Northeastern.
So just email you.
Yeah. So email me at Northeastern. I'd be happy to talk to anyone, get anybody access to these games. Absolutely. And, you know, it's the kind of thing that we want more people here doing this too. So if I can convince some of my colleagues to collaborate, that'd be fantastic. And I want to hear what Alison has to say, because he's a neuroscientist, but he has not dropped his neuroscience card. I feel like I've lost mine. I'm now all studying behavior.
Yeah, no, yeah, fantastic. I'm always happy to see what you're doing and how fast you went to this entire process. I remember when we met in one of these conferences, you were just presenting that. Fascinating. I'm so glad that you are also moving to the more severe type of autism. Yeah. That's where the needs are. So I have one question and one invitation.
Okay.
All right.
Great.
So the question is, if you have seen any parameters that is superior on autism, and I can't stop remembering the games that we put chimpanzees to compete with humans. And in short-term memory, chimpanzees are amazing. Mm-hmm. So they outsmart us by far. Yeah. And I wonder if some of those networks are also preserving autism, and if we could take advantage of that. So the second part is an invitation. How can we leverage what you level towards basic science? And for example, my lab has particular needs for lots of techniques that doesn't exist, that we're just inventing now, that see in your video games. I think maybe we can leverage the same tools, and while they're playing, they're solving complex neuroscience questions. They are classifying synapses, they are quantifying brain organoids. How can we leverage that? And if there's a way for us to move forward on that. Thank you.
Wow. Fantastic. And as always, there's so much in your question. So I think the chimpanzee plays the working memory games a little better than humans because they're not off spending half their brain thinking about what's in their Facebook feed. So there could be something there. And that goes back to the attention economy question. That said, there are legitimate, substantial benefits in processing. A lot of people on the autism spectrum that Alison Motri and his colleagues have characterized a lot of spatial benefits, some interesting motion processing differences, sometimes benefits, sometimes not. So there's a Steve Mitroff has characterized, there's some attention, so some focus kinds of benefits that a lot of folks have, and he's been characterizing behaviors that are kind of aligned with like TSA, what you need to be a TSA agent, right? That people on the autism spectrum tend to have the ability to, let's just say there's not just one gun in the suitcase, but two, right? Most of us stop at the one, found it. Whereas the individuals on the spectrum actually find the second one too, right? So there's just a different set of assumptions. And I think that actually could be really beneficial. And that's partly what we're seeing our game colleagues say. And the second, absolutely. Yeah. So there's a history of serious games for things like EyeWire, you might have heard of, right? For understanding retinal circuitry. So those sorts of things. But we also can connect our games with EEG and sort of looking at subtle signals. We're not quite there yet, and movement and EEG don't tend to go together, as you well know, get all kinds of artifactual signals. But I do think that these are questions that are well worth spending time figuring out, because we should be taking more holistic, real world-ish behaviors and trying to tie them to the things that we can measure neurally. So I'm here for 2 weeks. Let's talk. Thank you very much.
Excellent. Thank you very much. Next up, I'm just looking at an email, actually, microphone work. Oh, yeah.
Wonderful. I think we need the other.
You're the email. You want to give them your email?
Oh, I'm sorry. There you go. Sorry. I was paying attention to Dana and not to you. So do you have my email?
I suppose that's the way your life goes. Is Garrett here? Well, do we know how to handle all this? I mean,
they will be, yes.
Okay, so there was an email came out from ATPF. Thank you, ATPF family. We did it. It's official. County of San Diego will be launching the first public neurodiversity workforce initiative of its kind. This groundbreaking legislation, championed by Supervisor Tara Lawson-Remmer, passed with a unanimous 5-0 vote, June 14, 2022. That makes a first step in establishing San Diego as a national leader in hiring autistic individuals. So this Autism at Work panel, it will be looking at that. And I think we're starting first with a message from Supervisor Lawson-Remmer. Yes? I'm sorry.
Hello to all my friends at the Autism Tree Global Neuroscience Conference. I'm San Diego County Supervisor Tara Lawson-Remmer. I wish I could be there with you all for today's panel, but I'm so grateful to today's event organizers at Autism Tree for bringing us all together and for all the work you all do for our community. I'd like to give a special shout out to Garrett Hoff for all of your effort in our endeavor, and good luck in your first semester in law school. At today's panel, Autism at Work, you will hear about a subject close to my heart. Back in June, the San Diego County Board of Supervisors passed our initiative to support neurodiversity in the workplace. I hope this will serve as a model for other jurisdictions around the nation. Join me in taking a look back at some key moments from that special day.
This proposal is also about jobs. It's about finding individuals who have a lot to give and matching them with jobs in our community that need to be filled. You know, we know that there's work to be done. We know that we have for a long time faced staffing shortages in our county, and there's so much opportunity and need to serve.
Although I enjoy my job and take pride in serving the public, there are times when being the only autistic employee in the department is isolating and exhausting. I feel like I do as much educating as I do my regular tasks. This landmark legislation will benefit not just me, but those around me. like coworkers who wonder why their colleague doesn't smile or laugh, or don't know how to interact with someone who doesn't fit their preconceived notion of how someone should talk or act. And supervisors and managers who may get frustrated when their employee is feeling overwhelmed and experiencing sensory overload in response to a particularly stressful situation, or who are not used to having an employee who is blunt and says things that they don't want to hear, but must be said.
Idea here is we're going to lead by example. As an enterprise, as an institution, we have 20,000 county employees, and we're going to be leading by promoting equity across cognitive differences within our county workforce. And by doing so, I think we can show other local employers, large and small, that they can do the same. That this is something that we all can and should be doing as a matter of course in how we run our community, run our businesses, run our government. And by leading by example, I think we are going to be able to not only make a difference within the context of our own enterprise of the 20,000 people that work at the county, we're going to be able to make a difference for employers across our region and other counties and other government agencies across our country.
As I talk to different people in my life who are in a similar situation to us, one of the main things that everybody is talking about is what happens when they're an adult. Who's going to take care of them? Where are they going to land? Who's going to be a part of their lives? This is so, so important to all of us. It will have a ripple effect in our community. I think that San Diego taking this on and agreeing to be a neurodiverse workforce employer is going to have such a huge impact, not only on the individuals that you're going to be hiring, but also all of us, parents, aunts, uncles, grandparents, who have stood by our children through their whole lives and want to see them be successful. So thank you so much for this opportunity for all of us.
In my young career, I've seen enough implicit bias to last a lifetime. For neurodiverse people, bias is a fact of life. Even so, I still believe it doesn't have to be this way. That's why the first time I read this legislation, I felt the proudest I have ever felt to be a San Diegan. Now, I'm always proud to be a born and raised San Diegan, but my latent San Diego pride was whipped up into an unprecedented frenzy because for the first time, I really felt like I was reading legislation by people who see the community of neurodiverse individuals in this county, both those currently in the workforce and more importantly, those struggling to enter it. It recognizes autistic people's determination, not just to work, but to excel in our work. To see San Diego striving to be a leader on an issue so important to me as an autistic person is a great joy.
For all of us, we know someone in our community who is touched by autism. For me, it's my daughter. I'm going to try not to cry. I think this is so important to me personally, because I know that everyone living with autism and neurodiversity deserves to have the same opportunities to thrive as everyone else. And there's so many people with so many talents and so much to give and so much to contribute. But when we have society structured in ways that create exclusion, we exclude people who have so much to give. And I think that for so long, we have just accepted that that's just how it is, but it doesn't have to be that way. And, our county over the last 18 months has taken a really big pivot in terms of who we are and what we do here in San Diego. And thanks very much in part to the leadership of Vice Chair Vargas, and really a partnership with the whole board, putting equity front and center on everything we do. And certainly creating opportunities for inclusion for neurodivergent individuals in our community is an absolute central and core part of that commitment and that work.
Okay.
Garrett.
Hi, everybody.
Garrett,
My
you're
name
there.
is Garrett Hoff. Yes. Hi, Roger. Hi, everybody.
Hi. Good to see you. You're at Duke.
Yes. So for those of you that don't know me, my name is Garrett Hoff. I'm the director of Autism Tree's Autism Advanced Program, which supports autistic individuals in advancing their professional ambitions. As Supervisor Lawson Reamer alluded to, I'm in my first year at Duke Law School, and I'm excited to be coming to you from Durham, North Carolina today. I'm incredibly proud to have been a part of Autism Tree's efforts to support a remarkable initiative that Supervisor Lawson Reamer put forth earlier this year. The board letter opening more doors to workers who are neurodivergent, including autism in San Diego, of which you saw some of the public testimony that occurred when it was considered by the governing body of the County of San Diego. The County of San Diego, with over 20,000 employees, is one of San Diego's largest employers, and with the unanimous passage of this initiative, has now been formally instructed to promote an inclusionary hiring process for neurodivergent individuals, which includes instructions to increase efforts to recruit neurodivergent individuals for jobs at the county. This initiative is, to the best of our knowledge, the first neurodiversity hiring initiative enacted by any local government, let alone one the size of San Diego County. This is a tremendous first step towards combating the pervasive unemployment of autistic and other neurodivergent individuals, with which the Autism Society of America currently estimates to be around 66%, but has been estimated to be as high as 85%. And I'm very pleased to be talking about it today with Megan Elledge-Lavoda, Brian Lafferty, and Brandy Winterbottom. Panelists, thank you all so much for being here. And I would love it if you could all start by introducing yourselves and talking about what this recent legislation means to you. Megan, if you could start us off, that would be fantastic.
Thanks, Garrett. Can you all hear me okay? First of all, thank you for having us here today talking about this piece of legislation. My name is Megan Elledge-Lavoda. I'm a senior advisor with Supervisor Tara Lawson Reamer. It was my first week on the job that she said, I have this initiative and I want you to lead. And to be honest, what it means to me is this is why I'm in the job. This is what government is supposed to do. And so this, for us, is a flagship piece of legislation that we led. And being able to work with Autism Tree and so many in our community, really that's what it means to me. It's the people that hopefully we will be helping with this legislation.
I'm Brian Lafferty. I am a legal support assistant 2 for the Department of the Medical Examiner. I just recently transferred from child support to the medical examiner, and I am, as you saw in that video a few moments ago, I was one of the people who testified in favor of that. I was part of the advisory board that helped Tara Lawson Reamer draft and ultimately pass this legislation. And this legislation is huge for people like me. When I started with the county 9 years ago, I had a former coworker said that she was a supervisor for over 25 years. And she said that, I was a pioneer, like wherever I go, I will be someone's first autistic coworker. And it was a huge honor and privilege to help draft this legislation, because there's going to be one of the big things about this legislation is that there's going to be 2 trainings that are going to be required for for employees. There will be about neurodiversity. One is for all county employees and one for supervisors, managers, and those who sit on interview panels. And it's going to make this is going to make it easier for people like me to not only thrive, but be accepted. And thank you again for Autism. I would like to thank Autism Tree Project Foundation for allowing me to be a part of this. And all right.
All right, good morning. I'm Brandy Winterbottom. I'm a Deputy Director for Human Resources for the County of San Diego. I oversee the areas of recruiting, talent development, and equity, diversity, and inclusion. So I have a lot on my plate. I was thrilled to see this legislation come through. I worked with Megan and Brian on really incorporating what we could into the workforce for the county. So I am thrilled to be working on this program. And to me, it really means this is the epitome of public service, serving all our diverse communities through the County of San Diego through many different programs that we offer.
Wonderful. Thank you all again, everybody. So Megan, you're with Supervisor Lawson Reamer, and you were really instrumental in making this happen. Can you tell us a little bit more about the process for getting this initiative, opening more doors to workers who are neurodivergent, including autism in San Diego? How did this happen? What was the process from your perspective?
So the process was, so the way we really like to create legislation and policy in our office is we do it with our community. And especially with a topic like this, we knew that we wanted to create an advisory group. So the first thing I did is I started reaching out to organizations in San Diego who have just been instrumental in this space. So Autism Tree, Autism Society, Regional Center, we had a lot of folks. Brian was the first call that I made, to be quite honest. And I said, Brian, we're thinking about doing this. Can you join our advisory group? Garrett was instrumental as well. And so we formed this advisory group. And the first thing we did is we try to create, what does the sandbox look like? So you hear a lot about private industry and businesses having some of these initiatives, but with government, we have charters, we have a lot of more legal constraints. And so we knew we weren't going to be able to just mirror a program that was already out there. So the first thing we did was we convened and we really just talked about a whole bunch of different ideas of what in a perfect world, what are some of those things that we would like to see in this legislation. And then I met with HR and legal, a lot. That was like the really big stuff. So it was like, okay, here's our wish list. And we really, I will just give a lot of credit to the county for being willing partners with us to be creative and be innovative as we were looking at this legislation. And we really then started saying, okay, here's what we can do. And this is the box that we can kind of live in. And then we went back to that advisory group. We went back to the community. And like you heard Brian and Garrett, they helped draft this. Yes, I was coordinating this effort, but we really wanted our community not just to okay the legislation, but to really be part of drafting it and to really be part of the initiative. So we drafted the legislation, went back to HR and legal again multiple times after that, and then once we got the green light, then the day of the board meeting, which you all saw, we wanted an avalanche of support. We did not want this to be political. We wanted this because this impacts everybody across the county. So our advisory group really championed and spearheaded. I think Garrett was in DC at the time and was making phone calls, and we had over 800 e-comments in support of this legislation. To give you perspective, it's like 10. Like sometimes 0. So this was a big show, there was a big showing for this piece of legislation. We had folks coming in and giving public testimony for it. The supervisor, you heard some of her comments. This is very personal to her with her daughter. So that was the process in getting this piece of legislation at the County of San Diego. The one really thing that we worked on a lot was we didn't want it to be performative. We really were working with the county and we were working with legal because we wanted this to be something that really could be implemented when it got passed. And that was certainly a huge focus as we were drafting this legislation.
Thank you so much, Megan. And I'll say from my perspective, it was really remarkable in that I've been very engaged at a local political level for a long time, both at the city and county levels. And we never really been reached out to as an organization like this before. And we as ATPF, and as somebody who's involved with ATPF, we really are for our families, first, last, and period. And so we've always been very hesitant to engage in this way. But it was important to us because this is something that we firmly believe is going to help autistic individuals in the workforce and autistic adults. And yes, I was a lot of late nights texting, but I probably a few of the people here in this room I texted asking to do e-comments, and I want to thank those in the room for that. To give a little bit of a better sense of what exactly this means, Brian, you're an autist. You are a professional. You've been with the County of San Diego for 10 years, which is amazing. And you're also on the autism spectrum. Is there anything about your professional journey that you feel is important for people to know about what it is like for an autistic person to find fulfilling employment?
Well, like I kind of start like 9 years ago. I did a paralegal internship with the DA's office, and it was the first time I had ever worked in an office. And it was Yeah, and it was, I had, I learned a lot in that internship. Not just about kind of the work that's done in an office, but also about office culture. And, and I will say that when I started out at the DA's office, I did not tell anyone I was autistic because I was worried that people would look at me differently, look at me unfavorably. But then my trainer, after a couple of weeks, started making comments like, "Oh, you don't smile, you don't laugh," and so that's when I went to my boss and said, and I was really worried I would do this because about doing this, but I told her I was autistic, and she embraced that. Even gave me several resources that I still use to this day. And then when I was hired full time for the Health and Human Services Agency, again, first day, didn't tell anyone I was autistic, and a supervisor was showing me and the other two new hires around the building, made the same comments. And that's when I realized that I can't hide this. Like, there's a term that we use called masking, which is trying to suppress stuff that would have people perceive us to be autistic. And so thereafter, every time I started working for a new office, every time I have a new supervisor, I would disclose that. And it has made things a lot easier because, it's, people have, I've said in there, I feel like I do as much educating as a task that I do. And it's very new to a lot of people. And it's even new to I think for a lot of people in HR, because we don't see a lot of neurodivergent people in the workforce. And in the 9 years I've been with the county, to the best of my knowledge, I know of only 2 other employees who are on the spectrum, who are autistic. And and I would say my finding fulfilling employment, my journey, it's kind of made things a little interesting. Had probably almost more than half a dozen job interviews, and had a lot of practice with that. In fact, when I interviewed for the medical examiner, it was, and eventually got the job. And so it's I've been able to find fulfilling employment, and, been with the county for 9 years. So but at the same time, it's like, a lot of my coworkers, they don't understand like what autism is. Like, for example, just one last thing I'll say, like, for example, I've had coworkers who, I have a conversation with them, small talk, and they kind of look at me like, okay, yeah. But then it's like when I see like 5 seconds later, they're talking to someone else and they're the complete opposite. And that just kills you because it just makes you wonder, So there's still a lot of work to do, and I feel like this initiative is going to really help with that. So thank you.
Thank you, Brian. And I'll say, as an autistic person also in the workforce, I was in the private sector before I came to law school. I definitely can relate in terms of feeling this burden that, one, you're kind of alone in the workforce. Normally it's just you. And the other piece too, that there's this expectation almost that you as a neurodiverse person, as an autistic person, have the burden of educating your peers and coworkers on how to support you best. And in a sense, that makes sense, but it ends up being tiring, especially when masking comes into play and these other dynamics come into play. So I'm curious, Brian, when you have that situation where you're struggling with a coworker, they're just not getting it, how do you approach that discussion of being autistic and just dealing with it in a professional context? Any advice for those that might be entering a job and dealing with these new challenges?
I will say that the vast majority of my coworkers, and I probably had close to 1,000 in my 9-year career, I would say the people that I just described, that Garrett just described, I'd say it's like I could probably maybe count on both hands, and I think as a former supervisor gave me some advice and said, it's them, it's not me, it's them, if, I don't expect everyone to, like me or, or just, I think just the key is, like I just said, if at least 95% of your coworkers get along with you really well, and really like being, having you around as company and things like that, the 5% of coworkers who just, look at you weird or, just don't know how to talk to you, like that doesn't define that. So just remember, it's not you, it's them. And when you think about that, when you look at it that way, that makes it so much easier.
Thank you, Brian. And so, Brandy, I want to also bring you in on the conversation as well. Now that we've passed this initiative, now kind of comes what's next. And so you've been tasked with implementing this first of its kind employment initiative. What do you think will be the easy part of making this happen? And what do you think will be the hard part?
I think the easy part is that everybody that we've worked with from top down, from across the organization, everybody wants this to work. And it really has made a difference. People have opened doors. People have started talking about, hey, I have a child that's neurodiverse, or I have a sibling that's on the spectrum. So it's really created a conversation about raising awareness in the organization. And people are really feeling, our employees are feeling more open to talking about their individual experiences. The challenges, as Megan kind of alluded to, is we are a government organization. So we deal with a lot of authoritative documents that talk about how and what circumstances we can hire particular employees in. So we have to be mindful of each and every one of those kind of guiding documents. Look to make changes where we need to, obviously. And the second part of that is also making this successful. We absolutely want this to work. We want it to be successful. But as we have talked about, there are a lot of differing opinions in the communities as well. So we need to get it right. So we'll be bringing on a consultant to help us initially do this work. And also, we definitely have we are deliberate about a culture of inclusivity at the county. So I think that will help be foundational as we begin to roll out different components of the program.
Thank you. And I just want to, again, really applaud the work that you're doing. It's fantastic. And so I know that we probably have a few employers in the building as well. In your opinion, as somebody who's doing this work, as somebody who's an HR professional, how do organizations create trust with the neurodiverse community in the same way that the county has been making this very intentional effort to do?
That's a great question. I appreciate that. Thank you. I know for the county, I can speak for the county. Again, we're very deliberate in setting an inclusive culture and really establishing values that allow space for individual differences and different abilities. We want our employees to be their authentic selves at work, and trust is one of those values that we've worked to incorporate into the organization. So again, we've started by raising awareness. Again, there's been absolutely more dialogue in our organization talking about individual circumstances, individual situations. Again, we'll continue to work to bring training to the organization, as Brian alluded to, not only for all staff and coworkers, but for our supervisors as well. We're also looking to make some changes to our recruiting and outreach programs so that we're reaching more diverse candidates and welcoming and showing that we have a welcoming organization for candidates of all abilities. And then finally, we'll be looking to revamp our interview process to make it a more comfortable and inclusive process. So it may not just be a series of questions where you're sitting across a table from somebody. That may not be a comfortable circumstance for everyone. So we're looking at ways that we can incorporate different abilities to demonstrate skill sets in our interview processes.
That is absolutely wonderful. And one thing that I'm curious about too is one thing that I firmly have believed in my experiences in the workforce, and Brian, I'm probably going to ask you a variation of this question in a minute, is that the supports that we need as neurodiverse individuals are the same supports that everybody else needs as well in terms of what makes a good workplace. Do you feel supported in your job? We're reaching the same goals here. And so my question is we mentioned as we were preparing for this, Brandy, that HR has a role in this, but it goes beyond that. And so can you talk a little bit about if you're just a manager or just an employee in a workplace, what you should be doing to build an inclusive workspace?
Oh, absolutely. And I appreciate the question, Garrett. I have to be transparent. Garrett and I went back and forth on this a little bit because the question was initially, how does HR establish trust? And I'm like, whoa, you can't put this in HR's bucket because as both Brian and Megan mentioned, we're a 20,000 person workforce. Garrett and I work at the same campus and we never met, I'm sorry, Brian and I work at the same campus and we've never met each other before working on this initiative together. So, I think it's critical for our supervisors and our managers because they are the folks who are having those daily interactions with their employees in the workplace, employees of all different abilities, to really set an environment where we're focusing more on what our employees need to be successful in their roles versus asking, do you need an accommodation? I think we've really tried to change that mindset and we'll continue to do so, so that employees, Garrett mentioned, I don't know why I keep calling you Garrett, Brian. Brian mentioned that there was a point in his employment where he knew that he had to have a dialogue with his supervisor. And that to me extends trust on both sides. So I think it's really our employees feeling that they have that environment of trust where they can talk with a supervisor or they can talk with a coworker, or finding that right individual. It could be an HR person too, but we all need to work collaboratively together to make sure that our employees feel like they have that environment of trust, and have resources available to them if there's struggles.
Thank you so much. And Brian, I have to say, I feel particularly honored getting to talk to you because as autistic professionals, we don't get together enough and talk about the issues that we're facing in the workforce because they're so much more universal, I think, than we give them credit sometimes. And so I'm curious, let's trying to think of what's the right way to phrase this. Let's say you get a new boss, you've had multiple bosses. What have you found works best in terms of building that relationship with your coworkers, with your boss, to thrive in your employment?
Well, I think the first thing I say is one of the first things I say after I say hi, I'm Brian Lafferty, is I mentioned I'm autistic. And in fact, right before I joined the medical examiner, the senior departmental human resources officer asked me to submit a short bio, and I included in that I'm autistic, and I helped Tara Lawson pass the legislation, and so that way everyone just knows. And the reason I talk about my autism first is because I know that I don't think my brain doesn't work the same as those of neurotypical, those who are neurotypical. Like the way my brain works, I kind of described it probably as like jumping over hurdles, like Olympic hurdles in a lot of different directions. I kind of let my supervisor know, like, this is how my brain works. This is how I, do things, this is, that's because, like I said, like, I've had, we spend more time, for those who work in an office, we spend more time at work than we do with our own families, and people are going to pick up on things that, just being in close proximity. And so I just let my supervisors know, hey, this is like, at this point, I'm not saying these are the accommodations I just say, this is how I think, this is the way my mind works, this is my approach to things. And so it gives them an expectation. Okay, then, so when they assign work to me or so they can be a little bit more patient and everything. So I have felt like just disclosing my autism has really helped in that. And it's really helped my supervisors. And I've had 10 of them over the last 10 different supervisors. Like I've been able to develop a really good rapport with them that way.
Thank you. And I'll say the pro of having to come in and face these challenges day in and day out is that we are making change in our place of work. And so that happens on two fronts. It happens on the individual level, like with Brian being in the workforce, educating his supervisors, educating his coworkers, being out there, being open about it. But it also happens on a structural level in terms of getting to that inclusive workforce that we want to create. And I'll say, there's a lot of things people don't realize about making structural change. I think it's a lot more doable than people realize. And so, Megan, I'm curious, as somebody who's been at the county, who's been involved with legislation, what do you want people to know about making change at the county level and continuing the momentum of this initiative at a structural level?
Thanks, Garrett. Systemic change, when we talk just in equity and inclusion in general, that is always the goal. It's structural change. We're so happy that the supervisor is here, that we have a board that's supportive, that we have Brandy and her team supportive, but we can't have it depend on people. We have to really look at the structures and the processes that we put in place so that it can outlive anybody who is at the board of supervisors and anybody who's in HR. I think that what I love about local government is that we're close to the people. There's proximity to the people. And I think that if more folks know that your city, your county, you can get involved in these kind of initiatives, that there is a real appetite right now for figuring out how to get great talented workers in our workforce. We've said this from the beginning. We are not doing anybody a favor by creating this initiative. They're doing us a favor. This is a talented pool of folks out there in our community that we haven't tapped into. And we're struggling to get folks working for us and getting them, in these positions. And so I think that, looking at the structures, looking at the systems, I think we were really intentional in the legislation then of looking at what is the recruitment and outreach. Yes, but then what are we changing about the interview process? What are we looking at those implicit biases? And then once individuals are actually working at the county, we get them through the door. How are we wanting to keep them there? How are we really creating, again, those structural changes where we have a culture that Brian is feeling like he's supported and other individuals feel supported. We always want our employees to feel supported, but I think that, the onus can't always be on the autistic individuals coming into the county. We all have to own that. And I think that's part of this legislation, is really trying to teach and educate so that we can create a more inclusive climate and culture for these individuals, again, that are bringing a tremendous amount of talent and to our workforce. We want to keep them.
Thank you, Megan. And when we're talking about reaching out and being a part of this structural change, I think a lot of times people get intimidated. And I definitely remember in my earlier years, especially, whether it's because you're young, because you're busy, you've got plenty of things going on. What is that bar to entry actually? How do people get started on this first step? Because I'll say, I've definitely felt intimidated at times, and the more I learned, the less difficult I realized it was actually to do it.
Yeah, I think that there's a lot of entry points getting involved in initiatives like this. I think that you can find a leader, somebody who's elected, or a business that you just really like an organization or a business, and you can go and try to make a contact there and get connected that way. You can get connected with great organizations like Autism Tree and have that more issue-based, because we always, I think government does a pretty good job about working with community partners. you saw the public comments, I always think that's super effective. You can go down and just give a public comment about an issue that's really important to you. And hopefully that gets on the radar of somebody, you know, in kind of local government at the city and the county level. I think that it's a lot easier. I mean, coming from somebody I've worked state government, I've worked local government. We serve you. Don't forget that you elect these individuals, and people want to engage with the community. So I would say like really small efforts to just engage with within government really can yield some great results. This legislation, I can say, if I sat in a room and I just wrote this, it would have not been as good. It would have been, I mean, I couldn't imagine of doing it without all of the advisory boards and board members and the county and working with folks. So I think that also remember that your voice makes policy and makes laws better. Like we are missing that, in my opinion. We need more of that when laws and policies are made. So don't be afraid to get involved, to reach out. I think that, again, especially at the local level, I think you will find we really are pretty warm. We're not intimidating, as Garrett says. Like once you get to know us, then we want to bring you in the fold and bring you into the process.
Thank you. And I'll say it. I think a lot of times we underestimate just how much we can do on this issue of being inclusive, whether it's the public space, whether it's the private space, just in what we do on a daily basis. And Brian, one thing that I don't think a lot of people would probably realize is that you're also a leader in the union. I was wondering if you could talk a little bit about what that experience has been like, and how you ended up connecting and doing that work as well.
Yeah, so I am a member of SEIU Local 221, Service Employees International Union Local 221. It represents government employees and other employees of like from the county of San Diego, like National City, like Imperial County, et cetera. And so I became more involved 5 years ago during the contract negotiations that took place in that year, because I had always been a member since I joined the county, and I was just, paying dues. And then, contract negotiations start, we almost went on strike. And when I realized like these were my wages, like this was all affecting my wages and stuff. And so that's when I became more involved. Like I was, and so last year, so earlier this year, our contract expired. And last year I was elected to a spot on the bargaining team, and I represented with 28 other people, I represented over 10,000 employees. That's a lot of pressure, because it's not only you're not only bargaining for your wages and benefits and those of the 28 other members of the bargaining team, but it's over 10,000 employees. And I would characterize the negotiations as having gone very well. We met at least once a week with the county, the county side, and and it was a very good experience, because we ended up getting the best contract in the history of the union. I mean, I just remember 8 years ago, I observed a coworker filling out a CalFresh application. That's SNAP outside of California, on their break, and so, so I just became very involved in that, and testifying in public and things like that. And it's, because, being a part of the union, like in doing all this, because it's, not just, for my benefits, but, all over 10,000 other employees. So it, and so it was a very good experience. And, and I've, so yeah, that's how I became involved.
Wonderful. Well, everybody, I think this is as much a testament that whoever you are, you can be a change, and you can be somebody that is building the inclusive workforce of the future, building the workforce that includes our neurodivergent individuals in it and bringing value and maximizing the potential of our community. I'm afraid we're out of time, but I want to thank all of our panelists one more time. You're all fantastic. And I look forward to continuing this work, and we're going to get that unemployment rate down. I 100% believe it.
Just one announcement before we break to lunch. There are a couple of people who wants to tour the labs. My lab manager, Lila, is here. Just meet her outside for a quick tour. Thank you.
So, welcome back after, I hope you had a wonderful lunch, great morning. One thing I didn't get a chance, I mentioned this to Dana, one thing I didn't get a chance to mention at the end of Garrett's panel was that when the original, I've mentioned this to a couple of you, when the original email came out, said, you know, groundbreaking legislation, first in the nation, da, 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 da. I read it and thought, how wonderful. And then I thought, what? This is the only place in the first place to do this sort of thing? How ludicrous. I mean, so I said to Megan, as I talked to her earlier, that it would be a great opportunity, in fact, since this is such an extraordinary achievement, to actually do a manual and tell people and lawyers and so on how to do this everywhere. I mean, anyway, just my little rant. Right, so now we're moving on. I was thinking that family story I was telling you earlier about the connections to, first there was Alison, then Leanne. I mean, we go back a way as well. There's Torres, Elizabeth Torres, wonderful speaker. But the most amazing thing about this is the New Jersey Autism Center of Excellence to transform autism research, education, and services in New Jersey. 2018 to 2023, $4 million they got for that. And then there's quite a number of other grants as well. Liz is one of the most extraordinary raisers of money for dealing with this that I've ever encountered. And I had a great opportunity last year. Was it last year? No, it's 3 years ago now. She was in the office next to mine at Salk, at the Salk Institute, and we had a great time talking about all of this. But anyway, computational neuroscientist originally, but now obviously a PI at the Sensory Motor Integration Laboratory at Rutgers, and the New Jersey Autism Center of Excellence is what she directs and is a principal investigator. The rest you can read in here. There's just an enormous amount of stuff. She's just a wonderful person and wonderful speaker. And please come and join me. I must say that this title is, come here. This title it makes me think of all sorts of things. Derailment within our grasp. I see trains flying off. And anyway, look forward to hearing this.
The introduction, and so I'll tell you a little bit about recent work that we're doing in the lab, and it came to us a bit serendipitously through two places. One was the state of New York, and the other one was the state of Florida, who had archived records of neonates that they do the auditory brainstem test, which is the routine test that is done at birth. And they asked me, I had invented some new ways of analyzing biorhythmic data from biosensors, from mics and video cameras and wearables, and your Apple Watch, your cell phone, anything that you actually carry in your purse these days. And they said, you know, maybe there is something here. So I said, okay, let me have a look. And so I'm going to report to you what we found analyzing these waveforms that are routinely taken. And so with very little effort and cost, we could use it to detect neurodevelopmental derailment very, very early. In one case, we looked at over 200,000 records in the state of Florida. And in the other case, we looked at about 70 records. But the interesting thing there in the state of New York is that these records, they not only took it at birth, they tracked these babies, and by the age of 3 or 4 years of age, they saved the ones that had the diagnosis of autism, so we could retrospectively look at what was different. And so that's what I'm going to be reporting to you. This is the team that we worked with. I don't have a pointer, but the first two, Eric London and Dr. Haffan, they are in, one is a child psychiatrist and child pediatrician that direct the New York State Institute for Brain Research in Developmental Disabilities. The other three folks are in the University of Florida, in the University, of Miami in the state of Florida. And in particular, Rafael Delgado has a company that creates these portable devices. He's an electrical engineer by training, and these devices, you take them to the NICU and to the birth ward and in pediatrics, and we can then administer different pulses, and it's all very controlled and so forth. So the other on the bottom row are three. The first three are graduate students in my lab and a pre-med, and the last two are at the South Trzymanowski. I did my sabbatical there, and Rusty Gage, I also worked with his lab, and I'm not going to present the results from that work, but it was an analysis of human embryonic stem cells, transcriptome data, utilizing this same concept. And we just recently published our first paper on that work, and it's broader across neuropsychiatric and neurological disorders, but also we found 72 genes that overlap with those associated with autism from the Simons Foundation database, and a set of genes associated with brainstem related disorders and cerebral palsy. And so I won't talk about that work today for lack of time, but it's very interesting as well what we found there. So the motivation, of course, is that we have a tremendous raise in exponential rate of diagnosis in autism. It's gotten out of hands. We have a lot of problems with the diagnosis system in terms of how broad it has become, covering sensory issues, ADHD, cerebral palsy, and a number of rare genetic disorders. So it's a highly heterogeneous disorder now, and that has an impact in the administration of therapies and the training, the lack of training, rather, to handle these neurological issues. And so part of the problem also is that the diagnosis is far too late. So the average is 4.3 to 4.5 years of age when the diagnosis is given in the US. So we would like to go earlier, because then we could take advantage of that window of plasticity, very early plasticity in neurodevelopment. And what we know is that babies already come to the world with a system that is measurable, with, in particular, the brainstem has tremendous importance because it connects to the spinal cord where central pattern generators are, and you need to develop connections in the neocortex to control those top down. You have subcortical structures like the basal ganglia, the striatum, that also are participating in the control of movement. But most importantly, you have their processing of sound, which propagates throughout the various layers of the brainstem on its way to the primary auditory cortex, and the signal there is at microsecond time scale. So this structure will have to coordinate different timescales at the millisecond timescale. The microsecond timescale is much finer, and it does provides a center for coordinating, so like an anchor, so to speak, for coordinating all these disparate timings from information that has to be integrated into the motor stream, and then serve as feedback to the various centers of the cerebral cortex, the cerebellum, the basal ganglia, the spinal cord. So it's in a way, it's a conduit of information sharing all these multiple timescales. So it's very important for the coordination of all behaviors in humans. And it's accessible at birth, and that's why these archived records dating all the way back to the late 1990s until present time are important. And I want to notice that part of the importance of doing this so early is that humans are altricial mammals. They are born with immature systems that need a lot of nurturing, and they will mature within the first year of age. And because of that, the early stages of development are in a very nonlinear accelerated rate, and a lot of things happen every day counts. So the sooner that you understand the problem and develop a methodology that can address those problems with a specificity of personalized approach, the better. This is a power law scale of across species, and I just wanted to that relates the weight of the brain with the amount of time that it takes to start walking. And I want you to notice that the human babies are right up there. So it takes a long time for them to mature and develop before they start to walk. So therefore, we have to really pay attention to that first year of life and take advantage of the early plasticity and how the brain evolves over time in that very early period. So we need to diagnose earlier, and we have this routine auditory screening test that tests for auditory function for the intactness of the cranial nerve 8, and it's portable and it's available, and it's done routinely, and with different types of input we can measure the outcome, the responses. And because it's being done routinely for so long, it has been well characterized. So we have a waveform that has 7 peaks, and these peaks occur at particular millisecond latencies that we can measure and estimate, and they are in correspondence with different layers and different stages of the brainstem. So starting from the ear, these regions using Roman numbers to denote them. So starting from the ear all the way to the pons, the midbrain, and then finally the primary auditory cortex. And the type of input that I'm going to be utilizing here is 70 decibels, 75 decibels, and 80 decibels, which is audible already range. And this is data collected at 25 kilohertz. So it's a very high sampling resolution, so we can get to the type of millisecond timescale resolution. So what I noticed when they gave me access to their data is that the underutilized information that lies in these little wiggles before the big peak, the big delta, Dirac delta response that you get from the brain, and the little wiggles after that, in the refractory period after that spike. And I noticed that that was not being utilized, only the peak information was being utilized. But if you zoom in, you see that there is a lot of variability there that we could utilize. And likewise in the refractory period. And so this is zooming in into that Dirac delta response, what it would look like. And that also can be can be looked at in terms of the features of that waveform, not only the amplitude in microvolts, but also the prominence of the peak, which is from the base of the minimum there to the peak, the width, which has to do with the sustained time of that waveform, and so forth. So there are a number of features in the waveform that you can look at the variations, at the fluctuations. And so that was the first thing. And here I'm showing you one motivation for this is the nonlinear rate of growth and the fact that babies grow daily, and the head also, the head circumference changes over time very rapidly. And so when you are utilizing this information, these responses, you need to take care of the anatomical differences across babies to standardize the waveform and be able to compare across different babies and different ages and so forth. And so this is an ongoing study in my lab that we send out a cell phone just to prove the point that maturation occurs very early and is tractable with simple means like a cell phone, and we take 3-minute videos and get the babies and do movements and perform analysis that can tell us how the baby is growing, the rate of growth, and the rate of neuromotor control development, the rate of volitional control, and so forth, and the different transitions. This is, we measure for 30 weeks, and we the parents send us the data, so they're doing this at home. It's scalable, and so again, the rate of growth is highly nonlinear. So here I'm plotting estimated gestational age in weeks versus body weight, and you see that there is a nonlinear curve there, and we have differences between non-ASD babies and ASD babies, because this set of babies, we had the diagnosis 3 to 4 years later. And so to adjust for those allometric effects that would confound the analysis because of differences, anatomical difference in head circumference, we then take this ratio and we invented something called micro movement spikes, which are the fluctuations that take place above the empirically estimated mean of the waveform. And when you look at all those little wiggles that are otherwise not utilized in the current analysis, you see that there is a potential for a lot of information there that we are missing. And so after we scale for allometric effects, we have a standardized waveform unitless from 0 to 1, but we preserve the timing of the peak, so we know exactly, we maintain exactly the same number of frames and information that we started with. And so one of the problems that we have across the board in the analysis of these type of variations in waveforms is that there is an assumption of a Gaussian distribution or normal distribution that then applies a one-size-fits-all model treatment to the data that is very disparate, and it shouldn't be done that way. So when we looked at this positive rectified waveform and the wiggles that we got, then we realized that what the field is really doing is taking epochs of these waveforms, superimposing them, and taking an average, a mean based on the Gaussian assumption. This is a theoretical assumption, but the question then is, if we don't theorize about it and actually empirically estimate the distribution or the family of distributions that characterize the statistics, the underlying stochastic process, these variations, what would happen? How would we gain information? And the reason why we're losing information is because when you take the mean, which is the theoretical mean, the Gaussian theoretical mean, which is the black curve there, and then 2 standard deviations above that is the red curve there. What is being done is wasting all of the gross data, all of the other information that falls above the 2 standard deviations. So instead, what we're going to do, instead of taking epochs and assuming a distribution, we're going to estimate the family of distributions that these fluctuations above the empirical mean are having. And when we do that, we discover that there's a whole family of distributions underlying each person, and how you navigate through those distributions and shift through those distributions is a fingerprint-like signature of the person. So we can then truly personalize the treatment of the data, and in any biosensing data or any other data that contains time series or series of measurements across the board. So that's the family, and then we tested different families, and the gamma family of distributions, the continuous gamma family of distribution, was the one that, based on maximum likelihood estimation, which is a methodology that we use in signal processing and so forth, allowed us to fit the data the best. This is the formula of the gamma distribution. It has two parameters, the shape and the scale, which tells you the dispersion of the distribution. That's the scale, and the shape tells you the shape of the distribution. And you can track then information both in deliberate movements and in spontaneous movements, and in signals that are consciously beneath awareness, but also in signals that are largely beneath, above awareness and beneath awareness. So we can track all this information, and we can come up with parameter spaces where we can plot each, the data points of each individual as one data point with confidence intervals, and we can track the stochastic shifts for that individual as a function of, for example, the decibel levels that we are giving them, like the different decibel levels. So these distributions change, they're non-stationary. This is very important because imagine that the field across the board is assuming that it's a Gaussian distribution, and that the system has a steady state, static state, and it's quite the opposite. It's a family of distributions, is unique to this person, and it's non-stationary. So we have to pay attention to that. It shifts, and the rate at which it shifts will give us target information for devising different therapies and so forth. So here is an example of our project with the babies that we were tracking in tandem with the ABR waveform. We were also tracking their growth and their neuromotor control development. And we were able to plot in a parameter space and characterize, and this has been done over thousands of individuals of different ages. And so now we have an empirical characterization of a parameter space where we can know when something is off the chart, when something is significantly deviating from the normative data. So we first characterize the normative data, and then we characterize the deviations, the departures from normality. But importantly, we can track the distributions as they shift, and we can tell you then, for example, that in the left upper quadrant, that localizes processes that are exponential, that are random, that are very different from the ones in the right lower quadrant, where processes are predictive, are tending to Gaussian, and have low signal to noise ratio. We have discovered power laws that tell us about these maturation processes across different types of data, and we can track these stochastic trajectories on the right-hand side of your figure there as they change over time. So over weeks of development, and in autism in particular, we have been able to uncover a maturation power law, a maturation of the motor signature that tells us that in typical development, neurodevelopment, you have a transition across from exponential all the way to Gaussian. But in autism, this transition is not happening. There is excessive motor noise. And so there is a lack of predictive code that needs to be shifted towards when you dampen the noise, and it's possible to do that. And so that gives us information to characterize these parameter spaces and derive a target for treatment, which is dampening the motor noise, so that the system begins to have a better chance to integrate the information from multiple sensory modalities. So with that in mind, we have defined the worst case scenario, the best case scenario, a target for treatment, and we have now a pretty good empirical characterization of the biorhythmic activity of normative data and of data that departs from normative states, such as the case of autism. And it's very, it's possible to apply this to the various features of the waveform. So for example, here we're doing it with the peaks prominence and separating unambiguously the autistic case from the control case. So when we looked at the information from the neonates, utilizing this methodology and looking at the waveform of the response of the auditory brainstem response, what we found was that there was a fundamental difference in these signatures between the preterm babies that did not get a diagnosis of autism, even though they were preterm, the preterm babies who got a diagnosis of autism, the full-term babies who didn't have a diagnosis of autism, and the full-term babies with a diagnosis of autism. So this methodology enables us to unambiguously characterize these differences and track them over time. And in this particular case, we were able to separate based on the decibel levels of the input as well. So it gives us a way to now develop input signals that can induce different levels of responses. And in this way, we can then begin to characterize types of inputs that will have therapeutic value, because then we can identify the target and shift it. This was also the information reflecting differences for babies who went on to have the diagnosis of autism versus babies who didn't, for the different decibel levels. So these are stochastic trajectories that tells the unambiguous differentiation between autism and controls. And we can also identify clusters of babies that have different, combined with different weights and different patterns of growth and other clinical information. So when we went those were based on the features of the waveform, and now we're going to report to you what we found in terms of the latencies of the peaks for each one of the 7 peaks that people traditionally analyze in these data sets. So for each one of the peaks, we found a shift, a significant shift in the latency. So there are delays in the latency. So the sound processing is delayed in the babies, in the neonates that went on to receive the diagnosis of autism, and is delayed at each stage of the brainstem. So by the time it gets to the primary auditory cortex, it's significantly delayed. So these babies are processing the input, the sound processing is delayed. So when they have to integrate at microsecond time scale, when they have to integrate all of that sensory information with other sensory modalities like vision or proprioception or movement, pain, temperature, all the other afferent information that is a form of sensory input to the brain and has to be integrated at the brainstem, talking to everybody else, to the spinal cord, to the neocortex, to the cerebellum, to the subcortical structures, these babies are in trouble because the information is delayed and is also very different in terms of the bandwidth of information that they are receiving. So the babies that are that did not get the diagnosis of autism went on to develop neurotypically has a broad bandwidth of information. They have access to all the frequencies, and the latencies are broad, okay? The babies in this spectrum of autism has a very narrow bandwidth of information and are shifted in a delayed fashion. So these two things are now quantifiable. I'm here now zooming in the information that I just told you. And so for each one of these, I'm plotting the empirically estimated probability distribution function as a function of the latencies in milliseconds. And so what you can see there is for each one of these stages from 1 to 7, you have a different, an unambiguous different probability distribution in the autistic neonates, the neonates that went on to receive the autism diagnosis in comparison with the neurotypical neonates. And this again, I'm going to reiterate what I just said. There is a shift, a delay in these latencies, significant, and there is, we're not talking here a p-value or a significant difference in a p-value. We're talking two different worlds. These are two different planets altogether, okay? So, and if you see the dispersion of the distribution is very narrow. So that means that the variability that you want, the broad bandwidth that you want at first, welcoming all the sensory information is not there. They have access to just a slice of that sensory information in the sound domain. And so here we obtain in milliseconds the delays, and there are synaptic delays. There are delays that are natural that you expect because the signal is traveling. So these are physical delays. But what we're talking about is in addition to that, what's happening. And when we cumulatively add all of that, we come up with 1.74 milliseconds delay in the autistic system. And this is in a system that is computing at microsecond time scale. It's an order of magnitude. So this is very, very delayed. And so no wonder that there is this, I'm putting here this picture to kind of give you a sense of how they must be sensing the world. And again, just to re-emphasize that the brainstem is this conduit, and it has connections to all these regions of the neocortex, the cerebellum, and the subcortical structures. So that is an essential structure at the beginning of life that we are already quantifying in a way with this test that is routinely done. And I want just to invite you to think about all this symptomatology that we see in the babies, in the toddlers, spinning and shaking, and all these so-called obsessive behaviors is nothing but a way of trying to get an anchor, a frame of reference that aligns the timing, because all this temporal information is disparate, is at different time scales, and the brainstem has a chance to align them at microsecond time scale. It's not happening in their case because it's delayed and it's narrow. Then we went on and did the same type of analysis in toddlers all the way, and young children all the way from 1.8 years of age to 6.8 years of age. And I won't go into the details, but it was consistent. So this remains throughout the childhood if nothing is done about it. And lastly, I want to just call your attention to something called entropy that we measured. And you don't have to know the details about it, but just the color code in the blue, which is very low entropy, is the neonates that are preterm, and in the yellow scale is the neonates that are full term. So when you are born full term, there is a better chance that this entropy in the signal is broader, and that means that you're ready for welcoming all the sensory input and making sense out of it, as opposed to having a very narrow range of variations. The curve there is from over 233,000 babies from the state of Florida. We didn't have access to the diagnosis of autism there, which is just preterms and full terms. Of what you can see immediately in the first 2 months of life, week by week, is that these babies that were full term, and I'm plotting here females and males, had a sharp decrease in the delays, and the preterm were flat, no changes in the delay. So if you don't do something about it, it's not gonna fix itself. So that means that a very early intervention that shifts these delays and broadens the bandwidth of information is really critical. Okay, in conclusion, there is information buried in this gross data that is currently discarded. So I invite people to think about that. The broad variability in latency ranges at birth at each peak is in the brainstem region. So normally there's broad variability, and that's a good thing at birth because it welcomes all of the information. So if it's narrow, it's not good. There are quantifiable delays, and with very little cost and effort, we can then transform a routine hearing test that is already being done into the earliest screener for autism spectrum disorders and all disorders, because if we do this study at a scale, we can then differentiate different subtypes of autism that now in the very large heterogeneous set of disorders that we have buried in this very broad diagnosis system. So this is a winner for us, for everybody, and I hope that people see that so that they fund our study. We're asking to do this at a scale in 3,000 babies, and it would be much like the growth chart that you have to build a growth chart of the brain stems and the latencies and the information buried in that variability that we're throwing away otherwise with wrong assumptions. So I'll take questions.
So questions, Alison, you must have one.
Dr. Torres, I just remember when I was trying to figure out what was going on with Garrett, we did take him to do a hearing test. And I just remember he wasn't able to do it. Like, it was kind of a nightmare. So I was wondering, have they changed perhaps like in like the last 20 years, the way they're actually implementing it so that all children could participate?
Yes, I mean, this is done, over 233,000 neonates, and then we got like 70 records from toddlers and children. So this is being done now routinely, and with means that are not uncomfortable or anything like that. And as a neonate, they just do it. Later on, they do it as well, at least in Florida and in the state of New York. Yeah, it's a routine test.
Is that the difference? Like 20 years ago, they didn't do it on babies? Because he was,
Yeah,
I think he was a
20 years ago, it wasn't being done. This is more like the state of New York gave us data since like the early 2000s that was readable. So
thank you.
Yeah.
Elizabeth, that's fantastic. I'm so glad that you can dig on this data. So for the New York, you are talking about hundreds or thousands of babies?
Yeah, they have, but so they have thousands of records, but we only had access to about 70 because a variety of reasons. We needed to report it on a paper, we needed to have the same machinery, the same kind of, and they had shifted different machines over the years because the technology obviously advances. And so also the same stimuli. And so we wanted to have these 3 levels of decibel. So it really, and then we needed to have babies that were tracked over time until they got the diagnosis. So that funneled down the data to just about 70 records. And then you know, it just declines the number of records that you can have.
Okay. No, that I'm glad to know that. My question is related to, I think, the last point that you mentioned, because it's such a heterogeneous population, right? And nonetheless, we're still seeing like something that is quite robust. How do you explain that? I mean, do we have any other either genetic or clinical information on those 70 cases? How similar are they? How different are they? Because I mean, you are getting something that seems like so fundamental early on that might be like common across different subtypes.
That's right. So excellent question. And that was one of the reasons why also the number of records that we were able to analyze decreased, because we wanted to have a balanced heterogeneous ethnicity. So we had to have all kinds of ethnic babies that we try to include. We excluded, for example, babies from drug users or or alcohol syndrome, or babies that came from a lot of trauma during the birth, things like that, we had to exclude. But the group here is very heterogeneous in terms of economic status and ethnicity. And we were able to do that because the state of New York has been doing this since the late 90s. So the records are quite broad. And we also had the motion data from the videos. So that's another reason why the number of babies was reduced. But that's an excellent point. That's why we need at least 3,000 babies so that we can look at, because it'll be about 2.something% of that is going to bring the diagnosis of autism. And then there is the other issue of females that you know that it's only like 4 to 5 males for each female. So none of these records with the diagnosis of autism has enough statistical power to get females accounted for. So that was another thing. We had to get enough females so that we knew we could compare across males and females. But these are all very good questions, but these are current problems with the diagnosis system that we have. And, you know, it'll be a challenge because 3,000 babies, you think it's a lot, but it's not because of the heterogeneous. Statistically, it's not. So, but it will give us a chance to subtype at least a few. So hopefully we submitted a grant to the NIH. And so we're hoping, fingers crossed, we get it and we can do it. Yeah.
All right. Thank you very, 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 very much. We're going to go to the Understanding Diversity in Autism Research panel now with Brad and Connie. I don't know how we get wired up here. Sorry, we're a little bit late wherever you are. Leanne, are you going? Okay. Just have to keep on saying sorry, sorry, sorry. The only wisdom we can hope to acquire is the wisdom of humility. Humility is endless. Sorry. T.S. Eliot. Ah, there you are. Good to see you. Can you hear me?
Yes, we can.
All right. Well, in that case, you don't really need me. Why don't you? Brad? Are you going to be the introductory? Connie, are you going to do that?
Connie. I will. Thank you, Roger. Hello, everyone. Thank you for joining
Dr.
us.
Constance Smith-Hicks will now introduce the panel of wonderful people, 3 of them from Kennedy Krieger. If you ever watch LinkedIn, you'll see how many posts there are about Kennedy Krieger Institute and what wonderful work it's doing. And we've also got Omar Shanta as well there. So off you go. Connie.
Thanks, Roger. Hello, everyone, and thanks for joining us for this panel discussion. It's titled Understanding Diversity in Autism Research. I'm Connie Smith-Hicks. I'm a child neurologist and neurogeneticist. I direct the Center for Synaptic Disorders at the Kennedy Krieger Institute. and I was the former medical director for the Center for Autism and Related Disorders. I'm an associate professor of neurology at Johns Hopkins, and my research efforts are directed at expanding the clinical phenotype of neurodevelopmental disorders with known genetic etiologies. And I'm also focused on developing outcome measures and quantitative metrics to support not only the care of individuals, but to help facilitate clinical trials. Joining me today, as you heard, we have Dr. Bradley Schlager, Dr. Ebony Holliday, and Omar Shanta. Dr. Schlager is a pediatric neurologist and developmental cognitive neuroscientist. He's the president and chief executive officer of Kennedy Krieger Institute, Baltimore, Maryland, and he holds the Zanville Krieger Faculty Endowed Chair. He's also a professor of neurology and pediatrics at Johns Hopkins University School of Medicine, and he directs the NICHD-funded Intellectual and Developmental Disabilities Research Center at the Kennedy Krieger Institute, as well as the NINDS-funded Child Neurologist Career Developmental Program, supporting the early career development of aspiring child neurologist physician scientists. Dr. Schlager's research efforts are directed at understanding the development of the brain's functional network architecture in typical and atypical developing children. Ebony Holliday is a research scientist at the Kennedy Krieger Institute's Center for Autism and Related Disorder. She is a school psychologist by profession, and Dr. Holliday's research and clinical interests include early identification and assessment of social communication differences and autism spectrum disorder in young children. In addition, she is interested in family-centered interventions that are inclusive and culturally responsive. Her work also centers around the training and professional development of early care and education providers who serve young children with developmental disabilities and delay. And our final panelist, Omar Shanta, is a PhD candidate at UCSD. He studies bioinformatics and systems biology. He's a member of the Copy Number Variant Working Group of the Psychiatric Genomics Consortium. He conducts his research in Dr. Jonathan Sabat's lab, and he focuses on copy number variation in people with psychiatric disorders. Congratulations, Omar. I heard that you'll be defending your thesis in the spring. It's awesome.
Thank you.
So today we'll be talking about diversity in autism. And as you know, diversity is a hallmark of autism. Yet research in autism does not capture the full heterogeneity of individuals on the spectrum. Some misconceptions about autism, and this may that all autistic individuals have the same skills or difficulties, that they are all nonverbal and unable to communicate, or that they all have savant abilities and are either violent or aggressive. The statement, when you meet one person with autism, you've only met one person with autism, speaks to the heterogeneity of this condition. And today we'll spend some time divining both the diversity in autism, but in addition, the current state of diversity research in autism. And in addition, we'll end with a discussion of the roadblocks to include individuals of diverse backgrounds and abilities as participants in autism research. And we'll also try to touch on some of the strategies that one could use to address some of these challenges. So Ebony, we'll start with you. You know, I like to say that autism is an equal opportunity heterogeneous condition. It affects all races and ethnicities equally, yet differences in prevalence rate exist. So what are some of the factors contributing to the difference in prevalence rates, both at the global, national, and regional level? And to what would you contribute the delay in the diagnosis of people of color?
Thank you. Just want to say I'm happy to be here today and kind of joining in this conversation with everyone, as well as my fellow panel members. So thank you for that. So as we know, there have been some longstanding differences in the prevalence rates of autism, especially among individuals of different races and ethnicities. And when we think about that, also knowing that those prevalence rates for people of color are often lagging behind other individuals, and particularly individuals of white backgrounds. And so when we think about that, I think some of the good news is that we've made progress in recent years. If we think of some of the recent data from CDC that was released last fall, we know that we've made some efforts in closing some of those gaps, those race and ethnicity gaps that were there. However, it is still something that we need to focus on. So while overall we see the prevalence rates as 1 in 44, so we're familiar with that statistic, that definitely varies, and in some areas varies widely depending on the site or the state within the United States. So I think globally, or when we look at that big picture, I think we have made some progress, and that can be due to some of the outreach efforts, I think, that have been made over the years in certain communities of color. Different training efforts, I think, among clinicians that have been going on, different initiatives. So I think those areas are definitely benefits and pluses that are there. But again, I think we still need to do some work in that area with making sure that gap is closed even more, and particularly looking at different states. So while, as an example, as we know, the rate may be 1 in 44 in the United States, if we look at a state like California, that prevalence rate is 1 in 26. So if we're seeing that type of wide variability, that's also going to have some implications within race and within ethnicity in those areas. So again, I think we do need to kind of focus on that area, look at what else we can do to make sure that if there are specific areas, specific states, looking at regions, to make sure that we are identifying individuals and children that should have diagnoses and making sure that they are able to access the services and the supports that they need for that. When we think about the reasons that these differences might exist with race and ethnicity, I think it could be a variety of areas, a variety of factors. One is looking at access to services. So access to quality screening, quality surveillance, referrals that are there. We know that there are differences in referral rates. We know there are differences in referrals to early intervention programs. And those are impacted by race and ethnicity. So that's one factor that could be present. But also perhaps looking at under recognition of symptoms of autism, attributed it to something else, to other conditions, to other types of developmental areas or co-occurring conditions that might be present. So that's another factor that could be there. When we think about other areas and other factors, it could be biases. And so not necessarily things that are direct, but like implicit bias could be impacting clinicians, could be impacting the way in which we refer, the way in which we screen. So those are some issues that might come to play. And then also when I'm thinking about our communities, when I'm thinking about communities of color, there may be within group differences, there may be cultural differences that are there to play. There may be more of a wait and see approach in some cultures. There might be some distrust of the healthcare system in some ways. And so I think there is an interplay of various factors and it impacting clinicians as well as families, as well as individuals to either seek services or to also access those services. So those are kind of a few of my initial thoughts, but I definitely, am seeing this difference, and I think it is something that we definitely do need to focus on, and we do need to target as we're kind of moving along in this area.
Wonderful, thank you for that. You sort of touched on the idea of co-occurring conditions. And so, Brad, I'd like to direct this one to you. Psychiatric diagnoses occur, perhaps 10 to 41% of autistic individuals, and many of whom are reported to have at least one mental health diagnosis. So can co-occurring mental health diagnoses be separated from the autism? Or do you think it influences the manifestation of the autistic features?
Well, at the surface, it does seem that diagnostically, one should be able to pull apart autism from, say, ADHD or generalized anxiety and so forth. And I say that because the diagnoses are based on clinical criteria that are intended to, as much as possible, draw clean delineations between these diagnostic constructs. But the reality, underscored by the frequent co-occurrence that you just mentioned, Connie, is that it's not likely to be easy to do that. And I think there's an interesting history here. I like to point out that not too long ago, one was not really allowed to diagnose ADHD and autism in the same individual. And I remember there were a number of fascinating studies from some years ago that showed that if you do a data-driven latent class analysis of the phenotypes of patients who, using DSM criteria, were diagnosed with autism spectrum disorder and ADHD, you could pull out subtypes of those patients who could be indistinguishable from each other, so that severe combined ADHD looked a lot like, essentially indistinguishable from autism spectrum disorder. Now, more recently, we are now allowed to make both diagnoses in the same individual, in the same patient. So that's an example of the pretty significant overlap in the diagnoses despite the criteria. But at a deeper level, the issue becomes, I think, say generalized anxiety disorder, does it look different in the setting of autism? And on clinical grounds, I think it does. And I think in general, neuropsychiatric disorders don't simply overlay on top of autism in the background. Instead, I think these conditions interact in complex ways. And that makes sense to me on first principles, because we're talking about brain function as the basis for those diagnoses. And it's not really an academic argument. It has real implications. So if clinical trials, for example, let's say pharmacotherapy or cognitive behavioral therapy for generalized anxiety, if those exclude people with autism, with an autism diagnosis, from participating in the study, we can't know whether the evidence basis for treating anxiety coming from that study will or will not apply to people who also have an autism diagnosis. And intellectual disability is another good example here. Intellectual disability is more common in people with autism than in the general population, and the converse is true. Autism is more common in individuals with intellectual disability, and that's the case for a number of reasons. But the same issue is in play here. If people with intellectual disability are excluded from autism studies or vice versa, and they frequently are excluded, we can't learn how the co-occurrence of those interacting conditions influence outcomes or response to intervention.
Valid point. Now, Ebony, I'm going to direct this one back to you. Autism and co-occurring diagnosis of ID. There is some reports that the greater incidence of intellectual disabilities in Blacks with autism diagnosis. We discussed earlier a little bit about the delay in diagnosis. Do you think that this is this delay in diagnosis, is it cause or coincidence? Is there any reason to think that the delay impacted the development?
Thank you, Connie. I think it's a really important and interesting question, and I think topic to kind of talk about for a bit. As you said, we do know that there is a kind of an increasing rate of ID, especially among Black African-American population and in children as well. And I don't know that the specific reasons are completely understood, but there is definitely some suggested conceptual thoughts and models of that. One of the things I wonder about is, kind of this cumulative risk that is there in some populations. And so when we think about access, so if that is an issue, access to services, if we're getting later diagnoses, later referrals, then that is going to imply perhaps later intervention, later treatment of symptoms. And when we think about that whole cascade, we know about neuroplasticity and how important early intervention is. And so when we think about all of that being delayed, what that cascade, could implicate, what those implications could be. And then we also know that there are just other risk factors that I think differentially and disproportionately impact Black and African-American individuals. So if we think of things like even just poverty, right? And we know the impact that poverty has on development, on language, on all of these other factors that are especially important in child development. And then we know that poverty rates are also disproportionate among Black and African-American populations. We know that also impacts at times preterm birth and low birth weight. And we know that is more prevalent among Black and African-American populations. So again, we're just seeing some of these perinatal risk factors. We are seeing perhaps poverty in some populations. We are seeing less access. And then when we put all that together, that cumulative risk may be something that impacts these rates that we're seeing, these increasing rates or these differences in prevalence among, I think, African-American and Black population when we're looking at autism. So I think those are some factors that we can think about. Is it this, cumulative risk? Is it one or more of these factors? I think there are definitely things that we can look at and we can consider as we're thinking about why these differences are persistent, why they are increasing in certain race and ethnicity populations.
That's rather intriguing. So, what it suggests is that there are multiple factors occurring in any individual that not only contributes to their clinical features of autism, but the existence of these co-occurring diagnoses. So Omar, the idea there is this high prevalence of co-occurring diagnoses, primarily psychiatric diagnoses or neurodevelopmental diagnoses with autism, suggests that there is likely shared etiology. So can you comment on the genetics of autism and the interplay between perhaps gene and environment? Ebony shared the impact of poverty and among other things. Can you tell us a little bit about that?
Yeah, there's definitely a lot of evidence right now in the genetic studies of a shared genetic etiology between different psychiatric disorders. Like I see it in my own results, in my own research, and also read it all the time in different papers from other studies. For example, like in one of the known risk loci in neurodevelopment disorders on chromosome 16, 6p11.2, you can see that if you duplicate a sequence at a specific place, then you see an increase of risk in both autism and schizophrenia. But then on the other side, there's, along with the shared etiology, there's a lot of loci like that, but there's also disorder-specific loci that are only present in autism or only present in schizophrenia. And so on the flip side, if you, instead of duplicating the sequence, if you delete the sequence at the same location, then it's only a risk for autism and not schizophrenia. So you can kind of see like these, it differs by location and what the genetic effects are on the specific disorders. So that's actually been really interesting to see. And on the comment of like, are people looking at genetics along with environmental factors, there actually was a data set that was released not too long ago called the UK Biobank. and they actually have a lot of genetic data for different disorders. And they also included, for the same people, they included a questionnaire so that they can actually provide different environmental factors for these different people. So geneticists are using that data set to answer a lot of these questions. So, and that data set has like about 400,000 individuals already. So it's a pretty good resource for research advances.
Very interesting. So in addition to your comments, there are these common variants that are identified out of GWAS studies and rare variants that are identified generally from whole exome sequencing done clinically. And it's been rather interesting to read the literature and think about the interplay between these two kind of variants, the more common ones that could perhaps explain some of the broader autism phenotype, and the rare, powerful ones that have been attributed to single gene disorders. And I often wonder, since we're not able to screen for common variants clinically, it would be nice to design a study that allows us to look at the interplay between the two in populations in general. And that leads us to our next part, where we're going to talk a little bit about the current state of diversity in autism research. We know that research takes many different forms. We have epidemiological studies that are directed at understanding the neurobiological basis, or the psychobehavioral features of autism. And the goals are generally to inform not just intervention, but policy. We've already discussed and acknowledged the diversity of individuals who participate in autism research. And we acknowledge that it doesn't really reflect the heterogeneity of the condition. At least in the United States, we have fewer people of color they're generally diagnosed older, and are often less frequently identified. So, I've been giving some thought to what might be some of the factors that contribute to the disparity of the individuals who participate in autism research. And so, Brad, I'm gonna have you take this one. And my primary question to you is, based on your experience as a developmental cognitive neuroscientist, how would you describe the demographics of the individuals who participate in autism research, and how does the heterogeneity of these individuals impact your approach to research, or the approach to research in the field in general?
Yeah, it's a substantial question, Connie. And as you've highlighted importantly several times, autism is highly heterogeneous. And so I'd like to take a step back and just say that the situation with inclusion and exclusion in autism research is really a microcosm of the general issue, I think, of inclusion and exclusion in clinical investigation per se. I don't mean clinical trials, but clinical investigation, patient-oriented or human-oriented research. And of course, domains of diversity include race and ethnicity, socioeconomics, sex and gender, and those are separable constructs. Sex and gender often used interchangeably, but inaccurately so. And of course, often not mentioned is disability and neurodiversity, and the exclusion of people with disabilities and other chronic conditions, especially cognitive, affective, mobility. These are long-standing and pervasive. It's a huge problem that ripples through clinical investigation per se. And I think multiple factors are involved here, including, say, kind of in a euphemistic way, the tendency for investigators to collect population samples of convenience. That's particularly true with smaller sample size studies. And by convenience, I really mean, individuals who are easier to recruit, easier to consent, easier to retain. And just thinking about that idea of samples of convenience, underneath all of that, I think all of those factors that Ebony just so clearly delineated earlier in this discussion, those are all underneath this euphemistic samples of convenience.
Mm-hmm.
But exclusion is also driven by the way we think about clinical investigation, where the idea of too much heterogeneity is thought of as noise or confounding factors rather than an accurate reflection of the true clinical landscape, the true biology, if you will. So these ideas of noise and confounding variables, they are thought to interfere with traditional study design, power analyses, interpretation of data. So there are active, overt steps taken in study design to try to narrow the variability and accentuate homogeneity of sample. That's on purpose. So we end up in autism-related research having study participants who in their daily lives tend to require less services and supports, or perhaps not at all, and who are therefore easier to recruit, easier to consent, easier to retain in the study. And so a consequence of that practice of exclusion, in addition to the ongoing marginalization of people through such practice, including in the concept of inclusion in investigation, is to have research that ultimately doesn't really generalize broadly to the population as a whole. Like it's problematic to say, for example, that a study that provides evidence basis for, say, heart disease applies to me if I have a disability or a condition that would have excluded me from participating in that study. Fundamental question about what evidence-based really means. One of the starkest examples, just to digress a little bit, is the longstanding exclusion of people with Down syndrome from Alzheimer's disease clinical investigation. Over half of people with Down syndrome will develop Alzheimer's as they age, and people with Down they're living longer because of improved overall pediatric care. And finally, just relatively recently, the NIH has shifted to support not only the inclusion of people with Down syndrome, but to also recognize that their inclusion bolsters significantly our understanding of Alzheimer's disease for people without Down syndrome, which is an excellent example of how all of us can benefit from greater inclusion and investigation. I'll leave it there.
Yeah. As I was listening to you, it reminded me that another challenge in autism research is that there's a tendency to focus on younger individuals. And individuals with autism are living longer. Granted, it's also a challenge to provide care for them, but lifespan approach to care and research is critical. So thank you for that. That was insightful. Omar, we have talked often about the idea or knowledge of autism has come largely from individuals of European descent, and I assume it's reasonable to expect that the same is true for autism genetics and the genetics of other psychiatric or neurodevelopmental disorders. So has that changed over time? And how does the lack of genomic diversity impact the results of studies done with these large data sets?
Yeah, there's definitely a bias towards European samples in a lot of the genetic studies, and not just in autism, but like across the board of major psychiatric disorders. And it has changed recently, actually, like within the past few years. People have recognized that there's a huge disparity between the sample sizes of like 500,000 Europeans versus like 30 or 40,000 Africans within a study. And then usually what happens when you publish a paper And you see this kind of huge disparity is that the papers say, okay, these results are representative of Europeans because most of our samples are European. And it doesn't necessarily generalize towards other populations. And the reason that it doesn't generalize is because of the heterogeneity, actually, and because people are different. So their genes, because their genes are different, they have different characteristics, right? So according to those lines, like if you have a mutation within one population, but that locus is a heterogeneous locus and it's different in different population, then it can have different effects on someone from a different population. And we won't necessarily see that if we only have Europeans inside of a study. So that's why it's very important. And actually, right now, the disorder that's added African samples most recently is major depressive disorder. And they've been able to show some similarities, but also some differences between the risk loci inside of major depression disorder. So that's where it is right now.
Excellent. Brad, how does knowing the genetics of autism, how does that factor in to research questions and research design? For example, you do a study is done with, I don't know, hundreds of individuals with autism, but the genetic etiology is variable. Some may have known genetic risks. Others may have it, but it's not yet been identified. So how do we factor that in to research question and design?
Well, it's a complicated space right now because for the reasons that you stated and that Omar just stated, we're sort of in a nascent phase for understanding background, really, beyond European ancestry data. Autism describes a set of identifiable, observable traits. These are traits that are manifestations of brain function, and they're likely to be the result of a wide variety of factors, including neurogenetic. But just because those phenotypic traits converge, it doesn't mean that the mechanisms by which those traits in a given person that they're shared. It doesn't. You can't conclude that. There can be dissociation between overt phenotype and the underlying multitude of mechanisms, complex interactions of multitude of mechanisms. And just to underscore what Omar just laid out so nicely, a variant that produces an autistic set of traits in someone with European genetic ancestry may not necessarily produce the same phenotype in people whose genetic ancestry is African or non-European. Different neurogenetic factors are likely to drive different developmental processes, and they may emerge as comparable overt phenotypes, but the neurobiology can be different. So those differences really could be influential on how certain interventions are or not effective in a given patient. So understanding those neurogenetic drivers and how they interact with genetic background, as well as environmental exposures. This is not a nature nurture kind of argument. This is a nature by nurture, like interacting, cascading interactions across development, across the lifespan, understanding that complex biological landscape. That will very likely inform us of how to more appropriately design studies and trials, most effectively. Again, we haven't really touched on this, but the part and parcel of the focus on heterogeneity is the recognition that our clinical trial work should be focused on individual people. And ultimately want to get to individualized, precise treatments. So understanding all those factors, I think, I would assert, will make it possible for us to have wiser study designs that are more likely to yield better information for individualized decision-making for patients.
Thank you for that setup. It takes me to Ebony, where I'm curious to know what's the current focus and direction of implementation science in autism research?
Thank you for that question as well. So in our work that we're doing specifically in the Center for Autism as well, we are looking in early childhood, and some of that we are finding, taking the principles of implementation science has really been essential for making sure we're translating this work from research, from the research lab to the community. And implementation science is really just that study of the procedures and the strategies that we look to and that we can use to make sure that we're translating and that we're scaling research and putting it in the hands of the community. And the community might be, it might be an organization, it might be a community agency, it may be schools, it may be directly into homes with families. But it's really looking at how do we decrease and how do we shorten that that research to practice gap that we know can be like about 17 years, right? When we're getting, so the research that we know, when it actually gets into practice and gets used, there's unfortunately that 17-year that's there, or that no do gap that we typically use. So implementation science is a strategy and a principle, and it's really a field to better target that and to decrease that gap from research to practice. So it's really, and it can be used with any population, any intervention, but it's really that study of what do we need to do and how do we effectively and efficiently translate this research, our basic research or research that we're doing in the lab that works well under certain conditions with certain resources, with certain funding, and how do we translate that to the real world? And so when we think about autism, there definitely has been a push in the field with autism interventions and treatment. And my area and focus in my population is typically children and early childhood. So that's kind of where I talk from. But when we think about what can we do to really make sure these interventions that we're finding are effective and that are working, how can we make sure that these are then translated, for example, in a preschool setting, or how can we make sure in early childhood that parents and that caregivers and caretakers are able to implement these strategies that we know can be effective. And part of implementation science theories and conceptual models, a lot of times there are phases and there are stages in that. And some of the first few are really what we call exploration and planning. And so part of that really takes into consideration who is the population, what really are the issues, because at times as researchers, sometimes, you know, we may feel like we know best, this is what you need, this is what this is what will work best for a community. And implementation science is really about getting buy-in to a research, and so, or getting buy-in to treatment and getting buy-in to interventions, and really partnering with the people who are our population. And not us kind of coming in with a package and saying, here, you need to implement this, but really partnering and saying, this is something we designed. What do you think about it? You know, what changes might need to be made from this control trial that we've done until it goes into the classroom or until it goes into schools? What changes, what considerations do we need to take into consideration? Is this feasible to do? Because something that might be feasible in an RCT might not be feasible for a general, you know, teacher to do, for a parent to do, or for a community agency to do. And so those are things that we take into consideration in that planning stage and in that exploration stage of taking this nice research package or intervention that we have, and again, saying, what do we need to do to make sure that it can live in the community, that it can really do what we want it to do, so it's not just housed in academic institutions and not just housed in research journals, but that it's actually making a difference in that day-to-day. And so that is part of what we're doing you know, in autism and individuals that are using implementation science to really inform interventions that maybe we've proven and that we've shown are evidence-based. How then can we take that and translate it? And the last point I'll make is, you know, I think it's really important then to make sure that the research that we are doing is generalizable, is representative, because again, it's going to be hard to take it to scale for a larger population if it isn't inclusive in what we're doing. So that is always the first step, is to make sure that our research is inclusive in many of the ways that I think we've all talked about, especially Brad talked about many good points with that. And I absolutely agree with all those points that were made. We really need to make sure that our research is inclusive, that it's representative, and then we can think about taking it to scale, translating it for different populations in different communities.
Great. Now, is it reasonable to think that challenges with the generalizability of the research plays into the disparity in the distribution of services within the school system? Or are those two separate issues?
I definitely think there's an interplay of those that are there. I think, unfortunately, we do find that the research that we have is often on, for example, middle-class populations, may not include many children, individuals of color as well in the autistic community. So I think then we have that research, and while it may work, and it may be a fact that it is effective and it is evidence-based for everyone. But if we don't have an inclusive population, we don't necessarily know that for sure. So we need to make sure that, again, that research is generalizable, because then what happens is often when we have research and we're showing and we're saying that it's evidence-based, often then that may get changed into policy. So policymakers are going to make note of that. Often schools will do a global uptake of an intervention, and it's going to be in place for 10 years, in a school. And so if that research is not generalizable, if it actually was not inclusive of disability level, of cognition, of race, ethnicity, of all of those factors we've talked about today, I think it may be a fact that it may not be effective for everyone. And then we see those gaps, whether they are in race and ethnicity or gender or sex, we see those gaps perhaps widen. So again, I do think it's an interplay. I think we need to make sure our samples are inclusive in multiple ways. And then we need to really be mindful about how we are translating those, or making sure we are translating those in partnership with the individuals who will be implementing it, and or who our target population is.
Great. For the remainder of our time together, I want to touch a little bit. I know we've sort of discussed this a bit, but I want to give us an opportunity to talk a little bit about roadblocks and strategies for inclusion of autistic people from diverse ethnic backgrounds and abilities, socioeconomic status, you name it, in research. Brad, you commented on the inclusion, or rather the exclusion, of people with intellectual disabilities and psychiatric diagnoses from autism research. And you spoke to some of the challenges that results in this process. Are there additional roadblocks that you would want to mention at this time? And what are some of the strategies that you think we can use to become more inclusive?
So I'm going to unmute, and for you Baltimoreans, we got motorcycles coming right down the street. So just, wait a second. Okay. So so just taking a step back to what is the goal of clinical investigation, broadly speaking, and I think that has a lot to do with the exclusion approach in autism research of people with co-occurring psychiatric disorders, of co-occurring intellectual disability, and so on. And broadly speaking, the objective of clinical trials and or clinical investigation, and the coin of the realm in clinical trial investigation is the randomized clinical trial. It's really about studying not an individual, but the central tendency of, and in principle, a well-defined group of people, and how well that well-defined group of people responds to some intervention. And if that study can show a shift in the central tendency that's statistically reliable with a clinically meaningful effect size, then it gets elevated to evidence basis, evidence that intervention can be effective. So these trials tend to hone in on certain inclusion and exclusion criteria intentionally to assure that they have the cleanest sampling with the best likelihood of getting signal from that intervention. These studies are not designed to then understand which individual will or will not respond to the treatment. It's really whether the treatment will affect the central tendency of the group. And so this push to precision or individualized medicine must go hand in hand with a push for broadening inclusion criteria and the development of methods that we learn how to embrace and capitalize on heterogeneity of people. And that includes disability, chronic conditions, neurodiversity, in addition to race and ethnicity, sex and gender, socioeconomics, genetic ancestry, and so forth. We have to learn how to incorporate all that information. And so it's critical of the RCT, admittedly so, but in order to get to the next level, we have to recognize that that approach is intentionally not working towards individuals. And so within autism research, these issues are very much in play. And I think that to get to the strategies and barriers, we need review panels for studies who focus on seeing that clean sample of autism without co-occurring conditions like ADHD and anxiety and mood and intellectual disability, epilepsy, and so on. We need those panels to shift their thinkings, because in the real world, autism co-occurs with all those conditions in the real world. And so if clinical investigation involving autistic individuals excludes those, well, the work doesn't reflect the real world situations we face. And we will continue to make incremental at best progress and not have generalizable information. So very commonly studies addressing autism exclude specifically for intellectual disability or people who have the most significant forms of behavioral challenges that we heard about earlier in the conference, or for people who require significant support such as alternative and augmentative communication, as just some of the examples of the arguments for exclusion. But clinical investigation must reflect all of us in order for the work to benefit all of us. So we need review panels that critique study designs, and that includes IRBs to support these notions of expanding inclusion criteria whenever possible. And then funding and regulatory agencies need to pay attention to these issues if the intention is for the research work to ultimately benefit all of us, we have to shift into this next level of investigation. And we need to find ways to understand how to use the richness of data that comes from this diverse, heterogeneous large sample, like the kind that Omar just described a moment ago. Those really rich samples, we need to learn how to not shrink them down and squeeze out the variance, but capitalize on it, again, so we can get to large effects at the population level and then individual level characterization so that the work can ultimately be applied to individual people and patients.
As I listened to you, it made me think about the need, while being more inclusive, to within the larger group have ways of stratifying. And that would hopefully help us. We're going to have responders to some intervention, non-responders. What are some of the common features? And that will hopefully help us as we move towards precision medicine.
Well, I think stratifying is the bootstrap to get to individualized. So I know what I'm saying sounds just sort of grandiose, but we need to get there. And so I think the next step is to have in earnest real stratification that takes into account this information, and to not disparage allowing data to drive some of that stratification, and not have a top-down, we already know going in what the strata should look like, but to allow data to help us sort that out. we now have massive amounts of data and we have computational capacities to deal with it. We need to embrace that next level of analysis and learn how to do it.
Thank you. Yes. Okay, so implementation scientist, any thoughts about way forward, current challenges? What are your thoughts, Ebony?
I think it's both excitement for way forward. I think there are a lot of I think there's a good outlook, but I think there are also barriers to kind of take into consideration, especially with implementation science, and also pulling upon principles when we think about research, community engaged research. And that is somewhat of a similar field, but looking at how we get the community, how we get our populations, our target populations, to also participate with us in this. And so it's not just a top-down researcher, this is what is prescribed, but it's really making sure that we're hearing from individuals, what do you need? Does this sound like it is something, that is effective? And then being able to really create research together. That's not always going to be possible for every field and for every area, of course, but for those intervention programs or treatment areas where that could work and be feasible, I think it's definitely something to take a step to. And I do think it is a mindset change, perhaps for researchers. And I think this plays a lot with what Brad was saying, which I absolutely agree with. Looking at, I think, broadening inclusion criteria for research, but also thinking about, I think, the multiple factors that are at play. So looking at reviewers and, and individuals being more open as you're reviewing journal articles, as you're reviewing things that have been submitted, taking into consideration, what is this population diverse? Because I feel like there may be some barriers to researchers feeling like they can broaden those criteria, because is this something that's going to be then accepted? Is this something that's going to be in line with what reviewers are looking for? So I think there are those push-pull factors that may be in place. So I think having that mindset change among everyone, I think is going to be something, it's a big plan, it's a big take, but I think that's something that we should really strive to do. With implementation science and with community engaged research, again, it's really looking at that partnership. It's really looking at saying, what can we do together? So we are at our center, we have established some community and academic partnerships. And so we meet regularly with them, whether it's biweekly, whether it's monthly. We have community advisory boards where we sit down with individuals from the community who are invested in the work and the research that we're doing. And they give us really honest and genuine feedback about some of our ideas, things that we've done, things even looking to research flyers, and sometimes we give that information out and we get information back that we haven't even thought about that really makes our research better. Or they're giving us ideas about recruitment strategies that we may not have thought about. And so those partnerships with the community and those partnerships with other institutions in the area, we have found those to be invaluable in some of the research that we're doing. And I think it only makes your research better when you include more individuals, we have more buy-in. We are doing some stakeholder analyses. So what that means is we have our group, we started with maybe a group of about 6 people, and then they referred us to other people who referred us to other people. And, we have this network now of about 40 individuals in our regional area who know about our research, who either met with us, once or twice, who have done some type of a participation. And so when we think about how we scale up research, how we are translating ideas into practice, into the hands of the community, I really think we have to do some outreach for that. And so those are some of the steps that we're trying to take as it relates to implementation science. How do we take these ideas and really make sure that they're in the hands of the community, really make sure that they are feasible, that they are acceptable to individuals, and that ultimately it's something that can be sustained over time. We want sustainability. We don't want it to just be, an 8-week intervention that we do and then it's done. We want to make sure that it's something that can sustain over time. So we are using those principles of just exploring, planning, and really working together in partnership with populations. Again, I definitely understand that's not possible with all types of research, but with the work that we are doing and that I'm doing with, with schools, with providers, with early care and education providers, with families, those are some things we definitely can take into consideration, and they really help to make our intervention and our research more meaningful.
So Connie, can you hear me?
I can. Thank you.
Okay. The word has come down from on high that we must move on quite shortly. So that was a fascinating conversation. I have one tiny question at the end. I was just looking at this paper in Autism Research. I'm looking at in terms of what is the practical issue, which you almost answered in the last one. Practically, how does one do this? In other words, that paper by Anila De Mello and from John Gabrielli's lab, exclusion of females in autism research, empirical evidence for a leaky recruitment to research pipeline. Despite efforts to increase the representation of autistic females in research, studies consistently enroll small samples of females or exclude females together. You've seen the paper, you know the paper. Maybe this is something for you to talk about, Brad, but what does one do there? If you're sitting in a lab, or sitting at home, or sitting in a group, and you've read the paper, and it turns out to be really good and accurate and so on, what do? Do you march down the street? Do you go and talk to your congressman? I mean, do you try and get Nature to do an editorial on this? I mean, what's the practical steps that can be taken in these sorts of issues?
Well, I think what we are doing right now is about as practical as it gets to elevate this topic to make it part of a substantial and significant part of an all day long discussion about autism and autism research, to make sure that it is front and center, or maybe at the tail end of a long day. But but that's practical. The letter to Nature, the concern expressed to the editors of the journal, so that maybe the editorial practice changes to require studies that don't acquiesce to the sample of convenience sort of excuse, and request that studies that get submitted for publication actually have real representation. Yeah. So those are practical. If it's happening at a conference level where we're talking about it and encouraging others to think deeply about their own work, if it changes editorial practices at the publication level, especially high profile, if it influences the way study section thinks about study design, all of those are practical ways to shift. It's not going to happen overnight, but these are practical ways to shift the culture, I think.
Also, to add to what Brad was saying, to also help answer the question, I think another practical change is accountability. And what I mean by that is, like, right now in the consortium that I'm a part of, this year we added a subgroup called PGC Africa that's focused on getting samples from African populations in Nigeria and Ethiopia. So just making that subgroup makes people accountable to collect more samples from African descent. And also statements like the future goal is to include more diverse samples. Like those kind of statements will also increase accountability.
Yeah, and Ebony, your answer really did address this, your last answer, which was very clear, I thought. Lovely answer. And Connie, thank you so much for the questions you thought through and asked there. It was the other thing, of course, that we can do, that people can do, is make sure that we tell them where this video is, and that they can watch it and listen to all you smart people having discussed it, and then they can change their minds. Thank you all very much for being there.
Thank you. It's our pleasure.
And we can't do a 15-minute break, apparently. You can do a 5-minute break for bathroom break, 5-minute bathroom break, and then back in here. Thank you so much.
All right, everyone, maybe we should get started, making sure we have her slides here. So it's my pleasure to introduce Maria Montenegro. Dr. Montenegro is actually a colleague from Brazil, and she's came in from the same university as I got my degree in biological sciences, University of Campinas in São Paulo. She's joining as a clinician, a neurologist to the Rady Children. So she's helping us with the neurological division over there, and her focus is on epilepsy. I think for this community, lots of families actually were consulting with Dr. Nespeca, who retired recently. And I think Dr. Montenegro is filling the gap here. And most of your families, if you have someone with autism and epilepsy, would end up talking to her at one point. So it's a great pleasure to introduce her to the community and having her to explain a little bit about the intersection between autism and epilepsy. Again, for the families who have autistic individuals suffering from epilepsy and seizures, myself included, this is a very hard condition. So having someone who is very knowledgeable, who can teach us, who can talk to us, who can explain what are the options, what are the different types of seizures, different types of treatments, it's really helpful. So please help me to welcome Dr. Montenegro. Thank you.
Thank you very much. It's a pleasure to be here, and I'm really excited to start working at Rady Children's and UCSD. Today, we're going to talk about epilepsy and autism. And as you know, it is a common comorbidity, and the prevalence of epilepsy in the neurotypical population is 0.5%, which it's considered very high. It's a common disease. But in children or even adults with ASD, it is 5 to 38%. And this frequency varies so much because the ASD includes several different conditions in the same group. And the frequency of epilepsy will vary according to the comorbidities associated, like cognitive impairment, motor deficit, and other disorders. And that's exactly what we're going to talk today to try to separate three main groups. So we use the term autism spectrum disorder to diagnose kids according to DSM-5, but we have a lot of different diseases that can have autism as part of the symptoms. And this is why epilepsy has such a wide range of symptoms in this population. So it's not an academic division, but to try to understand and explain a little bit better, I am proposing to separate three different groups. The first group are those that has autism as the primary diagnosis. That would be the classic autism, textbook autism. Those kids that they don't have a genetic syndrome, they don't have a brain malformation, a perinatal complications. Autism is the main symptom. The second group are those in which the main diagnosis is a known neurological condition. They also have autism, but they also have cerebral palsy, a brain malformation, some genetic like Angelman's, tuberous sclerosis. So autism is part of the diagnosis, but not the main diagnosis. And the third group is a rare one, those with epileptic encephalopathy that also have autism. At the end, they're going to understand why we have to talk about this small group. In these groups, epilepsy is the main diagnosis, and the discharges are so frequent that the brain does not work well, and the kid may have cognitive impairment and often autism. So the first group are those that autism is the main diagnosis. In this group, remember, we had 5 to 38%. In this group, we have 2 peaks of epilepsy onset, usually in the very young ones before 5 years of age, and also in the late teens years, kids that are already in the teenage years, and the prevalence of epilepsy in this group is only 12%. That means that almost 90% do not have epilepsy. And usually, if you add a genetic syndrome, the prevalence of epilepsy will be much higher. This is a graph just to try to explain what's going on. It's not based on actual numbers, but as you can see, you have autism, and we can add some comorbidities, and the frequency of epilepsy up to 60% that some studies suggest. In this group, we have 12%, which is very low. And the type of seizures that we've seen in this group, it's focal, it's multifocal, generalized. There's not a specific type of seizure associated with autism. And it's usually well controlled by anti-seizure medication. That's a very important information, because I know epilepsy and seizures are very scary, but usually the kid will have a few seizures, and once we start the medication, seizures will be controlled. We are talking about this specific group. But the most important issue in this group is the EEG. If you get an EEG in people that never had a seizure in their lives, you're going to have 1 to 4% of abnormal EEG. So you can have an abnormal EEG and never have a seizure in your life. If you do this in patients with ASD that never had a seizure, the frequency varies. Again, the studies include different types of patients. It varies from 6 to 61, but most others agree with 30%. And the type of EEG discharge can be generalized, focal, multifocal. There's no specific pattern. But the most important thing is that an abnormal EEG does not correlate that the patient will have epilepsy. And we should not treat the EEG. If the EEG is abnormal, we should not treat it if the patient does not have seizures. This is very important, because once you got an abnormal test, sometimes it's tempting to do something about it. And during this talk, we're going to talk about, well, you shouldn't even have gotten the test, because it's highly experimental. I know there are studies trying to explain, oh, we should treat the EEG, but it's not used in clinical practice. So what does an abnormal EEG means in a patient with ASD? We don't know. And neurologists are very comfortable saying that we don't know. We don't know a lot of things, and that's okay. But what we know, we do know that people with autism, 30% will have an abnormal EEG, but only 12 will have epilepsy. So when we ask a test, it's supposed to bring us answers. And in this case, sometimes it gets in the way of the diagnosis. So many patients with an abnormal EEG will never have seizures. So EEG is not recommended as a routine baseline evaluation if the patient ever had seizures. Just to remind ourselves, like, how do we diagnose epilepsy? Epilepsy is a clinical diagnosis. You need recurrent seizures to have epilepsy. You don't need an abnormal EEG. The EEG helps to characterize the type of seizure. And the type of epilepsy is the combination of the type of seizure, the frequency of seizures, and the EEG. So if I know the type of epilepsy, I can choose the medication more specifically for that patient. And if you have spasms, you can have ACTH and so forth. So this is very important, because if you get an EEG in a patient with epilepsy, it will be really helpful. If you get an EEG and the patient never had a seizure and it comes with some discharge, you just don't know what you're going to do with this result. If the patient has some funny spells, some staring spells, strange movements, and you don't know if it is a seizure, sometimes we ask the family just to make a home video with a cell phone. And if you're an epileptologist, usually just looking at the video, you can tell, oh, this is a seizure for sure. Or, oh no, I'm sure this is not a seizure, but if you still have some concerns, the test that the child should get is a video EEG, not an EEG. And that's important because a normal EEG does not rule out, does not exclude the diagnosis of epilepsy, and an abnormal EEG does not confirm the diagnosis of epilepsy. This is the first group, those with the classical autism, with autism is the main symptom. Now we're going to talk about when you have a neurological disorder and also have autism. And this is a very, very wide group. You can have genetic syndromes like Angelman syndrome, Down syndrome, patients with brain malformations, tuberculosis, stroke, meningitis, perinatal complications. And just to frame this group a little bit better, for instance, this kid, he has 6 years old, and he has cerebral palsy, also cognitive impairment. And he had 2 seizures at age 1 that were controlled with anti-seizure medication. The MRI shows a bilateral malformation, and at age 3, he was diagnosed with autism. So the brain malformation caused the motor deficit, the cognitive impairment, the epilepsy, and also the autism symptoms. Another example, this girl, she's a 12-year-old. She has cognitive impairment. She has autism. The diagnosis of autism was at age 2, and she started having seizures of age 9. Remember that's the second peak. And MRI was normal, but she has a abnormal whole exome sequencing. In this case, the genetic mutation caused the cognitive impairment, ASD, and epilepsy. And the last one, just to remind you that it's a very, very different group. You have a girl with 8 years of age, and she has TS. And she also has cognitive impairment and autism. Her seizures started at age 2. They are very difficult to control. She needed epilepsy surgery to remove this large tumor. And the TS will cause the cognitive impairment, ASD, and epilepsy. So what these patients have in common? Almost nothing. And that's the problem when we're trying to establish the frequency of a comorbidity in this group. Epilepsy has a very wide spectrum because it includes a very different group of conditions, and seizure onset can be before or after the diagnosis of the autism. So the severity of epilepsy is associated with the condition you have. This is very important because in this group, if you have tuberous sclerosis, we know the natural history of the epilepsy. If you have a specific mutation, if you have a brain malformation, so we just can't say, oh, in this group of patients, 33% will have this, 12 will have that. We have to think according to each specific diagnosis because it's going to be very, very different. So again, if you go back to this graph, just remember, as we add comorbidities, we increase the frequency of epilepsy. And sometimes the family kind of complains to us. They don't mean to complain, but it's a complaint that, well, my son had cerebral palsy, he has epilepsy, and now only at 5, he was diagnosed with autism. And I know it's quite frequently to happen because in this group, sometimes it's also our fault. We are focused more on the main diagnosis. So symptoms of autism may not be really clear at this age. Symptoms sometimes are only clear after the higher demand of social interaction, especially at school. And before 2013, with the DSM-4, we almost had to pick one diagnosis. We're not used to add diagnoses. So the children, the we have to say, oh, she has ADHD or autism. Now you can add, and that's very important because as we add the comorbidities, it's easier to tailor the treatment for each patient. And the last group, that's the epileptic encephalopathy. Epileptic encephalopathy are conditions in which the epileptic discharges, they are so frequent because the brain cannot work well and there is cognitive and behavior impairments that are produced by the discharge. When you're doing an EEG, every page, there's several discharges. So it's very, very, very frequent. And we usually see developmental delay, cognitive impairment, and autism. So the most common epileptic encephalopathies are those that I think everybody is familiar with, but West syndrome, Lennox-Gastaut, Dravet, epilepsy with myoclonic-tonic seizures. And the natural history is that child that has he met the normal required developmental milestones for a few months, sometimes even years, has a seizure onset and developmental regression after that. And autism is frequently one of the symptoms associated in this group. Another interesting thing is that epilepsy is the main symptom. It's going to take usually a few months, sometimes years, to be clear the diagnosis of autism. Seizures are usually drug resistant. We try to treat them, but no medication works. But if you're used to that, you're going to see that if the seizures get a little better, the symptoms get a little better. The child functions better. And at some times when he's having a lot of seizures, the kid will walk slower, talk slower, because there is this, it's really an electrical abnormality going on. But we are talking about epileptic encephalopathies, especially because of this one condition. And that will report us back to the very beginning of the talk. The epileptic encephalopathy with spike wave activation in sleep. Now, this is since February, that's the new name, but it includes the ESES, that is electrographic status epilepticus of sleep, CSWS, Landau-Kleffner, that I think most people have heard about, and atypical benign Rolandic epilepsy. We have a asymptomatic period where the kid met all the required developmental milestones, and then epilepsy that is usually easy to control, behavior and language regression. And the EEG is almost normal when the child is awake, and it's really, really bad when the child goes to sleep. And the difference is that although there's some language regression, usually there is good social interaction. The child wants to communicate, sometimes usually use signs, and there's no restricted interest and no stereotypes. But this is the EEG, almost normal. This is very abnormal during sleep. And why we're talking about this? Because this is the only disease that if you treat the EEG, the child gets better. If you start anti-seizure medication and you achieve some improvement in the EEG discharge, cognition and language will improve. Very, very rare. But I know that there are some pediatric neurologists that they kind of hope that, well, if we treat the EEG in a child with autism, we're going to improve the symptoms. This has been tried and we can't do that. And this is very important because those medications have side effects. So language regression can be a confusing symptom. Sometimes people think, oh, it might be a Landau-Kleffner and epileptic encephalopathy with spike wave activation during sleep. Well, sometimes I know it's hard, but if you get an EEG during awake and sleep, it's very easy to make the diagnosis. But again, don't do this if you know it's not a Landau-Kleffner syndrome, because the differential diagnosis can be based on clinical signs. And that's the three groups that we're supposed to talk. And there's a bonus slide, because I've never forget about the adults. Just one slide, because the message is easy. It doesn't change much. This study is very nice. It got some adults only with autism, and the frequency of epilepsy it's very close to 12%, the same that we see in kids. But you have adults with autism and cognitive impairment, the frequency of epilepsy goes up. And adults only with cognitive impairment, it's a little bit less. So again, as we add comorbidities, we add the frequency of epilepsy. Just to summarize, in the group 1, autism is the main diagnosis. Seizures are not very frequent and easy to control. Group 2, sometimes you have seizures and autism later, sometimes first autism and then seizures later. It's a very wide range of symptoms. But in the third group, we have a lot of seizures, and later the child is diagnosed with autism. The take-home message is that I don't like to say like 12% have epilepsy. I'd like to say like almost no one will have epilepsy. Almost 90% will not have a seizure. And if they do, epilepsy is usually well controlled with medication. Drug-resistant epilepsy will be associated with other underlying conditions. And video EEG, that's the gold standard. If your child has some funny spells, don't get an EEG. Go for the video EEG. So we have to capture the event. And EEG should only be done if the patient has seizures. Remember that 30% will have an abnormal EEG, but only 12% will have epilepsy. Thank you.
Stay here. I mean, I'm happy to take questions here, and I might actually start. Sorry, Torres, but involves you as well, because I want to know your opinion about the CBD. But also, I mean, lots of families talk about the silent seizures that they cannot see. But there are, I think, in the past years, I would say, some comments about that some of these conditions have the silent seizures that you don't see it, but it's there only with the EEG, but because you don't have any reason to request the EEGs, you don't see it. And I remember lots of families coming asking about those things. If you could comment on these two topics.
So 2 parts, CBD. I'm going to talk about CBD and epilepsy. I don't know much about CBD and autism, but everybody knows that CBD is effective for epilepsy since the 80s. Why it's not a big deal since then? Because we had a lot of different anti-seizure medications that started to be developed after the 80s, and the side effects are much, much, much lower than CBD. CBD works, yes, but they have a lot of side effects. So in my opinion, it's not the first option. You should try the more conventional anti-seizure medications because they are safer, but CBD should be considered in patients with drug-resistant epilepsy. And when I say CBD, it's Epidiolex. It's not the homemade or some more natural approaches, because the Epidiolex does not have THC. And I know that maybe THC works a little bit better, but THC can trigger some psychiatric disorders. And we are talking about a very high risk. So I'm a little bit scared. The second, the second question. The silences, subclinical seizures, very rare, but they do exist. And it's scary, but it shouldn't be. I know when we say, oh, seizures, it's a frightening thing. I know it is. It's ugly. But you're not going to lose a neuron when you have a seizure. And it's very, very, very unlikely that it will lead to something really bad. And the silent seizures are usually very short, and they last for 20 seconds, 10 seconds. there will be no brain damage because of these seizures. And nobody knows what it means. Nobody even knows if you should treat it. It's different if you're gonna have a seizure that lasts more than 5 minutes. 5 minutes is the magic number. So if you have a seizure that lasts more than 5 minutes, there is some evidence that it might trigger some permanent neuronal damage. Less than 5 minutes, which is a lot. Most seizures last less than 1 minute. So we don't want to have silent seizures. We don't want to have subclinical seizures. But I wouldn't worry, and I know some people do that, oh, every patient with autism should get a 24-hour EEG to make sure there are no silent seizures. Remember, there's a chance of 30% that the EEG will come abnormal, and you just don't know what to do with this information. But the family will be really scared. So I'm really not really worried about that. Oh, another important thing. I've never seen a patient have only subclinical seizures. Usually they have some clinical ones. So there's a little clue like, oh, those are the ones that maybe he's having seizures, staring spells. Sometimes you say, oh, they are silent, but there's a staring spell. There's some mild symptoms. If you bring the kid to the EMU, like the monitoring unit, and you see that the seizure, sometimes we can explain to the family that's what we're looking for, and it's very helpful.
Yeah, so we must see different populations of patients because I have seen, I'm over here, seen clinical, subclinical seizures with children with autism. That is only on the EEG and not clinically, but I would really like to comment on the issue of the EEG and autism, because that's a longstanding point of contention between epileptologists and neurodevelopmental disability specialists. And people who deal with neurodevelopmental disabilities, and the literature is now, more and more frequently showing this, that people are realizing that seizures or possibly even not seizures, but severe EEG abnormalities may have an effect, I shouldn't say do, may have a negative effect in terms of cognition and or behavior in autism. And so, and there are a lot of people who routinely do overnight EEGs as part of the as part of the workup for children with autism because of that. And there are people who do treat them in certain circumstances, in particular when there are very frequent spike wave discharges during sleep. They have not been diagnosed with epileptic encephalopathy, but their EEGs pretty much look like that. And it wouldn't have been picked up if the EEG hadn't been done. So there is room for discussion about that.
I agree. Yeah. What I think, if the EEG is horrible, it's different. I'm talking about sometimes you have a couple of discharges, you have to report it as abnormal. What I think that probably happens, we just can't measure what's going on, because in the discharge, you have the spike and wave. The wave is the repolarization. So I'm sure that group of neurons are not working well. And the question is, a few discharges, I'll just let it go. But if it's very frequent, the 85%, I don't think we need that much to consider, at least discussing with the family, what's the next step.
All right, I think we should move on. Let's thank again, Dr. Montenegro. Thank you so much. And we are moving to the next speaker, Irui, who is a colleague here at UCSD from the biological sciences. He actually was a student here at UCSD, so I'm very glad to see that his trajectory. He went for a postdoc in the East Coast, but is smart enough to come back and become a faculty here with us. And you heard in the panel that sometimes, I mean, we discuss or we study other syndromes or other conditions to gain insights about autism. People who have been in this meeting for several years know that we had like lots of speakers talking about Williams syndromes or other syndromes that might give us insights about autism. And here is not different. So Hirui would talk about Down syndrome and how we can extract information, perhaps copy what he's doing for Down syndrome to understand better autism. So please welcome Dr. Hirui. Thank you.
So how do I switch it? There we go. Good. Thank you so much for the wonderful introduction, and it's wonderful to be back in San Diego, and thank you all for coming. Today, I want to share with you some of the work that we've been doing on Down syndrome and how we took a roundabout way to end up at something that is very unexpected. So one of the things that we are now finding is that in individuals with Down syndrome, there's this aging-like phenotype, which is known as senescence. And this is something very abnormal. Usually when we say senescence, we talk about aging, Alzheimer's disease, neurodegeneration, not development. So I want to first start by giving you a brief description of the clinical features. I'm sure most of you know about Down syndrome, but Down syndrome is a multi-organ disorder where there's varying degrees of penetrance of the different organs. However, the brain is one of the organs that's most vulnerable to triplication of chromosome 21 or Down syndrome. Nearly all individuals have some level of intellectual disability. There's development delay of the brain. And there are other features as well, such as craniofacial features that are unique to individuals with Down syndrome, and that's also penetrant. My lab is really interested in the brain and how it's impacted by intellectual disabilities. So just to give you a brief introduction, the adult brain is composed mostly of 4 different cell types, which is the neurons is what we mostly talk about when we think about when we talk about the brain, which is the functional cells of the brain. And then you have the supporting cells of the brain, which are astrocytes, which are shown at the bottom right corner here. These are the supporting cells. They provide nourishment, oxygen. They connect the neurons to the blood vessels. And then we have oligodendrocytes. These are the cells that basically myelinate or put wraps around the neurons so that they don't have misguided communication. So this is how these cells basically have directed communication in a very strategic way. And then we have microglia, which are the immune cells of the brain. They surveil the brain and ensure that everything is working properly. And interestingly, all of the 3 main cell types, which is neurons, astrocytes, and oligodendrocytes, come from this cell type known as neuroprogenitors. These are the developmental cells that give rise to the functional cells of the brain, basically. And in Down syndrome, these cells have been shown to be dysfunctional that lead to hypocellularity or reduced volume of specific regions of the brain, which include the cortex and hippocampus, which are the regions that are required for learning and memory, as well as cognitive processing. And these are regions that are very vulnerable at early stages of development. And at the cellular level, a lot of studies have looked at postmortem brain tissue samples, as Alison was mentioning this morning, that there's dysfunction in cell proliferation. So during the mid-gestation period, cells that have triplication of chromosome 21 do not divide as fast as the euploid counterparts, or the cells that are typically found in neurotypical individuals. In addition, there's altered differentiation where cells that have neuroprogenitor cells that have triplication of chromosome 21 generate more glia and less neurons. So changing the content concentration also alters the function of the brain in adulthood. And the third thing that we found is that there's deficits in migration. The brain develops at different increments, and different cells have to migrate to different layers of the cortex. And this is the cognitive part of the brain. And when you have triplication of chromosome 21, we don't have proper migration to the different regions. While the cellular level and organ level manifestations of Down syndrome have been somewhat well characterized, the molecular mechanisms underlying this dysfunctions are still not very clear. So in 1959, Jerome Lejeune figured out that Down syndrome was caused by triplication of chromosome 21. And since then, a lot of studies have been focused on identifying specific genes that could be leading to this disorder. And we've been very unsuccessful. But with the advancement in next generation technology, studies have been finding that triplication of chromosome 21 not only disrupts the genes on chromosome 21 that you see here as the higher barcodes, we observe that there's genome-wide disruption in gene expression. So this includes chromosome 1, 2, 3, and all these other chromosomes, and not just chromosome 21. Which is interesting because we've been always focused on the upregulation of genes on chromosome 21. And as Alison mentioned, my training was here. I was trained as a structural biologist. So when I first started my postdoc, I tried to think of it as a structural problem. So my initial hypothesis was, if you were to think of the nucleus as an elevator full of people that's at capacity, and in Down syndrome, there's this additional chromosome that's trying to come in. So how do you accommodate this additional person? My hypothesis at the time was every chromosome in that elevator, every person needs to reorganize themselves to accommodate this. And as a consequence of that moving around, then the gene expression genome-wide is changing. And this has to be somewhat of a structural problem. So this is the idea that we began with. So just to give you a brief introduction of how the genome is organized inside the nucleus, we have 23 pairs of chromosomes, and you all know that it's about 6 feet of DNA is what we have in every single cell. And that 6 feet of DNA is wrapped around these proteins known as histones, and it's all squished up into this very, very small structure known as the nucleus. So it's basically 1/100th of the width of your nail. So decades ago, we used to think that the genome was just randomly distributed inside the nucleus. But what we've been finding is that every single cell type has a very specific organization of the chromosomes. And this is known as chromosome territories. So for example, here we show different colors where purple is one and blue is another chromosome. And we can say that this is, as an example, found in fibroblasts or your skin cells. And if we were to look into neurons or let's say heart cells, the organization of each chromosome in space is going to be very different. So, and then if we were to take a cross section and just look at how every chromosome is interacting with each other, is thinking of each chromosome as a person, basically, there are two types of interactions. One is the interaction that the chromosome will have with itself, which is known as cis interactions, and then there's chromosomal interactions in between chromosomes, and this is known as trans interactions. So how can we understand the consequence of having 3 chromosomes on this global 3D genome architecture? So we utilize stem cells, and Alison is an expert and has talked extensively about this, but we utilize stem cells derived from different individuals, and we generate the different cell types independently instead of having them harmoniously to create an organoid. And the reason we want to do this is because we want to understand the specific consequences at each level of development where you have either 2 or 3 chromosome 21s. So far, we've been building a larger cohort of individuals, but for this first study, we utilized 2 unrelated individuals, a male and a female, that only have 2 chromosome 21s that are shown on the left side. And then we matched them by sex to have of cells that were derived from individuals with Down syndrome. Additionally, we utilized mosaic individual samples, which is there are 3% of individuals with Down syndrome are mosaic, where they have both trisomy and euploid cells within their body. So we have these cell pairs where the genetic background is identical, allowing us to eliminate in-person variation. So we've generated induced pluripotent stem cells, which are shown here on the left side from both euploid and trisomic individuals. And then we differentiated these induced pluripotent stem cells to neuroprogenitor cells that are shown in the middle. And then we karyotyped these neuroprogenitor cells to ensure that the cells that we're generating are either euploid or trisomic. that's shown here, that the bottom cells have 3 chromosome 21s, and then the top ones have 2. Next, we looked at the 3D genome architecture. I'll show a few maps, and I'll try to give you a brief introduction, but I'll try to do a summary of what our findings are. So as I said, there's significant differences in cell types. So as we go from an induced pluripotent stem cell state to a neuroprogenitor state, there's significant alteration of how chromosomes are interacting with each other and within themselves. And here what we're showing is that the red indicates that neuroprogenitor cells have gained interactions as compared to stem cells. And then the blue indicates that neuroprogenitor cells have lost the interactions in between chromosomes. So each box represents a chromosome 1 through 22, as well as the sex chromosomes in both the horizontal and vertical. However, when we look at the same sort of distribution in transitioning from stem cells to neuroprogenitor cells with trisomy 21, we observe significant reduction in interchromosomal interactions or interactions between chromosomes. So neighboring chromosomes are no longer interacting with each other in neuroprogenitor cells. So if we were to animate this, what we are observing is that with the addition of this 3rd chromosome, all the chromosomes are becoming introverted, where they spend more time interacting with themselves than they are with their neighbors. And what we observe is that these changes in the 3D genome architecture are actually directly associated with the genome-wide alterations in gene expression. So this was very exciting. However, it was interesting because induced pluripotent stem cells or stem cells don't have this feature. It's a very unique neuroprogenitor feature. And so we wanted to see if anybody else in any other circumstance had actually observed phenotypes that look like this. And in 2015 and 2016, several studies came out, or a few studies came out, looking at the 3D genome architecture in aging cells or senescent cells. And what they found was similar features to what we were observing in Down syndrome. So this was very puzzling to us. And here it's just showing you the interaction increased short range versus lost long range. The green line or the teal line represents neuroprogenitor cells. And in the studies, the red line represents senescent cells. So it's very similar features that we're observing here. So senescence, as I've mentioned, is associated with aging, and it was first described in 1961 by Leonard Hayflick. And the way the study happened was Leonard Hayflick and colleagues were really interested in understanding how many times can a cell divide in a dish. And they identified that it could only divide for about 40 to 60 times before it exits cell cycle, which is one of the key features, becomes anti-apoptotic, the cells don't die, and this feature was then labeled as senescence. And since then, several studies have found that there's significant alterations to the different organelles in the cell, and then that they release the senescence-associated secretory phenotype, which is toxic to the tissue microenvironment, leading to more or aggravated aging in the neighboring tissue. So this is not only bad for the cell itself, but it's also bad for the tissue entirely. And one of the interesting things is that a lot of these organelle dysfunctions have also been observed or have been classified as hallmarks of Down syndrome. This includes dysfunctions in mitochondria or increased DNA damage, which is part of the nuclear feature. And one of the main key features is early exit of cell cycle or loss of cell proliferation, which is associated with the developmental brain delays that are observed in individuals with Down syndrome. And since 1961, studies have found that there's numerous ways that you can induce senescence. This includes, in addition to aging or replicative exhaustion, oncogenes or oxidative stress or radiation and chemotherapy can also introduce senescence. And this is how resistant cancer seems to be developing. So we wanted to understand if it's actually globally, the genome-wide disruption in gene expression that we see, is it similar to Down syndrome? So we capitalized on this data that's publicly available. So we performed RNA sequencing on all the samples that we had. In addition, we took all the published RNA sequencing from all these different modes of induction of senescence. And interestingly, what we observe is that only neuroprogenitor cells, which are shown here as the last 3 rows, are very similar to the signatures that are observed in senescent cells. So overall, if we look at some of the classical markers such as P16, P15, P21, we see that neuroprogenitor cells are entering senescence. However, induced pluripotent stem cells are not. So this seems to be a time and age and cell type specific problem that we're observing with neuroprogenitor cells. And globally, this feature is very similar to oxidative stress. And as I mentioned, individuals with Down syndrome have higher levels of mitochondrial dysfunction. So there is this intimate interplay between the nucleus and the mitochondria that we're now starting to work on. So the next thing we wanted to do is to try to confirm if actually our neuroprogenitor cells are becoming senescent or they're just showing features of senescence. And we observed that neuroprogenitor cells that harbor trisomy 21 are in fact becoming senescent. So as I mentioned, senescence was first described in aging. So the field has done exhaustive work to identify anti-senescent or senolytic drugs to treat various aging-associated disorders, which includes Alzheimer's disease, Parkinson's disease. And there's been clinical trials, actually there's clinical trials ongoing for Alzheimer's and Parkinson's, and there's published data looking at diabetic kidney disease disorder. And the combination of dasatinib, which is a tyrosine kinase inhibitor, and quercetin, when individuals are given this combination, this drug can not only remove cells that are in deep senescence, removing these toxic cells, but also can ameliorate some of the molecular features that are disrupted as a consequence of senescence. So we wanted to see if we can utilize these drugs to alleviate some of the features that we are observing. And so we treated our neuroprogenitor cells with the senolytic drug combination of dasatinib and quercetin for 5 days. And then we looked at various cellular and molecular features that we see. So the first thing that we wanted to see was if we could remove these deep senescent cells. And so we looked at a marker for senescence, which is P16. And what we observe is that this increased senescent cell population as a consequence of trisomy 21 can be removed from our culture system after treating for 5 days with dasatinib and quercetin. And I just wanted to remind you some of the features that are key to the learning and memory disabilities observed in individuals with Down syndrome is associated with hypocellularity of the brain, which is associated with deficits in proliferation, altered differentiation, and deficits in migration. So we performed RNA sequencing to see if we can alleviate some of these features from the genome-wide disruption. And after 5 days of treatment, we were able to alleviate 54% of the genome-wide disruption that we observed as a consequence of trisomy 21. And some of these alleviated or ameliorated genome-wide disruption included pathways or genes involved in cell migration and cell proliferation. So we performed cellular assays to see if we were able to alleviate some of these features. And so we stained for Ki67, which is a marker of proliferation or cells that are continually going through cell cycle. And what we observe is that the trisomic harboring neuroprogenitor cells have significantly reduced cells that are in cell cycle or proliferating. However, after treating them with the dasatinib and quercetin treatment, we see that we're able to not only alleviate it, but even slightly higher trends of proliferation. Next, we devised a protocol to actually assess cellular migration. So we allowed these neuroprogenitor cells to self-aggregate into 3D, and then we embedded them in a scaffolding protein and allowed the cells to migrate out. And what you observe is that the top panel is showing cells. Each green dot that you observe here represents a single cell that has migrated out of this neurosphere. This here core is where the neurosphere began. And as you can see, there's a lot of cells that are migrating out from there in the euploid or the control samples. However, in the cells or neurospheres that were generated from individuals with Down syndrome, we see significant dysfunctions or loss of cell migratory capacity. And this is really crucial for brain development, as a lot of the cells are originating from one region of the brain and they have to end up at another region. So this is a very significant feature that needs to be ameliorated. And then after the senolytic drug treatment, we're able to alleviate some of these dysfunctions that we observe. So with that, I would like to just give you a brief summary that we went from looking at the 3D genome architecture to identifying that triplication of chromosome 21 or Down syndrome induces senescence. And we can alleviate some of these abnormalities that are introduced by triplication of chromosome 21 by utilizing the senolytic drugs or dasatinib quercetin treatment. And we're currently working on trying to identify more robust therapeutic avenues to treat some of these dysfunctions that we observe in neurodevelopment. But in addition to looking at this, one of the hypotheses that we have, which is, as I mentioned in the beginning, is that having an extra genetic material might be altering the 3D genome architecture. So in addition to looking at the other aneuploidy disorders, such as Edwards syndrome, which is caused by triplication of chromosome 13, and Patau syndrome, or 18, and Patau syndrome that's caused by triplication of chromosome 13, we're also interested in expanding into copy number variation. So we're looking at AP syndrome and also other copy number variations that are associated with autism. And a large majority of the individuals that have copy number variation, which is either the duplication or deletion of certain regions, also have intellectual disabilities, as well as some of the key features that are observed in individuals with Down syndrome as well. So there's a great convergence between all of these different genomic imbalance disorders. With that, I would like to thank my group and my previous postdoctoral lab, where a lot of the work that I shared with you today was done. This wouldn't have been possible without the generous support of our different foundations. I'd like to also thank our collaborators that provide us with the clinical samples, Dr. Brian Skako and Dr. David Switzer at Mass General Hospital in Boston, as well as Virginia Commonwealth University, where we get all of our aneuploidy patient samples. And I'd also like to thank the organizers for having me today, and I'd like to thank all of you for taking the time. I'm happy to take any questions.
Right. Fantastic talk. Questions? Maybe I should start. I mean, you mentioned the CNVs, and I'm glad you are working with the 8P as well. So the CNVs, I mean, it's very different from an aneuploidy where you have the entire chromosome trying to find its way on the cells. So I'm assuming that the level of rearrangements that you have in the chromosomes are way more subtle, right? So that's what one would expect. But I think my question is overall, I mean, considering all the CNVs, how much duplications and deletions in small pieces of chromosomes can actually affect the overall transcriptional profile? I'm saying that because I mean, we study Williams syndromes in the past as 7q, deletions and duplications. And when we do like a global gene expression, we don't get much. When we do see alterations in genes that are nearby the duplications or the deletions, but overall it's not like a lot that changes. I wonder if you have similar experiences.
Yeah, that's a really great question. So for the CMVs that my lab is actually focused on are these really, really large ones. So AP, which is about 20 megabase, as you know, which is the size of chromosome 21. So it's comparable to Down syndrome, basically. We work with Pitt-Hopkins syndrome, which has about 30 megabase deletion. So we're losing something larger than chromosome 21. And we're also working with Dup15q, which is a very weird sort of chromosome abnormality, but that is also, you know, larger than 20 megabase in size. So we're working with these larger, you know, massive amounts of gene sort of disorders. And yes, I agree that the smaller the size is, the more tolerable it is to the cell. And nature has already done this. So when we look at the trisomies that actually survive, we only observe that the smaller ones in size and gene content are the ones that actually survive to birth. So that's why we see Edwards, Patau, and Down syndrome, and not really chromosome 1 or chromosome 2, you know, triplications are not allowed by nature in general. But we do have all the aneuploidies just to get a fundamental understanding of the basic rules. But our lab is really focused on the larger scale ones. But the smaller duplications, you know, it becomes really tricky because the size is small and then there's very specific genes and maybe there's local alteration of the topologically associating domain. This is the structural unit of the genome. And so you might see a very concentrated alteration as a consequence of those CMVs. But, this is something we're all exploring, and we're really excited to work together on this.
Got it. Yes.
Thank you for this great talk. It was fascinating. I'm wondering, and maybe there is no connection, but individuals with Down syndrome often show signs of early aging that have been described as Alzheimer's-like. Are the kinds of things that you're describing in senescence in cells sort of, can you follow that to a point where you're really looking at this other feature that happens much later in life? Because that would be fascinating.
Yeah, absolutely. That's a very fascinating question. So right now, we have multiple avenues that we're pursuing. One is, as you mentioned, Alzheimer's disease. And so, APP, which is one of the main genes associated with Alzheimer's disease, is located on chromosome 21. So every individual that has Down syndrome will get at least Alzheimer's pathology by age 35. So this is a guarantee. However, the dementia phenotype and when it begins is actually sort of on a spectrum. So we're considering that, how much neurodevelopmental senescence you had might also be indicative of how early of the dementia will kick in. So we're utilizing different mouse models. So we have mouse models with aneuploidies, and we're trying to track all of these properties. But yeah, so far we actually don't know. Thank you.
I have another question related to the senescence part. And the question is related to the telomerase. Do you think that the lack of telomerase activity can actually lead to such alterations in chromosomes? And I'm saying that because we are growing now brain organoids at the International Space Station, and one of the features that we see in space is the lack of telomerase activity. And once we bring the cells back to Earth, somehow, we don't know the mechanism, but telomerase seems to realize that he's not been doing like a good job and overcompensate. So we have this acceleration of aging when you bring the cells back to Earth. So I wonder if that type of acceleration via telomerase will actually lead to any chromosomal abnormalities inside the cells. I don't know if anybody explored that link before.
Yeah, so that has not really been observed, but what we do know is that there is excessive telomerase activity in, for example, Down syndrome. One of the key features, as mentioned, the aging phenotype has always been quantified by the telomere deterioration that's happening in individuals with Down syndrome. So there've been these longitudinal studies looking at telomere length and telomere integrity, and they find that individuals with Down syndrome have a much faster deterioration of that system. Whether that telomerase overactivity is the reason that these individuals have trisomy 21, that is actually not known, right? We don't really understand why individuals or how aneuploidy forms both in the sperm and in the egg is not really very clear. There are multiple ways, multiple genes, multiple pathways. You can induce oxidative stress or manipulate Wnt signaling and you'd get aneuploidy. So, but in all of these individuals, we actually don't know. But that would be a really great thing to explore, and we can definitely do this in our models.
No, that's fantastic. And I think, researchers like that will not only help with neurodevelopmental conditions, aging, but also cancer, which is another condition that we see lots of chromosomal aberration as well. So, Hiroi, thanks so much. Thanks again. Nice presentation. Thank you.
Yeah. All right, so let's assemble the last panel then. Please.
Yeah. This chromosome, is a microphone be there, or each one has one, right?
Yeah, it looks like it. It looks like it does. All right.
So I'll leave this here. So in case
Or we can have a conversation. Like, what do you think has been the most interesting thing you've heard today?
Everything. Everything.
idea, which I hope you'll explain. This paper. The Canvas Rebel paper that Otto did. Are we going to, or Shelley, can you, somebody can explain Otto's paper or whatever? Or Otto can. See, I mean, what's interesting is that one of the interesting points you make, Otto, in the paper is about the amount of extraordinary ability you had to influence once you decided to do that, once you decided to put the information out there, despite the fact that it was typing and so on and so forth. And I was thinking back exactly 100 years to Hollywood. Aldous Huxley, Brave New World, had just arrived, was visiting. So are a couple of other interesting people. T.S. Eliot came along as well. And the one thing that they both, most of them that didn't like Hollywood, they didn't like Hollywood because they were somewhat intellectually arrogant anyway. But what they did like was Charlie Chaplin, simply because he didn't speak. It struck me as kind of ironic that we got into a situation like that.
It's okay. Most people don't like me until I speak.
I'm thinking that in talking about that, Charlie Chaplin was the original multimodality communicator.
Right.
And he used so many other means of expression that is only recently being acknowledged by these incredible young people, that being acknowledged by researchers in science and Hollywood itself.
Yeah.
So I love that analogy. Yeah.
I think that, yes, I think the point I was trying to make, not being cute or witty or anything, was that you can say an awful lot
Yes.
with your mouth shut. If you have the right way of doing it, and clearly he did.
Yes.
I mean, you know, there was just an enormous career. I mean, anyway. So do you want to rock and roll with this?
I will. Welcome to, I think, the last talk of the evening. This is the Living Autistically panel. And last year was the first year that we actually included this. And I'll discuss that a bit. It was conceptually a little bit difficult in how to approach the topic that we're interested in, given the nature of what we're talking about, and I'll be a little more specific about that. But we're going to go through a number of different scenarios. Conceptually, the Hollywood connection and such will make sense because we've tried to use that as the one thing that could kind of bring these things together as best as we could. I thought it was actually probably a little bit different than that in that Hercules being part of Hollywood or a normal part of Hollywood. And you think about the trials of Hercules, and I think about our children that go through many more trials probably on a daily basis. But more interesting, that's his wife, Hebe, who is the goddess of adolescence and immortality. And as a wedding present, if you will, gave him eternal adolescence. So not only did he suffer through the trials, but he was also perpetually an adolescent, which may be a good thing, maybe not so much. I work at UCSD and Rady Children's Hospital, and also a member of Autism Tree. So just getting back to the book club program, this was started in August of 2020 and was essentially a group of typers and books and trying to discuss books at a very sophisticated level. It became much more than that because we were dealing with typers and people that communicated in a very different fashion. So being online made a great deal of sense when it came to this type of a book club. This is something that was interesting to me. In May of 2022, UCLA was happy to announce that a man with autism was the first nonverbal graduate of UCLA. When my son was applying to college, UCLA wasn't that interested in people that were on the spectrum. And even if they pretended to be, didn't have the resources to help those kids anyway. So it's nice that this is happening. My fear is this is more about UCLA than that individual. But despite that, I think it's a move in the right direction. This was last year. So a year ago, this was the panel, the inclusion of typers, and basically discussed a book, Flowers of Algernon. And it's just taken up. This is Pretty Things, a novel by Janelle Brown. This was a group picture. Basically, this is a book that now has moved on into becoming a TV series. Otto makes things happen. I'm not quite certain how he makes things happen, but he just makes things happen. So here he's meeting with Anthony Doerr, Pulitzer Prize winning author, managed to mention the book club and ask him if he could take part in it, which he did about a week ago. His Club Cuckoo Land book was exactly what they were discussing. He actually pulled out of his pocket a map of the landscape and the setting of what his book was based upon, which was absolutely fascinating. And his involvement was fascinating, but even the involvement, which you can kind of see on the right side of the continuous interplay of feedback and such by the typers. And I really enjoy one of Otto's comments: "We are changing the world one letter at a time." So what I'm trying to do is combine disparate groups to increase our understanding. So there's not going to be any conclusions made here. It's not that type of thing, but it's to express an understanding of what the issues may be, what some potential solutions are, and where the problems are. So to do so, I wanted to identify the question. In my surgical background, we don't ever identify questions. That's more from my PhD work, where you try to identify a question. The question is, and this was a nightmarish question for, I think, anybody who has a child on the spectrum, is not in the short term, how is this going to impact my child, but will they be autonomous? Where will they be in 15 years, 20 years? You just don't know. And it is nightmarish for those of us that have had the opportunity to deal with it. So that's really the question. How do we identify autonomy, and what about these individuals in this work can help us further identify it based upon a really disparate group of people and a disparate curve. There is no normative kid or normative curve for the spectrum, and it makes it difficult. A personal joke I have with my son is you have those that are symptomatic and asymptomatic, and we call the asymptomatics the asses. So it's the spectrums versus the asses. But once again, this is to explore the issue, not to answer the question. So there's no doubt that adolescents may be an especially vulnerable period of development in autism, and this can make the transition into adult social roles and adult level of adaptive functioning very difficult. Life's hard enough, but there's something about adolescence in the milieu of a number of things that are occurring that can make this problematic. Some of the earlier studies that led to our nightmares were such as a study in 2009, that 2/3 of these kids fail to become autonomous as adults. And that's a large number. What you've noticed in the subsequent literature, that this number continues to go down and down and down. Obviously from greater public awareness, obviously from earlier intervention and more effective management. So what are the problems? Normative predictors of participation when you're talking about socialization, greater independence in activities of daily living, better socialization skills, and a greater number of services received. The current literature notes increased rates of several clinical problems in adolescents, with a significant increase potentially of anxiety and depression. There are numerous obstacles to our understanding what the best approaches to intervening in this population of patients are, given multiple diagnoses. And the potential for multiple benefits of treatment and multiple negatives of treatment. The biggest problem is that there's very limited age-specific treatment literature available. and an overall lack of research in adolescence in general on the spectrum. So the core symptoms of autism tend to abate to some degree in adolescence and young adulthood. And this is even more notable given the improvement in communication skills. Obviously, that represents a problem for a number of members of this panel. So as you get older, you have your decreasing core symptoms that are basically paralleled or exceeded by this improvement in communication. So all of these things are positives. It's rare that an individual will show enough gain that they'll actually move off of the curve or out of the spectrum. There's literature discussing what we call bloomers and kids that bloom off of the spectrum. But the fact is the characteristics are still hard. The socialization has allowed a lot of these individuals to avoid detection, if you will. But I'm not certain that anybody truly blooms off of the spectrum, and if there's even a need for such. Social impairment and repetitive behaviors tend to persist into adulthood, and not all individuals will show any improvement at all. Cognition tends to be relatively stable over time. A problem that we've always had is the fact that IQ is so closely identified with cognition, which I think is a big mistake, especially when we're discussing this population. So briefly, plasticity, we know that there are different periods of improved or enhanced growth, neurologically speaking, early childhood and adolescence being two areas of note. All of the research really concerns this first area and not so much the second. And all of our interventions are really directed at this first period and not the second. And it's essentially like gardening. I mean, that's what makes the most sense to me is periods of growth, overgrowth with refining of direction, of growth and pruning, a lot of pruning. And that essentially is kind of the pros and cons of plasticity, but I think it has an amazing impact. But what's important is I think this growth point during adolescence. ATPF has over 200 programs that deals with people along the entirety of the age range, which I think is amazing and was kind of an initial purpose 20 years ago. I always find that as kind of a satisfying anecdote, given the lack of understanding of this that many years ago. So my day job is dealing with brain tumors, and a lot of people like to talk about a two-hit model of dysfunction. The initial hit is disorders of neuronal migration, glial migration, disorders of cell differentiation, disorders of structural anatomy. So you're set up with this problem. The question is, subsequently throughout a lifetime, will you experience a second hit? In the case of a tumor, will you experience a second hit, which will create a tumor? Over basically the priming period of development and up through adolescence, the second hit in these kids is social demands. How do I get rid of that thing? There we go. Social demands, pubertal hormones, which are a difficult thing to remove, and neural reorganization. This is also kind of in conjunction with slowing down of the developing central nervous system, but a slow increase in CNS vulnerability. So there are many potential issues at about the time of puberty that are problematic and can lead to failure or speed bumps. So I talk about successful outcomes, the speed bumps, pubertal maturation, challenge of new developmental tasks, difficulty in developing intimate peer relationships, increased peer rejection, secondary depression and anxiety. The levels of risk-taking behaviors are different from normative populations, and it's hard to say what that means. And some problems with communication from the prefrontal cortex, allowing for cognitive control. And these are the speed bumps that essentially make it more difficult to reach what we are considering to be successful outcomes, which are limited. Formation of high quality friendships, acquiring autonomy from parents, forming romantic and sexual relationships. And all of these require complex reorganization of existing and underlying functional neural networks. But it's a much more complex problem that we really can address at this time. But briefly, we're going to try to do some of that here. So initially, I'm going to talk with Matt Dunford, the first member of our panel. And my first question is who is Matt Dunford?
Who is Matt Dunford? He was just a child from San Diego, California, born on November 30th, 1985. Birthday's coming up at the end of the month, so happy early birthday, me. He loved a lot of things in life, but first thing he ever loved was pirates. He loved pirates. In particular, he probably watched Peter Pan like 5 times a day. I don't wear hats anymore, but there was a period in time where I went around wearing a pirate hat every single day of my life, and I was obsessed with it, and I just could not be seen without a pirate hat anywhere.
You weren't the first.
Well, the first, yeah. And my favorite toys to play with were Lego pirates. And so Lego pirates were just my jam. Also, the Playmobiles had pirates, but they weren't as cool as the Lego ones. And I loved the Lego stories and just making, my own adventures with Captain Redbeard and the evil Imperials who were trying to tax the pirates. And I mean, come on, just give them the gold. Give them the gold. Just like, let them steal it. It's fun. And I had this encounter one day where my mom bought me a Lego pirate set from Toys R Us, and it had a comic in the back of it called Captain Redbeard and the Gold Medallion. And I was like, wait, these guys actually have their own story that like they follow. And so I was like looking through this, but I knew they were saying something in these little balloons and I don't know what. So I say, hey, Dad, what are they saying? Hey, Mom, what are they saying? And my parents had to read me this Lego pirate story something like 30 times a day. And to the point where, I think they just had to sit me down and force me to learn how to read in order to read this comic book so I could get my my little fix from Lego Pirates. And then in the year 1992, I was walking down the aisle of a Toys R Us with my dad, and all of a sudden I see the most beautiful thing I'd ever seen in my entire life. It was what we now know is a hologram cover to an issue of Amazing Spider-Man, which was celebrating his 30th anniversary at the time. And it was 30 issues of Amazing Spider-Man. And I just begged my dad to get that for me. And that was my first introduction in real comics. And so I became obsessed with this guy named Spider-Man. And I became a comic book fanatic since then. And comics are pretty much my passion, have been for the past 30 years. And I've done a lot of things that have led to this passion, and I knew that I loved them, but I didn't know that I had wanted to be, involved in them professionally. And it's led me into several roles over the years, such as being, I served as 6 years as board president of Little Fish Comic Book Studio, the comic education studio here in San Diego. I had also served as the editor-in-chief of Semantic Publishing and Keyleaf Comics. I had also moved ahead into an event called San Diego Comic Fest, which was started by the original founders of San Diego Comic-Con to create a more intimate, approachable atmosphere. And they appointed me as chairman of the event in 2017. And we'll be back in 2023 for another event coming soon. And my day job nowadays is I work at Upper Deck Entertainment up in Carlsbad, where I help oversee the Marvel comic trading card line, collaborating with artists to create these characters and bring to life these stories. Because as a child, comics were a lot more expensive for certain issues. It's a time before readily available graphic novel collections. And I hate that word graphic novel. They're comic books, not graphic novels. It's trying to dress it up into something. You don't always call a movie a motion picture. Don't be afraid of what it is, and don't be afraid of what you are. With these things, just, I learned about comic book characters from the backs of trading cards. And now that I get to do this full circle, like 30 years later, these very same trading cards that I learned all about all these characters from, I now get to play a process of that in my professional life. So that's pretty cool. So what is this? This right here is one of my old videos from another career path that I did back in the day. 10 things most people don't know about the BF-109. I used to work at an online publication called World War Wings, where I wrote basically clickbait about World War II all the time. And I thought, wouldn't it be cool if we started up a YouTube channel and just started doing stupid clickbait there? 10 things most people don't know about the BF-109. Is it the BF-109 or the MF-109? Like some people call it the Bayerisch Flugzeug. Some people call it and so these videos would accumulate millions of views. And I really liked doing them, but ultimately I had creative differences with the directors of the place, and I decided to ultimately leave World War Wings. And I told them if they don't change their direction, you'll be out of business in 6 months. But they proved me wrong. They're out of business in 4 months. So I really had a good time doing that, doing the World War Wings videos and articles. And I knew nothing about World War II history. And now it's at the point where I know everything about World War II history. And I mean, I can't even watch World War II history movies anymore because, like, even started becoming one of the most feared German fighters to encounter in a dogfight. That voice is annoying. 10 things most people don't know about the BF-109. Number 10, shaky landings. As dangerous as it was to other fighters in the sky, landing the BF-109 was incredibly dangerous for its pilots. With a very narrow landing gear and a heavy, powerful engine, these fighters were prone to tipping, making coming back to base tricky every time. As a fun fact, the most produced aircraft in history is the Cessna 172, with over 44,000 manufactured from 1956 to today. Yeah, so it's like dumb little clickbait facts over there. And I had a fun time doing that. And then, actually one of the first career paths that I actively wanted to do was getting into voiceovers shortly after college. So I brought back some of that and brought in this BF-109, World Fun Fact. But ultimately I learned about voice acting is competitive.
Who's this? Look,
and
you'll
who's
have
this?
to look behind you, but who's that?
I don't know. That's just our surprised guy. It's like, it's just like your standard YouTube reaction. Oh my God, I didn't know that fact. Because this guy looks like he would totally be in a World War II history. And of course, this is a wonderful moment right there with this, that is John Semper, who is the writer and showrunner of my favorite cartoon of all time, the Spider-Man animated series. And I thought I was a Spider-Man fan in 1992, but on November 19th, 1994, with the premiere of Spider-Man, the animated series, that shot my fandom into overdrive. Now, John is sitting here. This is at the 2019 San Diego Comic Fest, where John and I did a program called The Origins of Spider-Verse. Everyone had went out to go see the Spider-Man Into the Spider-Verse film, but I saw it at an opening night and I'm like, that movie was actually pretty good, but it wasn't original. So I told John, hey, John, I know you don't watch Spider-Man stuff after you did, but you should really see this movie. No, thanks, Matt. I don't really want to see it. No, you need to see it. You need to see it. And I finally convinced him, and he just says, they ripped me off. They completely stole my story and try to pass it off of their own. So we did a little panel there that said, he said when he was ending the Spider-Man cartoon in 1994, or in 1997 when it was ending, he felt that they had come out with so many toys that never made their way onto the show. And he always felt that kids would feel ripped off if these toys never made an appearance on the show. In particular, he's holding a toy called Web Armor Spider-Man, and it says now appearing on the Spider-Man animated series. But he never had plans to put that guy on the Spider-Man animated series. He just came out of nowhere as a toy. And he thought, wouldn't it be cool if when we're ending the show, we take all the Spider-Man action figures that we never used and then put them in an episode on the show where Spider-Man has to come together with these other Spider-Man from different universes to save the universe from ultimate annihilation from this sinister device at the hands of the Kingpin. Sound like any movies you saw recently? Yeah, but John did it back in 1997. And that was just a wonderful ordeal there. And this next picture of me that I see right here is at the 2019 San Diego Comic-Con when I was on the art during the Holocaust panel. I have another friend named Max Scheller who he goes around town to conventions, and people know him infamously as that guy with a Game Boy camera taking pictures of people with Game Boy cameras. But his mother was a Holocaust survivor named Ruth Sachs, and Max's mother wanted to put together a Holocaust exhibit in her honor called Remember Us the Holocaust, so Ruth. And she, Max had heard that I had done stuff in World War II history, and I had parted ways with World War Wings. And they said, could I provide some involvement in here? And I said, yes. And so I sat on this program talking about comic history during World War II and animation history during World War II. In particular, that scene right there is, I think, the probably best World War II comic ever done, called Magneto Testament, talking about the X-Men villain Magneto, where he got his intolerance for humanity and from the oppression that he faced as a Holocaust survivor. And basically he said, you know, mutants are going to be prosecuted and oppressed because it's happened before, it'll happen again. We need to get regular humans before they get us. And I think that is the most absolutely well done fictional story involving Holocaust or World War II history. There's only one disconnected moment. And in the attack on Poland, the Junker planes, the Ju-187s, are flying and bombing vertically. No, they don't bomb like that. They bomb horizontally. They are dive bombers. They go down like this, drop the bomb, and pull out like this, so the bomb continues to go in that direction. That's the only single moment that disconnected me from the story. Do you see how detailed I got in World War II history because of all this? I walked out of Dunkirk. I was like, this is wrong. 2/3 of these soldiers were from India. Why is it all white guys?
What is Matt Dunford's Double Ray Breakfast?
Okay, this is a cartoon done by my friend Jimmy Purcell when I was going around promoting the event San Diego Comic Fest, which is another event that I was going to do. And I'm dressed as Festivo, the mascot, who is designed by cartoonist Scott Shaw. And this is just because when San Diego Comic Fest is coming up, everyone knows that I will never shut up about Comic Fest. And this particular joke that Ray here is because in 2020, we were celebrating the centennial of Ray Bradbury and Ray Harryhausen, an icon of science fiction and an icon of stop motion film. And so the double Ray here. And in this particular scene, you see me with you see me at the Remember Us the Holocaust exhibit at the Chula Vista Heritage Society down in the Chula Vista Library with several of the local Holocaust survivors, and that debuted the opening of the first Holocaust museum here in San Diego. And this is one of my little fun facts because, you know, if you're on the spectrum, you're probably into Pokemon. And this was my World War II history thing. I made a cool observation here that no one had really ever noticed in history. I know in history you tend to beg, borrow, and steal, but I'm a big Pokemon fan. And these older Japanese biplanes, which had initially been painted with a red sun on them, they actually had a symbol. They actually painted them white at the bottom, white signifying death. And so in Japanese culture, older age, white is always symbolized by death. And so these biplanes were actually being used as kamikaze strike attacks because, I mean, rather than, you know, shooting bullets at a plane, you could at least cripple an entire destroyer just by crashing an aircraft into it instead. So, you know, sacrifice one just to you know, kill several hundred on a destroyer. And of course the Pokemon Voltorb, which is infamous for its self-destruct attack because it's a living bomb and blows itself up, you realize it was taken from the Japanese biplane kamikaze strikes. And this is a moment that I had it's a very defining moment where I realized almost kind of like what I really wanted to do. This is in August of 2009. This is at the Mysterious Galaxy bookstore. The man on the left, that is Mike Towery, who is one of the founders of San Diego Comic-Con. He founded San Diego Comic-Con when he was 14 with the idea of does anyone else out there like comic books? And so he got together this idea with the gentleman on the right, whose name is Richard Alf, who ran a magazine store, and that's where they get their comics. And Mike and his friends said, you know, wouldn't it be cool if there were people out there who wanted to do who like comics besides us? What if we were to gather some people together? And Richard's like, yeah, that's a cool idea. And I got this idea. What if we were to get a little convention together for comic books? They called it California's Golden State Comic Convention. San Diego's nowhere to be seen in that because ultimately San Diego wasn't a readily known city at the time in 1969, but they gathered this convention and they asked the great comic artist Jack Kirby to be their guest of honor. And then they also recruited Ray Bradbury as their science fiction guest of honor. They gathered this convention together and they founded Comic-Con. And so that was celebrating the 40th anniversary of San Diego Comic-Con back in 2009. And I'm there listening to them because it's my first time foraying into the world of comic history. I wanted to be a comic historian, so I wanted to meet these people and go there. And then in the background, there's this gentleman, the guy in the glasses, a guy named Batten Lash, who taught me everything he knew about style. He said, fashion is what is offered you, style is what you choose from it. But he was a great guy. And ultimately, Richard Alphys passed away a number of years ago, but they went on to start a smaller event called San Diego Comic Fest. And then Mike recently retired from Comic Fest and passed me the reins, and he appointed me as chairman in 2017. I'm like, it's kind of a really intimidating thing to help oversee a convention, but he said it really paid off because you brought a great vision to it and you love doing what you do. And you can't really go to comic cons without dressing up. You can fight it for a while, but eventually you do a costume. And eventually I started doing some costumes, and there was this event going around called Club Cosplay. And it's a nightclub where everyone dressed up in costumes, and I would always throw together the dorkiest costume the night before. And for me, there was this idea by Ivan, the manager of Club Cosplay. We want to get Weird Al to perform at Club Cosplay. Matt, do you think you could do a Weird Al costume? And eventually, my Weird Al costume became such a big hit, and it was considered scary accurate, and they would let me perform on stage. And we sold out the House of Blues. They loved it. And it got to the point where even Al himself called it scary accurate when he saw it. Is anyone going to check out that new Weird Al movie that debuts tonight? I think I am. Best of luck to Al and Daniel Radcliffe. I think that Matt Dunford could have been a shoo-in for the part, but just saying. And this over here, as we talk about stuff I do in my day job, this is Spider-Man Metal, a card set that came out last month that I was proud to work on. And here I am. I kind of just look at it. I finally did it. It came out, and it didn't really hit me until I held a pack in my hands. I made an actual licensed product with Spider-Man on it. And I even got to put Spider-Man hologram. There's even Spider-Man hologram cards in the end. And it's wonderful. And of course, I worked on the Spider-Man Into the Spider-Verse set, which is, finally debuting now with actor autographs and great holofoil cards. And I really enjoyed working on this stuff. And it's like, this is stuff I get to bring to life now. It's so cool. And I mean, that's just, a little smidgen here and there, as you can tell. So in answer to your question, because you'll notice you'll ask me a question and then I'll go off on multiple tangents over and over again. Who is Matt Dunford? He's a giant dork.
So is it Sari?
Oh yeah, Sari the beginning. I worked for a video game company called Critivo, and I did work doing the localization, storytelling, and helping to create video games to bring to life. So I don't really know anything about programming, but I could help with the storytelling along the way. And Sari the beginning was a really fun game. Its release has been delayed a little bit over the years because the developers are in Ukraine, and so they're going so. But next year it'll come out. And then the Universe Sim is another game we did at Critivo, and that was really fun. It's just sort of like the God simulator game. It's like what SimCity would have evolved into had it kept going in that direction. It kind of went over to the Sims these days, and everyone got to the Sims. But building your own world is the fun thing there, and so I really like it. And that is Adrian Perez. He's a student who is a student at Little Fish Comic Book Studio. I met him when he was about 13 or 12. He's a senior in college now. How do they grow up so fast? And he's also on the spectrum. He is very much on this. He's very much into comics and building his own world and building his own stories. And this was a night that actually happened 7 years ago to this day when I brought him up to meet his hero, the great Grant Morrison, the author of comics such as Animal Man and All-Star Superman, one of the most established artists. He came over to the United States in the early 1990s alongside authors like Alan Moore, Neil Gaiman, and just started the British invasion of comics. And Morrison adores Adrian because he's never seen a fan so young, so knowledgeable about these stories. And Adrian will just name off everything about the DC universe and going through in his own way. And we really got some style there at the late great Meltdown Comics in Hollywood. That was a wild night. 7 years ago to the day.
Would you say this is your ultimate job? Have you already reached the pinnacle? Are you looking forward to doing a lot more?
I am nowhere near where I want to be yet. I've done a lot, but there's still so much more I want to do. Because what I do is, I'm not afraid to start at the bottom. I'm not afraid to start in a grunt position. At Comic Fest, I was a volunteer, worked my way up to chairman. At World War Wings was a grunt, worked my way up to the senior marketing coordinator. And I'm ultimately, I think myself in a grunt position at Upper Deck. I would like to keep going further. I would like to keep doing more things. And ultimately, like the comic collector in me, I'm never satisfied. I just want more and more. I want to create. I want to do more. I want to see more. And I never have a dull day. And ultimately, I wish I did have dull days again, where I could just sit back and do nothing. And, I don't really make a whole lot of time for reading comics anymore, because it got to a point where my own life became more interesting than the stories I was reading. And it's kind of a bummer, because I miss comics.
Thank you very much.
Yeah.
Next, we're going to talk about the Miracle Project with Elaine Hall. And basically wanted to start it with 7 questions. What was your vision for the Miracle Project?
Well, I have to say something about Matt, because I'm so blown away, is that the idea of you following your passion and your family really following your lead, so that as a small child, what you are doing now was so encouraged.
Oh yeah. I mean, it wasn't so easy. I mean, I was an anthropology major, so I had the odds stacked against me. And the only job I could get back when I graduated college in 2010 was a job working as a chauffeur. And I did that for 3 years and it sucked.
Okay. But I just to say that, no, thank you. But
Yeah.
the whole idea of following our children's lead and knowing that they know intrinsically what's right for them. There's a mystical concept that everyone before they're born, they're missing on this planet. And when they come into being, they've been found. So I truly believe that we come here with a mission and a purpose, and that our children, our job as parents is to learn and listen from them. So it's not a direct answer of how the Miracle Project started, but you'll see from these guys and from my story how that evolved. I was working in TV and film, music, dance, drama specialist, and an acting coach for TV and film and writer. And I wanted a child of my own. I adopted my son. And when he came to me, he spun around in circles and stared at his hand and banged things. And being kind of a creative type person, I just did that with him. And when I did, we connected, we bonded. If he would flap, everybody flap with me, it's fun. And I just learned that flapping is what's done in Qigong, of getting out energy. And so when my son was diagnosed with autism, I mean, I would just flap with him and we'd be birds and we fly and we play. But the traditional therapies at the time were making him put them down and then rewarding him with M&Ms. And it made him tremendously anxious, which then made me tremendously anxious. So I started learning everything I could about autism. Dr. Stanley Greenspan, Dr. Barry Prezant, just learning as much as I could. I didn't know about Autism Tree. Oh my gosh, you guys are amazing. And I created a methodology to teach my creative friends how to follow my son's lead and how to join him. And slowly, by following his lead and learning all about all the incredible things that you do here at Autism Tree, but having it done in a creative way, my son merged into our world, still autistic, still non-speaking, but happy and connected, using multimodality communication, telling stories through movement, beating on drums for sound. And people asked me to create a theater program using what I had created for my son for others. And I created what's now known as the Miracle Project. And it's been the subject of an HBO 2-time Emmy Award winning documentary, Autism the Musical. We've presented at the United Nations and all over the world. And a lot of those kids that had passions, like, you shared, had passions as young, very small children to express themselves, are now in TV and film. Now, my son is non-speaking, and this is a creative musical theater program. People didn't think that non-speakers, actually, I'm using the wrong term, multimodality communicators and typers were not able to be part of the creative arts. And I didn't know any better. I'm neurodivergent myself. I'm a creative. So my son, naturally, the way I would reach him is through creativity. And he loves the camera. He loves being in front of people. He loves acting, and he types to communicate. I fortunately connected with Darlene Hansen, and she brought out my son's typing voice. Cut to number of years later, I wanted to have more typers in the Miracle Project, and most people did not think that typers could be part of this theater program, but I knew they could because of my own son. So when she introduced me to these incredible, you'll meet them, and I will not be talking because their words are so profound. When she introduced me to them, I had the opportunity of incorporating at the Miracle Project. We write original musicals and movies and films. And one of my first experiences with Otto is we did a show called Journey to Namu, which is human backwards. That's a whole nother thing. But we needed some music, kind of meditative music. And Otto composed the music, which was just extraordinary. And I said, like, we've got to do more of this. So we wrote a grant to Ability Central to create the first music movement creative program like this for typers and multimodality communicators. And these guys came to class with ideas, with they're artists, they're creators. And you'll see some of their work was all created with them. It was created, the concept of our video, the tone, the sound of the music, the rhythm, they wrote the lyrics, and we've defied the odds of what, Dr. Levy, I thank you so much for the presentation about what happens in adolescence. I remember when my son went through adolescence, I thought, someone came to earth and stole my child away, and because adolescence is hard for everyone. And when you have a son or a child on the spectrum, it's like putting Miracle-Gro on the hormonal whatever. But the arts were what helped him and saved him, just like it does others. So these incredible young people will share with you about participating, writing, creating, giving us the entire concept, a wardrobe, location, was all through their ideas. And we followed their lead to create a music video.
Can you tell me about the video just briefly?
Sure. Well, one of the things that I've always wanted is the best for our kids. So often our kids are given the smallest classrooms, and they, if they're going to do a play, then it's in like a little teeny room with no one watching or everyone's making noise or whatever. So we wanted to create a music video, and we've got state-of-the-art equipment. It was a full day shoot. We had call sheets. We had everything that would be done on a professional level production. And we were making transitions. We were had to wait a lot, just like on a regular shoot. We had to wait a lot. All the things that people say we can't do, our individuals on the spectrum can't do. Wear costumes and ties and anyway, we completely created an original video with a full production schedule, full production. And the concept came from these guys, which is most people think that individuals who may not use their mouth and tongue and to talk are a preschool level in their intelligence. But once they can type and share their thoughts and feelings, they are rock stars. They're like, we call it like them in black, they're higher evolved and elevated than not only people may think, but than most individuals. So that's the concept. And you guys correct me if you want to say it. I'm sure you can say it
Sure thing.
much better. So
So I'm going to show snippets of the video and integrate it with some questions to those involved with the video, just to get a sense of what it means to them, what the impact was. And then we'll move into some of the questions that I wanted answered about autonomy, the contribution of this to autonomy, and we'll just move on from there. So this is Bella and the green screen. So Bella, could you tell us about the Miracle Project's class and how it began?
My name is Bella. I am currently a high school student working towards my diploma. My goal after high school is to continue on to pursuing higher education. I'd love to study educational policy and continue my advocacy for access and inclusion. I have dyspraxia, a condition that makes everything I do difficult. I hope to change the narrative that autistics cannot access an education. This experience has been life-changing for me because I never considered myself capable of being a creator, nor a visual and performing artist. Perhaps because I never had the opportunity to participate in this art media, much less see someone like myself on screen. Thankfully, Elaine and her Miracle Project opened the door for us. The Miracle Project came into my life at a time when I was experiencing lots of insecurity due to my school situation. The focus for them was my weaknesses. I felt like my abilities were few. My creativity at that time was limited to writing poetry on occasion. On top of my doubting mind, I had another plaguing issue to deal with, my uncooperative body. I questioned whether dyspraxia could weed me out of trying. The process of moving on command was, and still is, challenging, but not impossible. In the video clip you can see what it takes to support my movement. Motor modeling and encouraging words made a huge difference. The weekly Express Yourself class helped me overcome my fears. I started seeing my abilities, not my deficits. I learned in the process of actively moving and creating that there is lots I can do. So in a nutshell, I was weary at first, but I have come to realize that I'm doing something that really makes me happy, and my input and participation is needed. You get knocked down enough that eventually hiding isn't an option anymore. This is what the Express Yourself class has done for me. It has made me love to shine.
What were others? What do you think other people's initial assumptions were about that? And what did you disprove, discover, or make known about your capabilities?
I proved myself wrong. I made a video. That was something I thought was never a possibility for me. The song and video tell our experience of being presumed incompetent. We had to make our message known so that teachers stop depriving students with complex communication challenges of an education. Elaborate. I am making it known that my challenges don't limit me.
Why is artistic expression essential and important for individuals on the spectrum, especially those requiring multimodality communication?
Meaningful experiences are motivating and essential to all of us. I had no experience with this type of artistic expression before. Now I can share the value of seeing myself as a creative human being despite my differences. I think multimodality communicators have been ignored, overlooked, and not encouraged to seek artistic expression. We are no different than you. We can and do want to participate in all that life has to offer. I believe in my creative abilities and can see a future in songwriting and production. I needed the Express Yourself class to change my trajectory. It really has impacted my self-esteem in a profound way. I am grateful.
For all of us, what are your hopes that this video and others like it will do for our community?
Having more artistic expression opportunities, that is my hope. Our community is very much making waves for access. I hope to add momentum to our cause. A door was opened, but making doors is our expertise. Mod Squad is unstoppable. Believe us.
This is William, also in front of the green screen. So William, also tell us about the Miracle Project's class and how it began.
My name is William Del Rosario, and I am a 22-year-old Hispanic Filipino male with black hair, brown eyes, and my pronouns are he and him. I have autism, apraxia, am a multimodal communicator, and use AAC. Thank you for making the time to be here today.
What were other initial
There is
assumptions?
so much more to this. Most people might think this looks like the next viral video with a dancing dad. What they don't see is the perfect example of apraxia and sensory motor differences. My brain and body have a hard time working together, especially in new environments. My motor is being coached in a way that helps me get my body organized. Also notice that I am focused on my dad. More than a communication partner, I rely on my support to be my communication and regulation partner in each changing situation. No one is saying that I am unable to do the dancing or movements. Instead, they are saying, how can we support you, and giving me the time that I need. Big difference when you change your mind to see the possibilities instead of capacities.
Why is this expression important?
I liked being a part of the Express Yourself program because knowing how my body every week was becoming more informed with the ability to act was awesome. The chance to see myself on the big screen says that we are all able to reach for our dreams when we are given the time to become the person we are capable of instead of the one everyone thinks we should be.
Thank you, Willem. This was them celebrating finishing the video. So Otto, same question.
And Autism Tree for the platform. I am Otralana. I am 18 years old. I live in sunny Southern California. My pronouns are he and him. I am a Latin male with lightly tanned complexion and a smattering of sun-kissed freckles on my face. I have short brown hair. I wear my hair gelled back. I have blue eyes and a bright, inviting, irresistible smile. I am a high school senior. I am a motivational speaker. I am an advocate. I am the reigning king of inclusion in my kingdom, Otter Nation. I am an entrepreneur and have a thriving e-commerce business. I am an intern at Kindred Communication as well. I want to plug this business real quick. www.gokindred.com. Without Darlene Hanson, the speech therapist, I would not have this life or this current level of success. She taught me to type. This is paramount for your listeners and their families to understand. Without letters, without words, I would not have this wonderful life. Like the royalty that I am, I was born this way. I was born neurodiverse. This community, our community, needs outspoken leaders like me. That is my connection. That is why I picked this clip of the alley. It shows how we are so many layers. It takes a village. In front of the camera and behind the camera. Here is some crazy facts for you. 1 in 4 people are disabled. That is 25%. And there are less than 1% of disabled people represented in front of or behind the screen. Making a video like this is groundbreaking. We are jackhammers. We cannot wait for people to invite us. We are building our own possibilities. Positive images speak volumes. I want acceptance. We should be well aware technology exists. No one under the age of 30 leaves a voicemail. We all text. Let's be real. My mission in this medium is to show all the ways we are integrated into society. It is the A, B, C of A, C. A always. B, B. C communicating. Opportunities for communication are everywhere. You do not need a checklist or a data sheet for authentic communication. Be spontaneous. The Express Yourself class at the Miracle Project has given us a platform, and we are running with it. Experiences like this are perfect for forging friendships. The examples of our collaboration to make this video come to life are much richer than the IAP, your turn, my turn friendship goals. We were presumed competent. We were given the creative space to thrive. It began and ended with communication and support. This exemplifies our interdependence. People focus on autonomy and independence. But I want to make interdependence the focus. We are all connected. And that is why I chose the clip of me typing in the alley and having a conversation with the director, Shelley Pack. It is shameless product placement. This is Hollywood, baby. I am always repping. How am I going to be financially independent if I am not figuring out ways to sling my merch? I also want to touch on adult relationships too. It starts with friendships, but also a paradigm shift. That was the main point of our video. We are so often infantilized. Think about it. There is no human sexuality course in the special education curriculum. I think it will be up to the Mod Squad to tackle this topic too. Elaine, what do you think about the Express Yourself class doing a mashup of Marvin Gaye's song, Let's Get It On, with the A.C.
I love the idea, Otto. Only you could get the licensing. So I think we could write our own song.
I'm on it.
They're very fluid. I'm trying to load the video.
While Otto's typing it, I want to add that, oh, great, that also the team here helped raise the funds to produce the video. So, talk about entrepreneurial.
Let's hit the drum. You ready? Your turn. Here you go. Your turn. Ready? Ready, everyone? Let's do it.
Not just a thing, but it's mine. Open your ears, open your mind. That happens all the time. My voice matters like the sun. Receive our dream and feel the beat. Let the world show, 'cause I want you to know I'm a master. Let the world show, 'cause I'm full of nerves and harmony. Let my time and let you see. I type my thoughts and I feel just like you. Typing's how I roll, it's true. You do in fact understand the puns that we be dropping. We're free to think and wanna be heard too. My voice matters, please listen. Receive our dreams and feel the beat. Oh, let the world show, 'cause I want you to know I'm not free. Let the world show, 'cause I'm full of nerves and irony. Great minds are poets and live the words please. Let my talk and let you see.
So these are some additional questions I just wanted to ask, and we'll just hit a couple of them because we're running out of time, or run out of time, which is my fault. Would anybody like to talk about autonomy? I had talked about autonomy before. Anybody with any considerations about autonomy?
Autonomy is always going to be one of the difficult things because I, have a situation where people always say it's like, oh, you're like everything that you've done is because you have, because your parents are well off and they gave you a good life. And I'm like, so you're saying I can't do anything on my own? Well, they're probably right, but no. Yeah. And it's like, I wish, I could be one of those people that says my parents are jerks and just, look at them in the eye as they, or sit there in the fifth row and whatnot, but I can't. But, I have had supportive parents. And so the whole thing is, it's kind of hard to leave autonomy when they are, caring of you, but then eventually, there's only so long where you can, be a college graduate sitting on your mom's couch in your underwear before like your parents say, get a job. Okay, I guess I got to do this. But eventually you want to do things on your own. And I think it's like, because you can't really be defined by parents. My dad has always been supportive of my love of Spider-Man and Pokemon and Lego Pirates and all that stuff. Not always understanding. So it's like, I got to do my own thing. He's like, hey, Dad, can we do this? It's like, my dad typically understands he doesn't do anything fictional. In fact, every time I try to get it, there's only one show where my dad has really gotten into, which was the medical drama House, Dr. House, also on the autism spectrum. They did an episode of that. But, yeah. And it was a great thing of that. But the thing is, oh, cool, medical drama. My dad solves the problem in 10 seconds because that's how doctors are every single time.
So
But
Matt, I love the idea of what you're saying with your parents, and Otto had expressed that earlier on too. So thank you. Where Otto, where you were talking about rather than focusing on autonomy, focusing more on you stated to focus on interdependence. Yeah.
Can I share my thoughts on Bella and the clips of William? Yes, I have more to add. To be honest, I expected nothing more than another opportunity to Zoom with friends. I had no idea we could collaborate on this level. And now Elaine has unleashed the beast. The genie's out of the bottle. There is no going back. It was empowering. The Miracle Project threw rocket fuel on our creative spark. And now we are soaring. I see myself working in many areas of production. I enjoyed the pre-production, creation and planning. I most definitely enjoyed production day. The camera loves me. And of course, post-production, promotion of the work. I have a gift. People want to engage with me. Promoting this film has been exciting. I'm even exploring the finance side of production. William, Bella, and I have applied for a grant to fund our next project. Great art takes fat racks of cash.
Does anybody
We need
want
to
to
move past us and them, ours and theirs? We need to think in terms of we. We need to move away from autism spectrum disorder, ASD, versus autism spectrum symptomless, ASS. Are you saying those without a diagnosis of autism or autism spectrum disorder, and now known as ASs? A little comedy for the masses. But seriously, we need representation as leading roles. And we need access to meaningful careers. We do not need viral videos of the water boy who finally gets to play on the court or on the field. We do not need to star in this type of pity porn. I want to thank everyone for the open hearts and minds. Let's create a beautiful world together. Hopefully, next time you happen upon an AC user, you will think that person has some greatness to share. Let's have a listen.
Thank you. I very much appreciate it. Are there any questions that anybody may have for the panel?
I don't have a question, but
hi to Adeline's mom, because I remember you when we were in my preschool class up here at the district. And I'm very proud of you and I'm happy for your success.
Any other questions? All right, thank you.
I don't have a response to,
See, I have a funny year.
Oh, I got the shirt. Exactly, you're wearing the merch.
Thanks.
And just saying, we exist. It's oftentimes hard for us to reach out. We appreciate when you reach out to us. It's really nice because, new situations for people on the spectrum are always going to be intimidating, but it's something where we can develop comfort. Did you know that once upon a time, my greatest fear was public speaking? Yeah, just like 98% of the population. Now I actually really enjoy it. The truth is, I'm actually more comfortable talking to a crowd than I am to an individual person because there's a wide range of things that an audience can say. But it's not as wide as what an individual person can say. So it is very comfortable. And I thank you so much for this opportunity here. It's hard for us to break out of, those repetitive things that we do. Little things are always going to be, the whole difficult thing, and venturing out, we are not very good with eye contact. It activates the internal threat sensor in the brain, and basically, oh, this person wants to fight me. This person wants to fight me. So if you see me wearing 100 different pairs of sunglasses all the time, that's the reason why I've always got my eyes covered. And that's just a little bit of understanding for us and what we do. And I know that I have it different than others on the spectrum, and my panelists up here with me, I've learned so much from them, and but they've shown that they're geniuses too. You've heard them talk. You've heard them express themselves. You may not have heard them talk, but you've heard the words that they have expressed through typing, and that there's different ways to get your message across. And as long as you're getting your message out there, I think that's the most beautiful thing.
Thank you.
Yeah. Mm-hmm. Yes.
Great. Thank you, Michael. Thank you for the panel. That was wonderful. I believe that there is a movie to be shown. Yeah.
Hi, I'm Dana Hoff. I'm the executive director and founder of Autism Tree, and I just want to say I feel extraordinarily grateful that we've been working together, and thank you, Dr. Levy, for this incredible panel. I have to say it's probably one of the top favorite things I've been involved with in the last 20 years is this panel, and just this evolution that we've been working on this neuroscience conference for 10 years. And I'm extraordinarily grateful to the leadership with Dr. Alison Motri, our chairman, and Roger Bingham, our moderator. And I want to thank, of course, our platinum sponsor that made it possible, and they had to leave a little bit early, but the Epstein Family Foundation and the Julie Bronstein family. We just couldn't do it without support. And I hope that in the next 20 years, we'll even go bigger. We have a quick video, and I want to thank our board. Who is here from our board that could stand up, please, or even come up here with me? Our board of directors. Are there any board of directors here? Come on up. Yes, Dr. Michael Levy's one of our co-founders. Come on up. Alexandra Tacone and Linda Townsend. Hi. We worked really hard on the 4 pillars. And this is a video that I want them to see too. And it really captures the heartbeat of Autism Tree these last 20 years. EC. What do? We embrace every family. we connect every family, we empower every family, and we help them create the lives. And right here, I don't think there's a better example. Everyone on this panel is exactly what we're striving for. And yet they're taking us places. I always like to say Otto is taking us places we've never been before. He's like an eagle flying over, taking Autism Tree to new heights. So please enjoy this video with me. And thank you guys so much for being here and for being part of it. This is how we're going to end.
We could coin the term autopilot.
I love that, Matt.
Yeah.
Okay. Thank you, everybody. We're going to end on this video.
Autism Tree has been serving one family at a time for 20 years. And now we have focused our process into 4 core values: embrace, connect, empower, and create. And we embrace every family with love and acceptance. Before Autism Tree, we felt lost. We kind of felt on an island of our own, and we didn't have anyone that really understood us. Whenever we've gone to events, he feels embraced. He feels like he's at home, and his guard goes down and he's himself. We connect them to a community of resources.
I can remember how confusing it can feel and overwhelming. And to be able to say to a parent, you know, I've been there, you're not alone, and help a parent see the value of who their kid is beyond the word autism.
We empower them to create the best version of a future for their child.
Music really does help me to express myself. Felt empowered when I was a liaison in the San Diego Symphony.
And create organic relationships, meaningful opportunities, and lasting friendships.
Part of my love and excitement over, Autism Tree is, you create this massive umbrella where all these like-minded people, all these wonderful kids who all kind of have a similar but different approach, and give them an opportunity to, explore and enjoy.
The idea of the navigating neurodiversity is exactly for those families that are in crisis position, is to have someone say and help prioritize and say, to help your child, do this first, because this will unlock these resources.
And we connect them with ABA providers, mental health providers, family counseling that might help them as a family. We also look at their IEP and the services that they're getting from the school to ensure that they're adequate. IHSS that helps families who a parent has to stay home and give up their career.
Our goal is to go big and with your help, serve 200 families who deserve to create a brighter future for their child with autism.
So I'll thank the TPF one more time, and I think we're officially done. So through the day, we had a journey from all the spectrum, and I think we can unify. And as Otto said, for a better word, thank you.